Dana?
Yes. Good morning, everyone. I welcome you all to the second Pharma Summit Drug Discovery and Community Trial on 4th and 5th December 2023, a hybrid conference organized by Association of Pharmaceutical Research. Association of Pharmaceutical Research is one of the world's leading professional association for pharmaceutical and life science professional caring for people. APR is a non-profit professional association meant for research and development in the field of pharmaceutical science and technology. APR is an international forum for researchers, academicians, doctors, and participants for sharing knowledge and innovation in the field of pharmaceutical science and technology. APR aims to bring together worldwide researchers and professionals, encourage intellectual development, and providing opportunities for networking and collaboration. APR meets with its objectives through academic networking, meetings, conference, projects, research publication, academic awards, and scholarships. APR strives to enrich form its diverse group of advisory members. APR connect pharmaceutical professionals, research, exchange global innovations, and act as a bridge between researchers and academicians. By organizing international conference, world conference, research, and economics by organizing international conference, world conference, faculty development programs, international workshops, seminars, lecturers, short-term training programs, providing membership, establishing chapters, faculty exchange program, and implant training. About BioLeaks. BioLeaks is a not-for-profit professional association which prominently promotes research and development. We at BioLeaks have brought a revolution in the field of worldwide conference. BioLeaks conference bring together the professional wizards and leaders who have explored all avenues to reinforce the field of life science and medicine technology. Help in enhancing the skills of the people from diverse industries and form a common platform for eminent personalities, physicians, researchers, doctors, and academicians, professional business figures, and much more. Association of Pharmaceutical Research organized the second Pharma Summit, Drug Discovery and Community Trial on 4th and 5th December 2023. This conference aims to bring together leading academic scientists, researchers, and scholars to exchange and share their works and research result on all aspects of pharmacology, pharmaceutical biotechnology, pharmacovigilance, drug delivery, biodrugs, and pharmacovigilance, and drug safety, pharmaceutical microbiology, pharmaceutical research and development, pharmaceutical analysis and quality curatives. The drug discovery industry has become such a competitive market that it continually faces a challenge to bring better drug discovery technologies. This industry must discover and develop innovative drugs for a wide range of diseases in a marketplace that is likely to experience growing regularity challenges, pricing pressures, and various other bottlenecks. Pharma Summit 2023 will be welcoming brainstorming research papers from across the world in the field of pharmaceutical science and drug discovery. Now, I would like to welcome our eminent keynote speakers of the day 2. I would like to welcome our eminent keynote speaker, Mr. Girish Nilhani, Research Fellow, Day2 Pharmaceutical Incorporated, USA. Associate Professor Dr. Sadia Sultan, Head of Biotransformation Research Group, Research, Nexus, Health and Wellness, Faculty of Pharmacy, Development of Pharmacology and Pharmaceutical Chemistry, University Technology, Mara, Malaysia. Professor Murali Ellappu, Associated Professor at the University of Texas, Rio, Green Valley, Sioux Falls, South Dakota, United States. Now I would like to welcome our section chair, Dr. Padma M. Parhak, Principal, Department of Pharmacology, the Oxford College of Pharmacy, India. Dr. A.L. Akilandeshwari, Associate Professor, Chettinad School of Pharmaceutical Science, India. Dr. P. Muralidharan, Professor, Department of Pharmacology, Chettinad School of Pharmaceutical Science, India. Dr. Devi T. Professor, Department of Pharmaceutical Chemistry and Analysis, Chettinad School of Pharmaceutical Science, India. Thank you, everyone. Now I would like to call our keynote speaker, Mr. Girish Nihalahani, to give a keynote address. Mr. Girish Nihalani is a research fellow of drug product development at Day2 Pharmaceutical Incorporated based at Florida, USA. Mr. Nihalani earned his master's degree in pharmaceutical science in 2025 from Bombay College of Pharmacy, Mumbai University. Thereafter, has dedicated his professional career in formulation development of complex modified realize solid organ dosage forms. He has specific interest in emphasized dispersions of matrix-based system. Over years, Mr. Nihalani has developed and registered more than 15 complex products as ANGA, including first to file, first to market, and NCE first products, many of which have been FDA approved as generics for US market. He has received a special recognition awards by the employees for his expertise. Mr. Nihalani has held professional position of increasing promises for globally renowned companies such as Inventia Healthcare Limited, FTC Limited, Mr. Reddy Laboratories, Silfria Incorporated, Watson Incorporated, and Teva Pharmaceuticals Incorporated. In his current role, he continues to lead develop of drug products requiring expertise in formulation development and biopharmaceuticals. Being working in the private industry, publication of his research work is limited due to IP constraints. Mr. Nihalani, to his name, has about 9+ publications, including 3 patent applications, and his works has also been selected by CRS and AAPS for poster presentation. Let's welcome Mr. Girish Nihalani.
Good morning, everyone. I welcome you all to the second Pharma Summit organized by Association of Pharmaceutical Research, and we must appreciate the efforts by the whole team of Association of Pharmaceutical Research in bringing together the students, scientists, and professionals from the industry to share their knowledge and to benefit from each other. Let me share my screen. I'm unable to share my screen. Is someone else also sharing the screen? Because the message says I cannot. Yeah, no, maybe.
So you can share now.
Okay, thank you. Is my screen visible?
Yes, sir.
Okay.
Yes.
So little introduction about me. I have done my master's in pharmaceutical sciences in 2005 from Bombay College of Pharmacy and have over 18 years of experience in development of solid oral dosage forms, specifically the modified release dosage forms, complex generics or 505(b)(2). Currently, I work as a research fellow of product development at Teva Pharmaceuticals based at Florida, USA. My today's keynote speaker session, the outline of the is like we will quickly glance over the ANDA drug product approval process, which many of you might be already familiar with the main emphasis of what are the possible reasons of refuse to receive an application by an FDA, and then we'll touch base with what are some of the good practices and key product specific guidances one should keep in mind in order to avoid refuse to receive scenario. And then we brief about one of the technology which is recently being employed by across the industry, which is hot mill extrusion as a technology to form amorphous dispersions. So for a generic drug application, it is being reviewed by different departments at FDA. It's CMC department, chemistry, manufacturing and control, labeling department, bioequivalence department, and if deemed to be satisfactory in terms of the meeting the requirements or the checklist, it's easy go to approval. But in the last decade, since last decade, the requirements have becoming more and more stringent, and the industries are struggling in terms of the first cycle approval. So if the FDA is not satisfied with the application, they issue so-called deficiency letter or refuse to receive. And where refuse to receive is important. If you see the right bottom of the slide, if there is a refuse to receive, basically it's a loss to the ANDA, it's loss of the fees, and more than the fees or money to the industry, it is the loss of the time, and which further triggers the delay in the launch. So in this slide, I have listed down like some of the common reasons which lead to the refuse to receive an ANDA by FDA. In practicality, the reasons could be more than these, but I have narrowed down to the high probable ones. The first most is failure to meet the IID limits. IID stands for Inactive Ingredients Database, which is listed by the FDA, where it basically lists the amounts in terms of milligram of an excipients which we use in a formulation. So in normal practice, when we develop a formulation in order to meet the desired target release profile, some of the times one tends to overlook these limits and cross these limits for simple excipients like mannitol. And what happens for some of the products, the recommendation is the pediatric population. Now, what happens in terms of pediatric based on the body surface area, one has to calculate the dose in and reversely has to calculate the how much excipients will be safe for a pediatric population. So some of the occasionally it is being overlooked because the database which is maintained by the FDA, it gives the limits for a adult population. For a generic drug application, FDA also requires to submit the minimum of three batches to be evaluated with a minimum of 2 lots of API. And sometimes we tend to use more than one intermediates in a batch, and there is a tendency of mixing up of the API lots. For in terms of the packaging, FDA requires minimum 100K 1 lakh units to be packaged in the final configuration proposed to be marketed, unless and until the proposed commercial batch size is less than 100K. And uh, in FDA also has a database as a guidance for the dissolution of a specific product. Despite of that, FDA may not buy the listed method, and they ask for demonstration of the dissolution method to be discriminatory. That is, the dissolution method has to be sensitive enough to detect any formulation or process changes. So that requires a lot of efforts in terms of developing a dissolution method in-house. And some of the other reasons are like impurities, and another reason which is overlooked is the formatting or font size or legibility of the matter. Occasionally, in order to accommodate the very complex manufacturing flow charts in one page, one tend to do the font size like 9, 8, or 7, or but not accepted anymore. If anything is not legible, FDA has all right not to receive the ANDA or refuse to receive the ANDA. Now, recently FDA has come up with a very, very good tools in terms of like rather than submitting the ANDA, one has option to do a control correspondence to utilize the tool which FDA offers, which is basically a mode of communicating with FDA seeking clarity on any matter or on a complex product where the product specific guidance doesn't exist. And depending on the level of control correspondence, whether it is level 1 or level 2, the typical response times from FDAs are either 60 days or 120 days. So that saves a lot of time or can help in avoiding the refuse to receive ANDA by FDA. So that's a great tool of like FDA has a separate guidance on that, like how to communicate with FDA and through control correspondence. Uh, Quickly going through some of the good practices or considerations when we do generic drug product development, specifically for modified release. We the goal is not only to match the dissolution profile to that of the reference drug, but to also demonstrate the alcohol dose dumping studies, which are basically conducted by using 5%, 20%, and 40% of alcohol in the dissolution medium uh, to, to, to demonstrate the real life conditions, like if a patient consumes a drug along with the alcohol, it doesn't lead to the overdose. And FD also expects to include the whole time studies as a part of in um, determining the shelf life of a product. Like hold times are basically how long we can hold an intermediate in a production setting before proceeding to the next stage. And before uh, before I think there is some noise in the background. If someone can mute. So one more point very getting more attention in the last, I would say, 5 years or so, is the stratified content uniformity. In the past, what used to happen was like FDA used to accept the content uniformity as a part of the finished product specifications, but not anymore. Nowadays, FDA requires content uniformity to be demonstrated throughout the whole batch, like throughout the whole compression run or encapsulation run. We collect the samples, like say from as good as 40 locations into the content uniformity. So the reason it is very important is at first place is like based on the dose of the API, one has to select the manufacturing process which should be robust enough from the launch perspective. And for some of the other products like chewable, there are product specific guidance. We will glance one of the guidance on chewable tablets with respect to the hardness. And so again, in the early stages of development, we do compatibility studies to rule out incompatible excipients from the drug. We do a quick open exposure studies by submitting to the
Google Form name and mail ID they might have given over there. We have to.
and one of the requirement for a generic drug application acceptance criteria is it has to be pharmaceutical equivalence, like it need to have the same strength, same route of administration, same API form. And the for tablets for dosage forms like Scott tablets, FDA requires the dissolution to be dissolution and content uniformity to be conducted on the splitted tablets and to be similar to the whole tablets. That is also one of the important criteria in selection of the right tooling, the design of the score on the tooling. And in case of the product with multiple strengths, it is also important to have the strategy in place for the biwaiver of the lower strengths, whether the formulation has to be dose proportional, dose similar, or any other justification in place. Most of the drugs which we handle nowadays are either BCS class 2 or BCS class 4 having low solubility. And I think the dissolution method selection we already covered. And the other in the recent times, the attention, the parameter is getting the attention is the elemental impurities and the nitrosamine risk assessment. Basically, FDA is also limiting the limit for each and every heavy metal to be present in the formulation, and the iron content to be not more than 5 milligram per day, as it is related to some side effects in kids and adults as well. And FDA also, after development, FDA is also insisting to demonstrate what the process controls we have in place, which will ensure a consistent quality attributes of the finished product with justification of the specifications. So these are like some of the aspects which good practices we talked about, which one must consider before finalizing the final formulation. I would just glance through a couple of guidances which are like as an example, like where if someone is like overlooks, it could lead to the refusal to receive. The one guidance is which is related to the size, shape, and physical attributes of the tablets and capsules. So if the reference product is less than 17 in any of its largest dimension, our test product is not permitted to be more than 20% larger in any single dimension and not more than 40% higher than the RLD in terms of volume. Basically, this guidance is in place in context of swallowability by a patient. The dosage form has to be easily swallowable by the patient. And if the RLD is more than 17 in any of the dimension, then the generic drug is not permitted to be larger than that. One another guidance is which I would like to brief is on the chewable tablets. So for the chewable tablets, it becomes a very challenging because uh, on occasions it involves shipping or transportation between the two different sites. And at the same time, based on the nature of the dosage form, which is chewable, which requires less hardness in order to be make it chewable, and transportation requires higher hardness to withstand the um, handling of it. So FDA has a guidance on the hardness of the chewable tablets, which irrespective of the dimension of the dosage form, the hardness is expected to be less than 12 kp. And if it is more than 12 kp, it could be still acceptable based on very strong justification, like the dissolution is rapid, disintegration is rapid, or there is the hardness drops after brief exposure to simulated saliva. Basically, we are simulating the conditions of how one would take the chewable tablets. I would like to touch base on hot melt extrusion as a technology to form amorphous to convert active into the amorphous. And why this is important is um, it is very important in terms of like developing a 505(b)(2) application in terms of formulating anti-cancer agents which have very high doses. So by means of converting the crystalline APIs to amorphous, the dose gets reduced, and the manufacturing process involves very less number of steps as compared to conventional solid oral doses for manufacturing. So as we can see in this, we might be familiar with this chart on the left bottom, the BCS classification of the drugs, biopharmaceutical classification of the drugs, 1, 2, 3, 4. Class 2 and 4 are having the low solubility. So majority of the new chemical entities coming the market, they are either BCS class 2 or BCS class 4, and hence to achieve the bioavailability of those compounds is becoming challenging, and that's why the hot melt extrusion is becoming more popular, because that is a technique to convert those crystalline materials into amorphous. So why crystalline? The new chemical entities which are crystalline are not favored because they are having very poor aqueous solubility, high melting point, steric hindrances in the structure, high molecular weight, high log P, and this is how hot melt extrusion looks like. If you see on the right hand side diagram on the hopper, basically a powder mixture of polymer and API is fed into the hopper, and then which goes through series of conveying screws and kneading screws when that in a barrel which is very heated based on the melting point of the drug, which converts the whole dry mix into a molten mass, and eventually the mass is extruded as a strands and cools down, and that is cut and further used either for encapsulation or tableting. So the basic concept is like solubilization of the API by way of melting it. And by way of melting it, what we are doing, we are loosening the structure of the API and converting it to the amorphous. This is how a design of a extruder looks like. If you see from extruder from left to right, there are different zones in the hot melt extruder. The heated zones, which are set based on the melting point of the API, melting point of the polymer used, and any other metals used such as plasticizer or surfactant. And in the initial zone, if you see, as the material is getting conveyed, the temperature is always less than the melting point of the polymer or API. Then as the material proceeds towards the right through the conveying and kneading zones, it gets exposed to the higher temperatures, gets melted between the screws. And so the green ones, the green screws resembles the kneading zones, and whereas the blue zones are the conveying zones. So along with the heat and conveying, it is also the high shear through the kneading zone screws, which basically are responsible for converting the API into the amorphous. This diagram illustrates a difference between forming amorphous dispersion by hot melt extrusion versus spray drying. On the top, from the layer above, represents by the extrusion isochronization. Sorry, my bad. Hot melt extrusion, and a row at the bottom represents making amorphous dispersion by spray drying. So in a spray drying, basically a solvent is used, and during the spray drying, the solvent gets evaporated and the mixture gets converted to amorphous dispersion. Whereas a hot melt extrusion is devoid of using devoid of any solvents. There is no use of solvents in the hot melt extrusion technique. So to convert the API, the drug, into the amorphous, we need to use some polymer, which is which has to be identified or selected based on the properties of the drug. There are multiple polymers available, and this involves selection of polymer, optimization of ratio of polymer to the drug, melting point of the drug, melting point of the polymer. Some of the common polymers used are like povidones, HPMC, AS, acetate succinate. So the extrusion is carried out typically about 10 to 20 degree higher above the glass transition temperature of the polymer. And in cases where the glass transition temperature of the polymers is very high, we make use of the plasticizers. Basically, plasticizers help in reducing the glass transition temperature of the polymers and facilitate carrying out of the process at lower temperatures practically possible. And typically we use plasticizers in the range of 5 to 30%, and they also help in reducing the viscosity of the polymer drug mixture, facilitating further to carry out the process easily. So when we looked at the main component of the hot melt extrusion is the screw. So the different parameters are uh, related to the screw design are as illustrated in this diagram is like external diameter, like outer diameter, internal diameter, and the difference between the outer and inner diameter basically makes the gap or the free volume available for the material to pass through. And based on the properties of the materials, one can design the hot melt extrusion to have uh, different pitch of the conveying elements or number of kneading zones. The bottom left is, if you look at the diagram, there is a high pitch, low pitch, and medium pitch. So high pitch is where the basically the feeding of the powder mixture occurs. And if you can see, as the pitch is high, that facilitates higher transfer rate of the powder mixture. And then down the line, it gets to the either low or medium pitch. And there are heated zones as we saw before. So hot melt extruders are also referred like in terms of their name, they are also referred by their outer diameter of the screw. And during the scale up consideration, it becomes very important to keep the ratio of the outer diameter and inner diameter to be same. And for the two the two screws could be of a different design. Like they could be either a co-rotating, meaning rotating in the same direction, or counter rotating, like both rotating in the reverse direction. And also one can design the two screws of the two barrels like with respect to angles to each other, like could be 30-degree or 90-degree, in order to develop the desired torque or shear within the hot melt mixture. So these are the examples of on the top, on the left hand side, if you see the bilobal versus trilobal design of the screws. As you can see, in the bilobal design, there is more free volume, and in the trilobal design, there is less free volume. So trilobal we typically use for the R&D use, where the batch size is limited. So we prefer to have a low free volume, whereas in a on a production scale, we tend to use the bilobal screw designs. Talking about setting up a process and setting up a process by using the hot melt extrusion is an art, because there is a combination of three variables here, three types of variables happening here. There are one variables related to the material properties, which includes the melting point, rheological properties, the molecular weight, the ratio of the excipient with respect to each other, and there are on other end there are process variables, which are some of them are like independent, some of them are like dependent, like screw speeds, the residence time, the pressure, the temperature, and the third aspect is design variables. Design variables, on most of the occasions, we get a good help from the manufacturer of the hot melt extruder itself. And the like converting the API, what my point is, like converting the API to amorphous is not of the end goal. On many of the occasions, what we see, even after the API gets converted to amorphous, on some of the occasions we see there is no drug release, because based on the structure of the polymer and the API, they interlock so well that there is no drug release. In those cases, we use combination of polymers, or we add surfactant or different plasticizer to the same mixture to facilitate the drug release. Whatever we were talking just now on the parameters affecting the hot melt extrusion process, some of the important parameters are the screw speed, which relates to the resistance time. If the resistance time is not sufficient, material may not be subjected to complete melting and shear. Temperatures, which are set based on the glass transition temperature of polymer and API. There is in the screw design, the ratio of outer diameter and inner diameter of the screws, the feed rate. I hope I was able to cover some basics or some key importance in a 30 minutes window, and it was useful to everyone. Any questions? I would be happy to answer.
Attenders, do you have any questions regarding the present? Kindly ask. Thank you for your informative session, sir.
My pleasure. Thank you for the invitation. Thank you.
Next, I would like to welcome our keynote speaker, Dr. Sadia Sultan, to give a keynote address. Dr. Sadia Sultan is an Associated Professor of Pharmaceutical Chemistry in the Department of Pharmacology and Chemistry at the Faculty of Pharmacy, University Technology, Mara, UITM, Malaysia. Currently, she is heading the Biotransformation Research Group under Research Nexus, Health and Wellness. 5 PhD and 6 master students have already graduated under the supervision. She is having more than 18 years of experience in the field of natural product research. Her area expertise includes biotransformation of exogenous substrate using microorganism and plant tissue culture techniques, exploring bioactive secondary metabolites from plant, soil, and marine endophytic fungi using dereplication and osmic approach, modern MMR spectroscopic approach in elucidation of secondary metabolites, biological screening using antimicrobial assays, mechanistic studies to understand metastasis breast cancer towards the effects of microbial of steroids and preference using MDA MB231 cells include xenograph model.
Hello, thanks a lot for your very kind introduction. May I share my screen?
Yes, ma'am.
But I cannot start a screen share while the other participant is sharing. I think, yeah, now.
Now you can share the.
Okay. So, is it okay? You can. Okay.
Yes, ma'am, it is visible.
Is it okay?
Yes, you can start.
Thanks a lot, the chairperson and the other organizing committee members for giving me a chance to present some of my work done by my PhD student at this moment for your great conference. So you have already my introductions. I just start from my research. So here I'm going to, because basically I'm working on biotransformation of exogenous substrate using steroids, terpenes, and beside this, I'm also working on isolation of secondary metabolites from endophytes. And from currently we have done isolated some endophytes from the novel endophytes from the Arctic and Antarctic zone. My student, she herself, she went there and she collected and isolated some endophytes. So one of, I think, I have present in my previous conference, but this is the current one. This one, she used the endophytes and she grew endophytes on different media, and the media is used as elicitor. And it works as an epigenetic regulators to make the silent genes or silent compounds. Okay. So, as we all know, fungi can produce secondary metabolites, and usually we observe in external and laboratory conditions. As compared to if we use different methods to use the unexpressed genes or the sleeping silent genes. There are some microbes present. Endophytes or secondary metabolites are present in the endophytes or microbes, but they are silent under the standard screening conditions. So here we use a silent biosynthetic pathway which lead to the novel bioactive compounds, and we use the method to activate the sleeping genes present in the microbes, and we awake them, the silent genes, and use overall expressing silent genes as well. So we start with the culture condition. We use different conditions. We modify the conditions, and we have our approach to activate the silent biosynthetic pathway in the fungi. We use genetic manipulation, and we use the epigenetic regulator as well, and addition of biotic, co-culture, and abiotic challenges as well. So the different ways we can do, we can manipulate the genetic by pathway, or we can use the epigenetic regulators, or we can add the biotic, means co-culture, and abiotic challenges as well. So for the current one, we are using epigenetics, the study of the heritable changes in genes expressions that occurs without changes in gene sequences. So we our approach to, without changing the gene sequencings, we are going to changes in the gene expression, how the gene can express in the different ways using some epigenetic regulators. So the epigenetic regulators we use here is DNA methyltransferase. This is DNMT inhibitors, and we also use histone deacetylase inhibitors as well. So there are two epigenetic regulators, DNA methyltransferase and histone deacetylase. So our problem statement, we have the discovery of bioactive metabolites, that require the minimization of the consumable items. So we want to, instead of using, if we applying different elicitors or epigenetic regulators, we can have a lot of mess, use a lot of consumables, chemicals. So we just try to minimize all these consumable items and human resources, so make it in a short and in a minimized way. So we develop a method, and we did the fermentation of fungi instead of individual flowers for screening purposes. For labor, it is labor intensive and it's costly. This is a typical fermentation method. We use our Maxus method. As compared to the typical fermentation method, we use microtetra plates, elicitors, combinations of solid phase extraction, and ultra high profile UHPLC and static at the end static analysis. So this Maxus protocol only tested on a single fungal strain, which is Aspergillus. And we have to send the protocol for culturing this non-sporulating fungi, because the spores formation fungi culturing is easy and identification is also easy, but to culturing for non-sporulating fungi is a big challenge. Okay. And we use a series of epigenetic regulators. Combination could be applied as standard. Later, we can make it as a standard procedure to study the various fungi strands. So essentially, start with a combination of different, and at the end, we finalize few of the combinations on the basis of our outcome and results. Okay. So our research objectives was to develop the epigenetic manipulation procedures and applying them onto the fungi from Malaysian and polar regions. And these are the specific objectives. Okay. So limitation to culture this fungi at a small scale. We want to make it on a small scale, and we want to use a aero media using this Maxus protocols. So the newly developed this epigenetic manipulation procedures provide us for rapidly accessing the silent secondary metabolic pathway, and it also can be implemented in a lab as a standard method to screen the fungal secondary metabolites, and to contribute. It also can contribute in the discovery of compounds. This is a simple layout for our methodology. We did initial screening and method development, isolation of Arctic fungi, and we apply our method in which we have method imputation, and then we did the HPLC analysis of all the extracts, and then characterization of our fungi of interest. So we select which one have we give the promising peaks and results after bioassay, on the preliminary screening, and then we have isolated the selected one. From the selected one, fungi isolate, we have isolate the compound and purify them, and we did the again antimicrobial assay. So initially screening and method development is a big challenge for us because it includes the selection of fungi and epigenetic modifier as well. And we have to see the media comparison, natural versus synthetic. We can use a media as a natural ingredients or the synthetic ingredients, and it helps to method development for culturing, especially the non-sporulating fungi. And we culture the Malaysian fungi with the epigenetic modifier as well. And we did the extraction, we use solid phase extraction, and then we subject for the chromatographic profiling. Okay, so this is the, you can see this is a very, looks like some complicated, but it's a very, because it takes a long, maybe a year to go for this method and selection and all of this. So we use microtetra plate and sandwich cover systems. Okay, so this you can see the microtetra plate. So here you can see this is the stainless steel clamp. So we make it a sandwich using all this filters, and then we just clamp on this board. This is the custom made board. Okay, and this is, you can see the B is the stainless steel silicone mat, and this one is the EPTFE filter. This is C, and this one is the microfiber and spongy silicone, and this one is this is the cover. So as you have your sandwich, so that we just keep inside all the things and then make it like a sandwich. And this is microtetra plate. Okay, so these are the fungi we use. Okay. So these are the fungi. This is aspergillus. This is another species aspergillus. This is Telomyces. This is penicillium, and this is Aspergillus fumigatus. So the epigenetic regulators which are used for this research, we use for the histone deacetylase inhibitors, we use melperic acid and we use the sodium butyrate. And for the DNA methyltransferase inhibitors, we use SAHA and we use SAHA and we use another this 5-azacine, the azacitidine as well. So I'm using the abbreviation instead of using the full name. So this is the results. Selection of the growth media. So natural and synthetic media comparison. We use this potato texture again and synthetic dox media as well. Comparison on the morphology of the fungi and chromatographic profiles. For the morphology, full growth and heavy sporulation, what we observe, and the chromatographic profiles, good secondary metabolite production and separation is one of the target. And PDA, PDB, the potato texture burr selected as the growth medium. Okay. So this is you can see the method development for this culturing non-sporulating fungi. Fungus, then we take in a vacuum tube, and it helps for the homogenization. And fungus from the PDA plate, this is PDA plate, we just inoculate here, and we incubate it for 8 days for at the culture homogenized using genus grinder. And then we just use this one, were design plates for the fermentation of fungi, 8 fungi in 1 macrodated plate with the addition of epigenetic regulators. So we use 8 fungi at a time on this microdated plate, and then with the addition of epigenetic regulators as well. And then it is the cultures after this culture are ready in this microdated plate and is ready for extraction. Then we use SPE method for extraction of this MTP microdated plate. And after the extraction, we have extract. This extract was subjected to for the chromatographic profiling to the HPLC, and then we identify the compound. So we are using psychotrophic fungi 59% and psychophilic fungi 41%. We are able to isolate from this Arctic because when we are able this is based on the temperature, at which temperature they are able to grow. So we are find out 59% on psychotrophic and 41% of psychophilic fungi. So these are the collection site. From where it was collected, and these are the number of fungi. It's collected as a psychotrophic and psychophilic, and total we are able to collect 41 fungi from Arctic. These are some examples. As it can be seen on morphology, can be seen on the PDA plates. Okay, so for the fungi characterization, first we start with the microscopic examinations, and then we go for the molecular techniques. For the molecular technique, we do the DNA extraction, DNA amplification, purification, and then sequencing, and then submit it to the gene bank database for the confirmation. So for example, this is the first isolates genomyces, and you can see it is under the microscopes. And this is under the SCM microscope. We have this scanning electronic microscope at our institute lab. So we for the further identification, we use this SCM. Okay, this is another fungi. And this is on the microscope image. And this is again under the SCM. We can see it is very clear. We can see the hyphae. We can see the quantity very clear. So we can initially we are able to identify based on their SCM. And for the further we submit for the DNA extraction and gene bank. This is another fungi we are able, and this is the SCM for this Delonomyces. Okay, this is another one, penicillium, and you can see a very clear image for this SEM. For this fungi penicillium, we have very clear conidia, conidiophore. Okay, this is another one, Aspergillosis. So all this I identified, we are given this name based on this SEM and DNA profiling. Okay, so for the screening of fungi in the selected media, we use the media epigenetic regulators to the medium. This in the media was PDB 70%. And for the control, we use no elicitor in order to see what happened with and without the elicitors. So this is our elicitors. Well, this is SAHA and SAHA's, and this is VA, valproic acid. We combine synthetic and natural, right? So here we combine with SAHA, VA, and this is sodium butyrate. This is another valproic acid. This two we use as synthetic, and this third one as well. So this is the combination we use as a media constituents. Okay, so if you see here on after extraction from the marketed phase, this extract was subjected to HPLC, including the control. And here you can see for this control, this is without the media, without the epigenetic regulators or elicitors. Here we use one SAHA, here we use another one, and in the SAHA and in the valproic acid. So in here, we use in the incubation period, we use for 40 days and the temperature was 28. You can see that very clear difference with different elicitors and epigenetic regulators. Okay, so with the addition of SAHA and SAHL, this is for the 5 weeks, and we did this one at 10 degrees centigrade, where we did for 2 temperature with different 2 fungi. So you can see here for the intensity is very clear with using this epigenetic regulators. The yield of the metabolites, secondary metabolites in the fungi is enhanced. So it's a good results for us. And here again with for this extracts with if you compare with the control, again there's a bit increase in the metabolite at attention time P with using the combination of this epigenetic regulators. Okay, then we subjected for further finalizing our selection of our extract of interest. We did antimicrobial screening, and we use this pathogens, these microorganisms. And for the positive control, we use gentamicin. So this is the antimicrobial screening of the crude extracts here. And you can see here the B2, C2, and D2, CD, and SHF exhibited a broad spectrum of activity here. Okay, so selection of fungi. 3 fungi was selected for scale-up fermentation because we have more than you can see we have around 42 fungi. So based on that, we selected our this screening. We selected these 3 fungi for the scale of fermentation and isolation of the compound. And this selection based on the chromatographic profiles and antimicrobial activities of the extract as shown in my previous slides. Okay. This crude extract after this was subjected to preparative LC for the fractionations and for further for semi-prep, for compound purifications, and finally for the spectroscope method and comparison with the literature. Okay, so these are the compounds which are isolated, and you can see details are here. This compound is isolated in the presence of using this epigenetic regulator, and again this curvilion is we are able to isolate in the presence of using epigenetic regulators SSHCl and SSHG. Same is over here. So here we are able to have 4 compounds. Okay. So this is a HPLC fractionation of one extract SHSF. So here you can see these are, we have the selectivity and we have 2 compounds and which are in very good yield. So this is how is this a process, how we use recycling HPLC, because you can see the peaks are very close to each other. So this is a bit challenging to isolate this very close peak because the resolution is not very good for this between these 2 peaks. So here we applied our recycling HPLC, and every time we can recycle our, and then we can collect our desired, and we are able, but once we recycle further time, so you can see here the peaks are very close, then the distance increase, and at the end we can see the at cycle 5 the peaks are separated and we can isolate our peaks, and then easily after 5 cycle of 5 cycles. Okay, so this is the NMR. What is compound, because this compound is as a mixture, we have as a mixture. Okay, so this is the C13. Okay, so this is the results for the, because sometimes only for the synergistic effect we have more than one compound, we have synergistic effects, we have high good activity, but at the end for the pure the sometime activity may be enhanced for one compound, decrease for another compound, or sometimes we don't have we select just two and we just left a small peaks, so we have no activity because of synergistic effect. Okay, so this method for culturing for non-respiratory fungi in a microtiter plates is successfully developed and is incorporated in the METSUS protocol. And we just manipulate, we did our method for using this epigenetic regulators, and we just found out the standard operating procedures for in our lab, and we are able to develop. And this isolation of this 41 psychrophilic and psychotrophic fungi is also successful during our this research, and we have 5 active fungi are identified successfully here, and the 3 crude extracts show the significant antimicrobial activity, and we are, which is, this is just a part of this work which I'm going to present, because we just select a few fungi. From few fungi we select only 3 extract, or from this 3 extract we are able to isolate this compounds, and actually this is based on our method. So on this basis we can see we are successfully able to develop these methods. Okay, so this epigenetic manipulation procedure can demonstrate that it could be used in high throw output screening, which can handle and screen a large number of samples at a time, and we can narrow down our work, and we can just select, because if we take a lot of start with one fungi and at the end we have nothing, so it's better to start with some fungi and narrow down with 2 or 1 or 3 fungi, and then you can scale up with the selected fungi and can able to have something in our hand. So this crude extract we can, okay, so actually this work is done by my PhD students, so I really appreciate and acknowledge them. They really did wonderful and hardworking, and they were just persistent to do and keep the going and trying their best. And this is supported by my FRGS grant. And at the we have I haven't included my all the publication list here, but we are able to have published papers in tetrahedron letters and microbiology journals and molecular structures, bioorganic chemistry, and even we have published one paper in natural product, JNP, Journal of Natural Product, and it's the work still is going on, and now this student is working as an academic staff, and she is still working on her fungus, and which are not included. She is working more on that. So then I would like to thanks to all of you, and I would like to acknowledge my faculty, my pharmacy, my dean, and my grant and Ministry of Higher Education, mostly for giving me the grant and funding always to carry out my all this research. Thanks a lot. Thank you for your patience.
Dear attenders, if you have any query regarding the present, kindly drop your query in the chat box or raise your hands.
Okay, so I stop sharing. Okay, thank you.
Thank you for an informative session, ma'am. Next, I would like to welcome our keynote speaker, Professor Murali Ellappu, to give a keynote address. Murali Ellappu is a tenured associate professor of the Immunology and Microbiology Department and a member of South Texas Center of Excellence in Cancer Research at the School of Medicine, University of Texas, Rio Grande Valley, Texas, USA. He is currently seminar coordinator for DIM sector seminar series. His current research primarily focus on the development of nanomaterials for improved therapeutic potential of clinical drugs using nanotechnology. Additionally, through Cancer Immunology Institute, his laboratory will generate safe and effective nano formulations for cancer immunotherapy. He is also world top 2% highly cited scientist. He serves as additional board member, associate editor, and senior editor role of various journals. He has published over 160 peer-reviewed articles and reviews in journals, 12 book chapters, and over 125 conference abstracts, and over 25 invited talks. His work was cited over 12,430 times with an H index of 55 and I-10 index of 128.
Thank you, organizers. Can you allow me to share my slides?
Now you can share.
Okay, so can you all able to see this? Good morning, everyone. Can you all able to see my slides?
No, sir, it is not visible.
So, okay. Mute.
Now it is visible, sir. You can start. Sir, are you available now?
Good morning, everyone. I think, I have issues with the Zoom. So whenever I'm sharing, I think it's kicking me out from the Zoom. Can you able to hear me?
Yes, sir. You can proceed.
Okay, so I will try one more time sharing the slides. Otherwise, I can send it to you. You can change the slides. Okay. So I'll try to share the screen. Okay. So can you all able to see the slides?
Yes, now it is visible.
Okay, that's wonderful. So good morning, everyone. So I'm very happy to be here on this day, and I want to give some information about nanomedicine, how we use them for cancer therapeutic applications. Okay. Can you hear me well?
Yes, sir.
Okay, good. So today I will be covering some of the basic concepts, and also I would be giving advanced knowledge how we make a different type of nanomedicine for cancer therapeutic applications. So whenever we think about nano, we all might be thinking whether it is only or primarily
Sorry
meant
for the
for
interruption.
drug delivery.
Hello.
Yes,
Can you please make it a screen big? Full screen?
full screen.
Yes.
Okay, good, wonderful.
Yeah.
So now it looks good at your end.
Yes. Okay, sir. Now we can.
Okay. So whenever we think about any nanomedicine or any nanomaterial, always our impression would be whether it is only for drug delivery or any other applications. So this slide will give you some information that not only the drug delivery aspect, but also the same nanotechnology can be applied for various other fields such as nanoelectronic and nano concrete or nano other. Can you able to hear me?
Yes, sir.
Hello?
Yes.
Yeah. So whenever we talk about nanomaterial, we would be thinking that can be used for various other applications such as drug delivery, nano concrete, nanoelectronics, or nano detection. So that how these methods would improve certain way, either the delivery or the strength or the communication or the detection level. So what that means, that nanomaterials, okay, I was unable to move the slide. Okay. Okay. Yeah. So whenever we think about nanomedicine, we might thinking you have a drug molecules that are embedded inside the nanoparticles, right? So that's our concept for now. So where we use these nanomaterials, especially in the chemotherapy area, where you have your chemotherapy drug, and then those will be going inside your nanoparticle, so that we may experience better delivery profiles. So that was our concept so far. Then how we would achieve all these things that we will be discussing today in this seminar. So always we might be thinking that any drug molecule, especially anti-cancer drug molecules, if they are low in soluble and low in stability, those may not serve our purpose, because like all the drug must be formulated or injected through IV or orally taken up, so that they will go and dissolve and then they go to different parts of the body. If the poor solubility means it's not good enough, right? That's the number one issue. Another one is like, even if you have a good solubility, if your drug is very potent in nature, right? That means that drug is quickly going to go each and every part of the body. Therefore, the drug molecule will be having effect on all cells, right? Not only just cancer cells, but they will be having on other cells also. And same way, low targeted efficacy, like, we wanted to send this drug molecule to the tumor cells, but, if you have a poor bioavailability, maybe the drug is not reaching to the tumor site. Apart from all these, maybe drugs also, they develop, cancer cells develop drug resistance to particular anti-cancer agents. So these are all conventional issues that one can experience when it goes to a clinic, right? It could be a first line therapy or second line therapy or third line therapy, anti-cancer agent, but they all may experience either one or many of these issues. So the nanotechnology aim is to improve one or few of those issues that we are discussed, okay? So nanoparticles. So what do you think about nanoparticle? One, how we imagine them, right? If we have a glucose molecule or protein, DNA, viruses, the range of the nanoparticles that we are going to discuss them primarily from 1 nanometer to 100 nanometer. Those were like, if you take a protein molecule, okay, one protein, human serum albumin molecule, it's some, close to 70 or 60,000 molecular weight in general. Those kind of protein molecules that you will see on the DLS, the size would be somewhere 3 to 4 nanometer, right? That means the nanoparticle that what you are thinking are close to 100 nanometer. That means if the 10 or 20 of these proteins, bind together, then you will see a 100 nanometer particle size formulation, right? So that is that you can imagine, right? Simply. So this slide would give you brief information about various types of nanomaterial that are divided in the size range. Like, for example, 1 to 10 nanometer dendrimers, metal nanoparticles, carbon nanotubes. Those are all, close to 1 to 10 nanometer in size. But, there are various other nano materials, like, those fall 10 to 200 nanometer or 100 to 300 nanometer. There are some of them are, even close to 500 nanometer, but, our focus would be primarily today would be on the nanomedicine that is going to go for 10 to 200 nanometer, or in certain cases 100 to 300 nanometer. So primarily, polymeric nanoparticle, lipid nanoparticle, hollow or solid lipid nanoparticles and so on. So what's the advantage of having or choosing a nanomaterial, right? So the number one criteria, what we think about nanomaterial is that nanomaterial, they have a inherent property, not just because of the smaller size in nature, but they have a EPR mechanism that one can achieve with the nanoparticles. That is nothing but a passive targeting mechanism of the tumor cells, right? So here on your left hand side, you can see the yellow color nanoparticle, they can go into the blood circulation. Whenever they see a leaky vasculature at the tumor site, these nanoparticles can go in and lock up there in those tumor tissues. So if you have a small molecule, let's say anti-cancer molecule, like 1,000 molecular weight, or 250 molecular weight, or 1,500 molecular weight. Those are very smaller in size, smaller than your nanoparticle. They may go easily into the tumor cells, but also they can come back. Okay. On the other hand, I told you, drug resistance, right? So drug resistance case, what happens? The cancer cells are smart enough, they can detect okay, this is the drug is coming. Okay, they can throw out from the cancer cells. Okay, so so it's called they overexpress some of the PGP expression on these cells. Therefore, the cancer cells will throw out those drug molecules from the tumor cells. That means your nanoparticle, if you use the nanoparticle, they can go there and then they're locked up in the tumor tissues, and also the drug can be released within the cell so that there won't be any opportunity for the cells. They cannot identify this is the actual drug. Okay, that's one mechanism. And another mechanism is that active targeting. Active targeting is nothing but on the same nanoparticle, you conjugate the anti ligands or receptor antibody, or that can specifically detect the tumor cells. So these are the two very important parameters whenever you are thinking about nanoparticles. And earlier slide, it showed different nanoparticle size. You can prepare nanoparticles, but which range that we want to use them, right? This slide you can see clearly. So if you have a very smaller nanoparticle size range, such as 1 nanometer, right? So if you have a 1 nanometer particle, they will be cleared by the kidney easily. On the other hand, if you have a 10 nanometer particle size, those will be cleared by liver, right? The good size range is, excuse me. So 10 nanometer to 200 nanometer, good size for the achieving the EPR mechanism. So EPR mechanism is the best suit for cancer therapeutics, right? So before I go for explaining some of the examples, I want to give you a good example of nano-based technology, how that can be implemented. So this slide is giving you introduction of particle-based formulation for the same drug that was synthesized in 1887, that is aspirin molecule, right? So here you can see in various years and in various forms and various formulations were developed for the aspirin. So nowadays you can see the quick advertisement, it comes, okay, you take this aspirin drug, tablet that, your pain will be gone quickly, within a few minutes. What that means is that this pellet or the tablet contains, micro-based particles. Those will be, dispersed quickly in the blood circulation. Therefore, it can reach at the inflammatory site quickly, right? So that's the concept behind this. So same way, why we want to make nanoparticles in the first place, as I mentioned before. So either that has to increase the solubility of your formulation, or maybe overcoming the drug resistance, or maybe it minimizes the side effect of that particular drug. So now I will give you brief example, like for example, doxorubicin. So doxorubicin is a hydrophilic molecule. It's widely used as an anti-cancer molecule. Okay. The main issue of this molecule is that this is not going to discriminate whether it's the cancer cells or normal cell. Okay. What it does, it goes into the cell DNA, it binds there. Okay. It breaks the DNA and it damages the DNA. Right. It's a function of this molecule. So what it does, like, it reaches more amount to the heart and especially cardiomyocytes. Okay. So therefore, this drug has more effect on the cardiomyocyte other than your cancer cells. So what happens there? Certainly you have a cardiotoxicity there, right? So how we can minimize that cardiotoxicity? That one we can make a liposomal formulation so that you can minimize that cardiotoxicity. Therefore, you have generated this doxyl nano formulation, and where you can see complete options of cardiotoxicity here. You can see complete options of cardiotoxicity here, right? And when you go, the same technology can also be implemented for various other similar type of drug molecule. Here you can see amphotericin B. It's like antifungal agent or anti-microbial agent, right? Another example is that Abraxane. So what is Abraxane? Abraxane is a paclitaxel based formulation. So what is a paclitaxel? Paclitaxel is one of the anti-cancer agent. It binds the tubulin. Okay, it helps to stabilize the tubulins. Okay, so in any cancer cell in the division or any cell division, okay, this is very important. Okay, so those needs to divide into one cell into two cell into four, right? So what happens, like, if you have this beta tubulins, okay, it is continuously polymerized and it will not allow to divide those cells. Okay, so that means, like, your cancer cells will undergo apoptosis. Okay, so paclitaxel does this mechanism, right? So then it's also not a unique or not a specific mechanism for cancer. That means, like, wherever this drug molecule go, you will see same effect. Excuse me. So this is the best way that you can make a formulation of that so that you can minimize that particular systemic side effect. So this is the best example for you have a Abraxane. It's nothing but a albumin-based conjugated paclitaxel. It will increase the circulation and also it improves the EPR mechanism. Apart from all this, it also acts as an active targeting mechanism by binding to GP60. Okay, so it will exhibit both passive and targeting, right? At the same way, this type of PLGA based nanoparticle based drug depots that can deliver for over period of 6 months. This another example that is FDA approved formulation. So what are all these is telling us that when you encapsulate your drug molecule, it can prevent from the degradation, and also it can improve the solubility, and also it can improve the improve the targeted ability, and also minimize the drug resistance, and also minimizes the systemic side effects that are associated. Some of the nanoparticles, for example, iron oxide nanoparticles, they themselves act as a therapeutic molecules, like, so because they undergo, like, they exhibit certain property that can help to heal that nanoparticles. For example, like somebody has any brain tumor or a breast tumor, if you inject right into the tumor cells this, iron oxide formulation, and then you applied magnetic current. Therefore, the nanoparticles will be come under the motion, and then the heat would be generated so that the heat will kill the cancer cells. Considering all this, huge literature, current data shows that liposomal or polymeric nanoparticles are the best suit for drug delivery application and especially for cancer therapeutics. So here I want to make everybody attention that so the liposomal formulation, you all might be enjoying the privilege of this formulation. Formulation for mRNA technology. At the same time, so they received a Nobel Prize for that mRNA technology this year. At the same time, another, excuse me, another chemistry Nobel Prize for this year went for, carbon darts, right? So these are the two nanotechnology achievements that you all can appreciate. Excuse me. So with all this knowledge, what we did, we created our own new formulation, considering that, iron oxide based or polymeric based nanogel based. So we created a number of formulation, but before we go there, we tested, which formulation would be best. Now we tested one drug molecule, and we made a self-assemblies, polymeric based nanoparticles, polymeric crystals, dendrite-based formulation, and magnetic nanoparticle formulation. And we found out that MNP-based formulation was best in our case for that particular molecule. Therefore, we went ahead and then made various modifications, and then we have a patent on this particular formulation that we can use either for small molecule, drug molecule, or any nucleotides or microRNA, siRNA, a delivery purpose. So now on, just I will go briefly. We have number of examples that we can show. Majority of them are published. You can put my name on the Google Scholar on the PubMed, so you can see all my publications. So this one of the example that we employed for microRNA 205 delivery. The necessity was microRNA 205 was lost in certain type of tumor cells, particularly in prostate cancer cells. So our idea is supplementation of this microRNA 205 would benefit the cancer cells so that it will be stop the growth of tumor cells. Okay, so these are all the assays that we commonly employ. For example, like how much microRNA is binding, that we show from the fluorescence spectra and gel, DNA gel, DNA RNA gel, and then a CD spectra, and then we did some hemolysis assays, and then we did stability assays and uptake performance of that formulation, and then how that acting on the tumor cells in terms of proliferation, colony formation, and also we did how after encapsulating this microRNA 205, whether its mechanism of action it still remained or not, whether this after putting 205, whether we have achieved the reversing the drug resistance mechanism or not. This is all we apply. The same technology can be implemented for a wide variety of models that here we see a PSMA targeted nanoparticle formulation. You can clearly see that formulation and its characterization, and then a dose dependent uptake in the cancer cells by TEM, and also you see the molecular mechanism, and also we see the tissues from the mice after treatment with the various formulation, and then its parental drug molecule, and then targeted ability of these nanoparticles and so on. Like, there are so many examples that I can show, but, so just I will stop with this example, what we did here. So we created a nano nanoparticle formulation, but we quoted various type of cell membranes. For example, neutrophil, mesenchymal cells, raw cells, and so on, Jurkat cells, and so on. We put them on the nanoparticle, and then we thought, which formulation would take more at the tumor site. We tested them, both in vitro and in vitro in the mouse model, and then we figured it out. Some formulation was better than the other. Finally, we figured it out. One formulation was best. This is one of my PhD student work, and then we find out that way. Not only that single drug molecule, but also we created a nanoparticle platforms that can deliver dual drugs or one siRNA, one drug molecule. This is another example that we show, like, whether we achieve any synergy with this two dual delivery and so on. You can target this formulation for the bone and also for the macrophages. Macrophages are heavily exist tumor associated macrophages. Those are more than 50% of the cells exist at the tumor site. So targeting those macrophages also another way of targeting tumor cells. And also majority of the tumors, like those were metastasized to the bones, right? So you take, like, for example, brain and prostate, sorry, breast and prostate cancer cells, the first or majority of those metastasis, like, the breast cancer cell, they can go to the bone and then they make a bone metastasis. Same way, prostate cancer cells, they can go and then reside into the bones and then they develop bone tumors. So we developed some formulation that are specific to bone targeted delivery. So with this, I stop here and then I'll take any questions if you all have. And also, like, so you can have my email ID. If you all have any questions, please feel free to communicate anytime and we can discuss if you all want to have any future collaboration or anything like in the area of nanotechnology. So I'm presenting only related to nanotechnology, but our focus is also on other diseases such as diabetes, liver diseases, and also HIV, brain tumors, and any neurological diseases and so on. For example, ALS, Alzheimer's, and so on. So we will be happy to collaborate with any of your teams that if you are interested anytime in the future. Thank you. It's open for the questions.
Dear participants, if you have any query regarding the present, ask your questions now.
I think
Thank
if
you
there are
for
none,
your
thank you so much.
session, sir. Please attention everyone. Due to the cyclone at our location, certificate will be issued by the next week on Saturday. Thank you for your understand. Now I request all our participants to move their hall for the presentation within 5 or 10 minutes. Please attention everyone. I request all our participants to move the hall.
அதுதான் இருக்கும் mostly. ஆமா அனுப்பிடுங்க.
సోడియం హైడ్రాక్సైడ్ పెల్లెట్స్ రెండు ఉన్నాయి. ఒకటి అమోపెండ్ ఒకటి ఫైన్ డైసోడియం సిట్రేట్ అమోపెండ్
Good afternoon, everyone. Has everybody joined the Good afternoon, all. I'm Priyanashni. I will be moderating the main hall presentation. Let's welcome our session chairs of today, Dr. Padma M. Parag, Principal, Department of Pharmacology, the Oxford College of Pharmacy, India, and Dr. A.L. Akhilandeshwari, Associate Professor, Chettinad School of Pharmaceutical Science, India. Let's start our today's session with the presenter, Darshini AD, Wales Institute of Science and Technology and Advanced Studies. Please present your paper. Darshini AD, are you available now? She's unavailable at the moment. Now let's move on to our next presenter, Sangeeta Kumari from Birla Institute of Technology, Mirza, Ranchi. Please present your paper, ma'am.
Hello.
Thank you. Thank you, ma'am, for giving me the opportunity.
Thank you, ma'am.
Just give me a minute. Ma'am, please let me know, is my presentation is visible?
Yes, ma'am, it's just visible. You can continue.
Oh, okay. Thank you so much, ma'am. So I'm here to present my paper on determination of bioactivities and phytochemical screening of methanolic extract of Ficus bengalensis slim. And coming to the introductory part, Ficus bengalensis is mainly, it is known as the banyan tree, and it is most sacred tree in our country. And it is a evergreen tree growing up to 30 meters. And coming to the medicinal uses, it has a tremendous work in the field of medicine. It is used for the remedy of toothache, for wound healing, for improving the vision of eyes. It purifies us blood and many more. And firstly, my work is on, actually I'm a PhD scholar, and my work is on anti-obesity effect of this plant. So first, but before that, I have to do the chemical screening and bioactivities of this plant. So this is the first part of my research. So firstly, I have collected this plant, and it is get authenticated by BSI Kolkata under this number. Then I extracted the powder using methanol and chloroform. Under the rotary evaporator, it is evaporated and the extract are allowed to dry. Then I do the phytochemical screening as per the protocol, as well as total phenolic effect, as well as total flavonoid contents. And also other bioactivities such as in vitro alpha amylase, in vitro assay, DPPH for antioxidant, for reducing property assay, lipase inhibitions and anti-inflammatory effects, and also TLC profile and HPTLC fingerprinting were done as per the protocol. And here is the result. Coming to the result part of both extract, for chloroform extract as well as methanolic extract, it is viewed that in the methanolic extract, there is the more presence of glycosides, saponins, tannins, triterpenoids, etc. You can see this chart. And coming to the percentage yield, also I have only taken the methanolic extract because the methanolic extract so more potent result. And coming to the physical property of the extract, so under the swatch, ma'am, there is some disturbances. Okay. Coming to the physical properties of the extract under the Swatch UV, that is 254 nm, it is showed the dark brown color, and under the high UV, long UV, it is shown black color. And the total flavonoid content was found to be 92.04 mg per dry gram, and total phenolic content was found to be 99.5 mg. And similar were the result with total hydrogen peroxide scavenging activity. The highest concentration, that is 150 μg per ml, shows 72.5, while compared to the standard, that is ascorbic acid, it is 76. So it is so somehow similar result. And also we can see the graph, there is R square value is 0.994, which is quite good. And coming to the phosphomolybdenum scavenging activity, the highest concentration of my extract shows 72.48%, while the standard shows 93.03%. And similar with alpha inhibitory assay and DPPH radical, have the like IC 50 value is 7.2, where the standard show IC 50 value of 8. And here is the graph, like in the highest concentration, it is gradually shown increasing order, and the R square value of the extract is 0.989. Similar with DPPH free radical scavenging activity, the percent radical scavenging activity of the sample is shown to be 0.89%, and while the standard shows 97% with the IC value of 0.32, while the IC value of the sample shows 0.73%. Now, power reducing samples. Among the 6, I have taken the 6 different concentration. Of the highest concentration, the absorbance of the sample shows 0.62, while the standard show 0.99. Here the standard curve, and here is the comparison between the standard and as well as my extract. And as I am working on the anti-obesity effect, so I have done lipase inhibition also. So the highest concentration shows 76% of inhibition of standard, while the sample shows 69% and gradually it is increasing. In vitro anti-inflammatory activity I have also done. So for the standard, the results show 88%, while the sample show 77%. Here is the inhibitions curves, which show the R square value is 0.99, and here is the comparison between the standard as well as sample. This is the TLC profiling. So here I have taken the standard quercetin. So there is almost, it is similar to the quercetin, and in the HP TLC we can see like having the RF value 0.67 of standard, while the highest concentration of my sample shows a similar effect of quercetin 0.67 value. So as we know that quercetin is a flavonoid and it's having the antioxidant effect, and also in some paper it having the anti-obesity effects. So we can consider that this plant also have the anti-obesity effects. So coming to the conclusion part, so the bark promising bioactivity properties. So it having the antioxidant potentials, high flavonoid contents, high phenolic contents. Also it have antioxidant property, it having the phosphomolybdenum scavenging activities. So it can neutralize the harmful ROS, and also it having the alpha amylase inhibitory activities. So it suggests it can manage blood sugar, that it can treat the diabetes, and also it having the we can say a potential of lipase inhibitory. So we can say it is a bioactive potential for anti-obesity effect also. So thank you so much. These are my references.
Thank you for your presentation, ma'am.
Okay, ma'am.
Session chairs, if you have any question related to the presentation, you can ask to the presenter.
See, you have done this DPPH essay. Can you show the results once?
Ma'am, DPPH. Okay.
Yeah, DPPH and that slide you show, please.
Yes, ma'am. Okay, this is protocol. Result.
Next slide you show. Result part.
Okay, ma'am. Okay, just a minute, ma'am. Yes, ma'am.
Yeah. This IC50 value looks wrong. Calculation part is wrong. Otherwise, it's okay. Alpha amylase you have done. Then why you have done anti-inflammatory? You wanted to check obesity, na? So why anti-inflammatory you have included here also?
Okay, ma'am. Okay, ma'am. I will rectify it. Thank you so much, ma'am, for suggesting me.
Okay, fine. Anything else? Other speaker can ask the question. From my side, this is the question. Okay, thank you.
Thank you for your presentation, ma'am.
Thank you, ma'am.
Ma'am, good morning. Ma'am, am I audible?
Yes, ma'am, you are.
Yes, ma'am.
Next, now.
Ma'am, I'm Dashni from Wales University, ma'am. Ma'am,
Yes.
am I audible?
Yes.
I was the first presenter, but I due to network, I can't able to join. Can I present now, ma'am?
Yes, ma'am. You can continue your presentation.
Okay, ma'am, can I quit my
Yeah.
presentation?
Yeah, thank you, Sangeeta.
Or any other questions? Okay, thank you so much.
Thank you for presentation.
You cannot start screen.
Now let's call upon our next presenter, Darshini AD from Wales Institute of Science and Technology and Advanced Studies. Please present your paper, ma'am.
Okay, ma'am. Ma'am, is my screen is visible?
Yes, ma'am, it is.
Okay. Very good morning to all. I'm presenting a case study on autoimmunohemolytic anemia with bicytopenia. Okay. Autoimmunohemolytic anemia is like AIHA, which is a rare autoimmune disorder. It may be result in production of antibodies, which is of autoantibodies against our RBC, causing a shortened erythrocyte levels. The main incident rate is of 1 to 3 cases in 1 lakh persons per year. The indications and classifications of AIH causes can be done carefully. The main confirmatory test is done for AIH is positive direct antiglobulin T, that is PDAT. The treatment option for AIHA is still unclear. First line drug can be given as corticosteroids, rixutimab, which is of the second line option for AIHA. Splenectomy is a procedure which is done for relapsed cases, but only 20%
Sorry
of
to disturb you. Please make us a slideshow, no? It is not in slideshow.
Okay, ma'am. Ma'am, now it's okay, ma'am.
Yeah, it is okay.
Okay. Okay. Bicytopenia, which is of a rare disorder, occurred in few cases, like RBC level, WBC, and platelets are the three levels which is being based on that bicytopenia is being diagnosed. For this case, patient had WBC level and platelet count is being low. The treatment option for bicytopenia is being still unclear. And not evidence-based. Bone marrow aspiration is being performed for malignancy and all malignancy. In case of severe anemia, blood transfusion can be done. And first-line drugs like multivitamin is being prescribed for this patient. Early diagnosis for AIH with bicytopenia through the routine supervision of complete blood count of anemia patient and anemia patient who had undergone the transfusion during the previous years helps to identify the production of autoimmune disorder. And this is a case report. A 22-year-old female patient was admitted in the hospital with a chief complaint of severe anemia, fatigue, and knee pain and loose stools. Due to her low hemoglobin level, like 3.9 she had, at the age of 19, she had underwent blood transfusion. On examination, she had a mild splenomegaly, pallor is being very severe for the patient, and routine monitoring of hemoglobin, total RBC level, ESR, erythrocyte sedimentation rate, and platelet count was carried out during hospitalization. A bone marrow aspiration was performed to look for malignancy, like tumor, any cells are being present or not, but the patient had a negative. Bicytopenia was identified after the peripheral spur test like RBC, WBC, and platelet, and that revealed reduced RBC and WBC count. We report here a case of AIHA patient and bicytopenia, which was detected in blood disorder center. Introduction. Autoimmune hemolytic anemia is a clinical condition that develop over a time. It being characterized by production of autoantibodies that is being attached to the RBC and causes hemolysis, which causes RBC cells less survival. Based on the erythrocyte destructions, which can be caused by extravascular red blood cells, phagocytes in the spleen, liver, and bone marrow, intravascular complement medicated lysis of erythrocyte. The type of antibodies being divided into two types, C and W. C is nothing but cold and W is nothing but warm. The main symptoms of AIHA is like severe anemia, fatigue, breathing difficulties, and knee pain. The main diagnostic feature is like DCT and anti-IgG Coops test. The main lab investigation is being checked out like CBC level, total WBC count, RBC count, platelet count, and ESR count. Bone marrow aspiration is being done for malignancy. Bicytopenia is like RBC, WBC, and platelets are the three present here. And checking their levels is the final diagnosis for bicytopenia. The treatment option for bicytopenia is still being not evidence-based one. Blood transfusion is being done in case of severe anemia and multivitamin is being supplied here. This case study shows that bicytopenia was seen in blood disorder center as a result of which AIHA patient. Okay. On examination, she had she was febrile, mild here, hematosplenomegaly, had a pallor, and elevated level of SGOT and SGPT level. Based on the routine investigation is being done, alkaline hemoglobin electrophoresis pattern was being normal. Result from CRP, like C relative protein and anti-nuclear antibodies was normal. On bone marrow aspiration cytology, examination showed mildly hypercellular marrow with adequate triannular hemoprosis, mild hematocytosis, and reduced iron store. These all being done based on lab investigation they have been proved. Additionally, the peripheral spear test results in reticulocyte count was 0.5 and platelet count was 1 normal range. RBC level was predominantly microcytic. Total WBC count was moderately reduced, and yeah, they had increased level of ESR level, very low level of hemoglobin of 6.3. The most common causes for the most common finding was anemia with leukopenia and microcytic hemochromic RBC level. It is an uncommon condition for AIHA patient with bicytopenias that was observed. The treatment which was given for this patient is antibiotics methylprednisolone 600 mg with 500 ml NS infusion over 4 hours, and proton pump inhibitors like pantoprazole, all those is being prescribed. Back the red blood cells, and later she was treated with steroids 30, and then it is being reduced to 20 based on the lab investigations. She was afebrile after 2 days, later developed a fever spike in the patient during the discharge time, and it was treated with proton pump inhibitors. She felt better and asymptomatic. After treatment, her hemoglobin level has been 6.5 to 7.5. Her complete blood count was showed like this. Total WBC count on admission, she had 1.19, and on discharge 4.59. Hemoglobin level initially she had 6.5, and it is being on discharge 7.5. Platelet count was 193, and she on discharge 233. ESR level is like 98. On discharge she had 56. Discussion. Autoimmunohemolytic anemia is known as ALH. The report incidence of the ALH was 1 to 3 cases among 1 lakh persons over here. But ALH with bicytopenia condition is a very rare case. That is my novelty. And treatment of autoimmunohemolytic anemia is still a challenge to clinicians. The treatment of ALH is mainly influenced by the cause of severity of anemia and type of antibodies. We continue to lose the patient because of hemolysis and other infections by immunosuppressions. Corticosteroids is a first-line drug is being prescribed. In the case of corticosteroids resistance, immunosuppressants like cyclophosphamide, azithromycin, cyclosporin is being prescribed. Splenectomy is being given based on their pallor conditions. It's not being given for all the patients with ALH with bicytopenia. The type of antibodies is mainly divided into two types, like as I said before, C and W. C is for cold and W is for warm. And the main treatment, first line drug is given for cold is rituximab alone or in the combination with chemotherapy. It's like rituximab is like anti-CD20 monoclonal antibodies, and it is being given for second line drug for patient with warm autoimmunohemolytic anemia, proven to be increasingly effective in treatment for B cells malignancy. That is, when B cells they'll be having a tumor. The mainstay of treatment for the warm AIHA is glucocorticoids, and intra IV immunotherapy is being given. And the recent studies have been shown that adding the infliximab early has a positive effect. Okay. Later resort therapies like high dosage cyclophosphamide is being given in the case of very severe cases. But the treatment option for bicytopenia has not been discussed much in many of the hospital articles, and it is not much evidence based. According to this report, bicytopenia patient receiving the routine monitoring of the complete blood count and blood transfusion done as necessary. Though the AIHA patient are cured with certain medicines, the AIHA patient associated with bicytopenia is being curable or preventable, pharmacologically manageable is still a question mark. And I'm concluding like a rare disease of autoimmunohemolytic anemia with bicytopenia as a manifestation of severe anemia, leukopenia, low WBC count, fatigue and tiredness. The observed case of AIHA with bicytopenia or a family history of anemia and a mother who had a consecutive pregnancy is the main cause for this. Through this report, we could suggest like early diagnosis of AIH with bicytopenia can be done by routine supervision of CBC count of anemia patient and anemia patient who had underwent blood transfusion during a childhood days helps to identify the prediction of autoimmune disorder. Further research can be built up by saying that AIHA patient with bicytopenia to determine the cause, treatment it pharmacologically, and it can be prevent further complications. Patient perspective, I was worried when I was initially diagnosed with autoimmune hemolytic anemia. The day before checking the low level of hemoglobin level, CBC level, and LFT level was abnormal. And I would like appreciate the medical professionals and other the team for helping to diagnosis and hoping that by sharing my story, more people will be get aware of this rare condition. This is all these are reference. Thank you, ma'am.
Thank you for your presentation, ma'am. Respected chair session chairs, if you have any question regarding the presentation, you can ask to the presenter.
Only corticosteroids are used for the treatment.
Actually, the patient was using corticosteroids before the admission also, ma'am. If the patient had been stopped the corticosteroids, the patient had elevated level of again WBC count. So only the patient is being prescribed with the corticosteroids before admission. After admission, the patient had been given based on their condition level, ma'am. Pantoprazole is being prescribed, all those.
But still the hemoglobin content and is not increased that much. From 6.5
Yes,
to
ma'am.
7.5 is not a good sign again.
Yes, ma'am.
Maybe within a few days of treatment, she will again face the same problem.
Yes, ma'am. She's facing still now, ma'am. It's like a question mark for her. She's taking steroids so that she's having more side effects like gaining weight. If she's reducing her dose like 20 to 10, she's reducing now. She's again getting her WBC count low level of WBC count. So still a question mark for her, ma'am. Autoimmune for
Okay,
the
fine.
patient.
Thank you. Thank you. Thank you.
Thank you. Thank you, ma'am.
Thank you for your presentation, ma'am. Now,
Thank you, ma'am.
Move on to our next presenter, Mansi Pal from Sardar Bhavan Singh University of Palwal, Dehradun.
Good morning, everyone. So the topic for presentation is molecular docking in oncology, addressing the challenge of multi-drug resistance in cancer therapy. Introduction. Cancer is one of the leading cause of death worldwide. Cancer is a group of disease characterized by the uncontrolled and abnormal growth of cells in a healthy body. Cell grow, divide, and die in a regulated manner, maintaining a balance between cell proliferation and cell death. However, in cancer, this balance is disrupted, leading to the formation of a mass of tissue called a tumor. Factors that are associated with elevated risk of cancer are tobacco use, lack of physical activity, alcohol use, low vegetable and fruit intake, and high body mass index. These factors are thought to be responsible for approximately one-third of cancer death. Breast, cervical, lung, thyroid, and colorectal cancers are the most common types of cancer in women, while prostate, lung, colorectal, liver, and stomach cancer are the most common among men. Multi-drug resistance in cancer. Multi-drug resistance is another refractory outcome of chemotherapy and is defined as the resistance of cancer cells to multiple chemotherapeutic drugs with different structures and mechanism of action. MDR is a major cause of chemotherapy failure and responsible for increasing cancer-related mortality. Molecular docking. Drug discovery is considered as a multi-process platform in which a specific chemical compound with desired biological activity on the drug target can be selected and eventually enter the drug development as a candidate drug. In this platform, both chemical compounds and biological targets are evaluated from different aspects by using various approaches. Need of molecular docking in MDR cancer. As a drug discovery and development, both are time-consuming and cost-intensive program. They pose many challenges for researchers in drug designing and discovering for various diseases such as different types of cancer. Therefore, the use of new technologies can pave the way, discovering new drugs with high therapeutic potential and take a big step towards disease treatment to also help in rational drug design and personalized medication. How molecular docking is valuable in MDR cancer? First is understanding drug target interaction. Molecular docking helps researchers understand the interaction between potential drug candidate and the target protein associated with MDR cancer. It allows the visualization of how a drug molecule binds to a target, providing insights into the molecular mechanism underlying drug resistance. Second, identification of novel drug candidates. By screening large libraries of compounds, molecular docking can assist in identifying new drug candidates that have the potential to overcome resistance mechanism in MDR cancer. Docking studies can predict the binding affinity of these compounds to specific targets, helping prioritize candidates for further experimental validation. Third, optimizing drug design. Docking can be used to optimize the chemical structure of existing drugs or lead to enhance their binding affinity and specifying for MDR related targets. This information aids medicinal chemists in designing more effective drug with improved pharmacological properties. Prediction of drug resistance and mutation. Molecular docking can predict how mutation in the target proteins may affect the drug binding. This information is crucial for anticipating and addressing potential drug resistance in MDR cancer. Virtual screening of compound libraries. Virtual screening Virtual screening using molecular docking allows researchers to evaluate a large number of compounds in silico, prioritizing those with the highest likelihood of binding to the target associated with drug-resistant cancer. This accelerates the drug discovery process by narrowing down the pool of potential candidates. Combination therapy design. Molecular docking can aid in the design of combination therapies by predicting the binding modes of multiple drugs to different targets involved in MDR pathways. This approach aimed to overcome resistance by targeting multiple pathways simultaneously. Reducing time and cost in drug development. Computational methods like molecular docking can significantly reduce the time and cost associated with the drug development by guiding experimental efforts towards the most promising candidates. In conclusion, the use of molecular docking in MDR cancer research holds significant promise for advancing our understanding of drug resistance mechanism and involving therapeutic outcomes. Molecular docking techniques enable the exploration of interaction between drug candidates and target protein by providing valuable insight into design and optimizing of novel compounds by identifying potential binding sites and predicting the binding affinity of the drug. Molecular docking aids in the development of targeted therapies that can overcome MDR in cancer. Through virtual screening and the identification of potential drug candidates, researcher can prioritize compounds with high binding affinities and selectivity for specific targets implicated in drug resistance pathway. Thank you.
Thank you for your presentation, ma'am. Respected session chairs, if you have any question regarding the presentation, you can ask to the presenter.
Did you do any specific MDR scan, this docking?
I have not done this.
Okay, fine. Thank you. And I request the organizer, please stick on to the time because lot many people are there. Thank you.
Thank you for your presentation, ma'am. Now let's move on to our next presenter, Abhed Kumar, Sardar Bhagwan Singh University.
मेरी कैसे होगी स्क्रीन नीचे हुआ है ना शो
Sir, please make it full screen.
Good afternoon, everyone. I am Abid from Sardar Bhagat Singh University, and I am presenting my topic on creating tomorrow's healthcare, AI role in precision medicine. This is the content of my topic. Introduction: Precision medicine is a crucial approach to healthcare. Is a challenging traditional model by personalizing medical care to individual traits. Precision medicine aims to optimize treatment strategies by leveraging the vast and complex data set that characterize an individual's health profile. AI, with its capacity for rapid data analysis, pattern recognition, and predictive modeling, plays a critical role in decoding the intricate interplay of genetic variation and environmental influences. AI in precision medicine has the potential to improve progos- uh, prognostics, and therapeutic approaches across a wide range of medical disciplines. Now, the roles of AI in precision medicine is diagnostic and imaging. AI have the capabilities in interpreting medical images such as X-ray, MRIs, and CT scans. The next is personalized medicine. AI analyze genomics information of patient. This enable healthcare provider to develop personalized treatment for more targeted and effective intervention. Next is predictive analysis. Predict disease progression, identify individuals at a risk of developing specific condition, and optimize preventive measures. The next is drug discovery and development. AI accelerates drug discovery by analyzing vast data set to identify potential drug candidates and predict their efficacy. The next is natural language processing. As AI have the valuable information from unstructured clinical notes, it enables better documentation and communication with patients' report. Here is the advantages of AI in healthcare. It improves diagnostic features, next is personalized treatment plans. Also, it improves remote patient monitoring, enhanced robotic surgery, and patient engagement and education. There are some disadvantages also with AI, like data privacy and security concerns, bias in AI algorithm, lack of transparency, reliance on technology, high initial cost and resource requirements, ethical issues, and limited generalizations. Now, the ways to overcome disadvantages: prioritize data privacy and security. Also, we can do is mitigate bias in AI algorithm and enhance algorithm transparency, promote human AI collaboration, continuous monitoring and evaluation, interdisciplinary collaboration, investment in cybersecurity. These are the main ways by which we can overcome the disadvantages which is come by the AI. Here is the conclusion. The deployment of AI in healthcare has shown promising results in various applications, including medical imaging, drug discovery, predictive analysis, and patient care management. This advancement have the potential to revolutionize patient outcomes, reduce healthcare disparities, and contribute to overall improvement of public health. However, the widespread adoption of AI in healthcare also raises important ethical, legal, and privacy considerations. Safeguarding patient data, ensuring transparency in algorithmic decision making, and addressing issue of bias and fairness are critical challenges that must be addressed to build and maintain trust in AI technologies. Thank you.
Thank you for your presentation, sir. Our respected session chair, if you have any question regarding the presentation, you can ask to the presenter.
No, this is the future. Maybe now we are saying a disadvantage. This will come in future. No, already AI has been helpful in lot of health diseases monitoring as well as healthcare management. So let us see how this will progress. Anyhow, it is a good presentation. Thank you.
Thank you for your presentation, sir. Now let's move on to our next presenter, Ajay Kumar from Sardar Bhagwan Singh University.
Yes, ma'am. Presenter, good afternoon, everyone.
Good afternoon, sir.
Ma'am, my screen is visible.
Yes, sir, it is visible. Please zoom your screen.
Okay, ma'am. Today is my topic, metabolomic in drug discovery. Metabolomic technology is being used in many areas. Drug development, discovery, its impact growing significantly. Metabolomics are small molecules present in the biological system. Technology involve and cultural expectation will necessary to optimal use this powerful tool. Definition: metabolism is the comprehensive quantitative or analysis of all metabolite. Creating comprehensive metabolite profile is more than just collection data, remediation, xenobiotic toxicity, or drug target engagement. To name just a few. After disturbance, the metabolite profile directly indicate phenotype and biochemical activity depending on the biological system. Under study, metabolite might include both naturally occurring substance and pharmaceutical, as well as byproduct of their. Two techniques are regularly employed for metabolite analyze: mass spectroscopy or NMR spectroscopy. The main advantage NMR is lie in its non-destructive nature, reproducibly and simple sample preparation, whereas lack of sensitivity limited molecule at concentration above 1 micrometer m pose the core limitation of this technique. Traditional technologies such as enzymatic assay, electrophoresis, spectroscopy, mass spectroscopy emerge high versatile and valuable technology crucial to many aspects of modern drug development. In addition to more stabilized applications such as metabolite monitoring of identification or its application as a high sensitive detection technology or for pharmacokinetic or pharmacodynamic study, it can also highlight potential target for therapeutic development. Additionally, metabolomics can be applied in medication development and in the research of new natural compounds. Drug testing and discovery can be improved by metabolomics, which can also shed light on the defective of medication that are intended but not intended. Metabolomics describe a field dedicated to the high throughput identification and quantification of small molecule in biological system under different conditions. Number of different terms have been used for specific application of high through metabolite analysis, including metabolomics, target or untarget metabolomics, fluxomics, lipidomics, genometabolomics. Principle: these are tiny molecule can be variety of endogenous and exogenous substance included vitamins, peptide, amino acid, nucleic acid, carbohydrate, and organic acid. Metabolomics can be very useful to improve such failure as it involved in the study, profiling, and understanding of the biochemical process, which help in identification of target engagement marker as well as prediction mode of action. Currently, pharmaceutical companies are utilize the approach in the early drug development stage to combat failure. This chapter will mainly highlight the advantage of this concept over traditional concept drug development along with recent development of it. Metabolomics provide as a sensitive measure of health of the phenotype of a biological system. Gene determine what may happen and metabolites define what has happened. The interaction the interaction between the genome transcription, proteome, and metabolome with the environment in the biological system. What are the principal metabolites? Example of primary metabolites include carbohydrate, that is glucose, fructose, sucrose, raffinose, stactose, mannitol, xylitol, and sorbitol. The non-starch include lignin, amino acid, that is arginine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, tryptophan, and valine, vitamin B complex. Areas of metabolomics. Areas of target identification, mechanism of elucidation, discovery of T marker, and physiological response marker include therapy monitoring. In turn, the use of mass spectroscopy and nuclear magnetic resonance spectroscopy based metabolomics has become a fundamental technology across the early target of drug development. Technology advance in the experimental work flow, analyze, detection strategy, and bioinformatic tools have enabled metabolomics study to become increasing comprehensive, robust, and informative for the understanding of disease, drug action, and the development of biomarker. Numerous metabolomics investigation include these: pharmacology, toxicology, stem cell research, oncology, and system biology have made use of cell metabolomics. Metabolomics aim to comprehensive map all biochemical reaction in given biological system, divided into two categories approach: targeted or untargeted. Targeted require prior knowledge of the molecule interaction to design tailored analytical method. However, the establishment of intensive targeted method has become facilitated by the increasing availability of metabolite libraries, IRO technology. Untargeted aim to detect as many metabolites as possible in a simple with subsequent and annotations, and identification usually involve database research. Example, amitriptyline or MS-dial, as well as comparison with authentic synthetic material. General biological sample, उसके बाद measurement and acquisition, data processing. After data processing, multi-variation data analysis, database search and identification, biological translation. Application of metabolomics and viral infection, biomarker, HIV disease, hepatitis B, metabolism to hepatitis C, SARS-CoV-2, bacteria infection, biomarker, metabolism to Clostridium difficile, tuberculosis approach, etc.
Sorry for the interruption, sir. Please conclude your presentation.
Yes, ma'am. Yes, ma'am. New development. Metabolomic drive experimenter known as stable isotope tracing uses stable isotope traces. Example, 2 hydrogen, 13 carbon, and 15 nitrogen incorporated in nutrients such as glucose and glutamine and provide dynamic representation of metabolism. Tracing experiments are often coupled with simulated metabolic network to enable metabolic flux analysis. मैम ये चार्ट है, ये रेफरेंस से ही लिया गया है इसी के. अप्रोच untargeted LC-MC-MS metabolomics, advantage discovery of unknown broad best possible coverage. Disadvantage, hardly quantitative metabolite identification remain a bottleneck. Example, XCMS, MS, dial, TOX, CM. Other behaviors. Metabolomic drug development. Traditionally, the first step in any drug development process involves the selection of promising target of disease modification, followed by extensive screening procedure for possible interaction. However, novel candidate targets are now frequently identified based on omics data, such as genomics or transcription 77 linking specific genetic alteration with disease phenotype 78, for example. Recently, so that carrier of the hydroxysteroid 70 beta dehydrogenase 79 gene variation have a reduced risk of non-alcoholic streptohepatitis and cirrhosis. Target identification by population wide screen and association frequently enter the drug development pipeline without prior knowledge of their biochemical function, mechanism of action, and T marker identification. In turn, metabolomics approach have taken center stage for obtain a detailed understanding of underlying biochemical process and enabling mechanism of action TA marker identification. Additionally, phenotype drug discovery, a target agonistic process in becoming wide adapted in the pharmaceutical industry 18.
Sorry
in
for the
contrast to,
Please conclude your presentation.
जी मैम
Please conclude your presentation, sir.
conclusion no present in this.
Okay, sir. Respected session chair, if you have any question regarding the presentation, you can ask to the presenter.
Yeah, I can. Okay, it's okay, no problem. You can continue with the next person. Thank you.
Thank you.
Okay, thank you, sir, for your presentation. Now let's move on to our next presenter, Tripti Padalia from Sardar Bhagwan Singh University.
Is my screen visible?
Yes, ma'am, it is.
Good afternoon, everyone. My name is Tripti Padalia, and my topic of presenting today is revolutionizing the spinal cord injury recovery, the next approach of neural stem cells in therapies. Introduction: spinal cord injury is a damage to the bundle of the nerves and the nerve fiber that send and receive the signal from the brain. The spinal cord extends from the lower part of the brain down to the lower body. Symptoms: numbness, tingling, loss of or change in the sensation in hands and feet, paralysis, pain and pressure in the neck, back, head, weakness in the part of the body, unnatural position of the spine, head, etc. Now the injury site and the loss of the function. Over here, we can see a complete diagram of our brain and our spinal cord. If we get an injury in the sacral section, then there will be some loss of function in the hips, leg, little to no movement of the ball and the bladder, impaired sexual functions, patient may still be able to walk. But if we get in the lumbar area, all the below will be occurring and there will be a very little chance of recovery for the walking. When it happens in the thoracic, all the control of the abdominal, chest, back muscles and all the listed below. When it happens in the cervical, the lower cervical, there will be loss of arms and hands. But when it happens in the higher cervical, total paralysis from the neck down, plus inability to breathe on their own and reduced ability to speak occurs. Now there are types of SCI. There are two types of SCI, incomplete and complete injury. In incomplete injury, the spinal cord is able to send some message from the brain, but people with incomplete injuries still have some feeling, sensation, muscle control between the site of their injury. In complete injury, there is no nerve communication between the injury site, muscle control, feeling or function below the injury is lost. Now the degree of severity is basically classified into these both. Grade A, which is complete injury, and the grade B, which is incomplete injury. Now the phases of SCI, primary, secondary, and chronic. Primary, the initial stage immediately after the injury is known as primary injury with the features of both fragment and spinal ligament tearing. It is incomplete and partial injury state as few segments remain connected to the axons. In secondary state, the secondary injury occurs when the body's own response to damage exacerbate the situation. In the case of the spinal injury, for example, the bone fragments not only damage neurons but also tear the surrounding blood vessels, leading to inflammation, further amplification of the harm caused. In chronic, during the chronic phase, the body response to the damage initiates a reparative process that involves the formation of scar. These scar, however, impede the connection between the neurons. Over here, we can see the MRI scans of all the three phases. In the first scan, we can see there is a bit of swelling and narrowing of the epidural region over here. In the second one, which is secondary phase, we can see inflammation. And in the third, we can see a scar forming. Now the stem cell. Stem cell represent the body's foundational material as they can develop into large range of different cells. Forms with specialized function with brain and heart, blood and muscles. Sometimes they repair damaged tissues or self-renovate, becoming the new stem cells. Now the two key features of the stem cell is they can divide into more, like mitosis and to form more cells, and ability to differentiate into specialized cells. The stem cells can change broadly into 200 different cells. Now the stem cell therapy. The strategy of the transplanting stem cell in the treatment of SCI focus on the replacement of the lost and the damaged cells, especially neurons and oligodendrocytes, provision for tropic support of neurons, manipulation of damaged spinal cord environment to facilitate axon regeneration. Neural stem cells. A very large number of axons extend to over very long distance through the injured spinal cord and form new neural relays across the site of severe SCI. Over here, we can see a diagram where we can see the red lines that denote the wire that connects the nervous system is cut due to the injury over here. And in the next one, we can see the green. The green lines are the stem cells that enable the regeneration of the injury of the axon in the spinal cord, and it will extend and form new axons and connect the area below the injury. This is the concept to form the new relays across the site of the injury of neural stem cells. Now, why do we choose neural stem cells? Well, they are developing cells in the high growth rate. They can fill the cavity in the injury and form a physical tissue bridge connecting the ends of the injured spinal cord. They are unique environment that allows injured host axons to regenerate into this injury site. They can extend new connection axons below the injury to reconnect the spinal cord below the injury. They can bridge the gap and splice the circuit. The experimental result, the neural stem cells of a human was tested in an animal model that was a rat. So what they did was they took a rat and they spliced the rat spinal cord at T3. This is the control rat. There was nothing changes happened after the splicing. But when they applied human neural stem cells, so what they saw was there was connectivity and all axons were producing and all the connections were growing back. And now we can see the gap was like from the green. The green is basically genetically modified neural stem cells because it was used for checking wherever they are growing and all that. And it was genetically modified with the protein, green fluorescent protein. The future perspective, the reparative efficacy of implementing such strategies in SCI repair needs further verification. Secondly, differentiation control of NSCs in the spinal cord is needed. Current results have shown that the transplant NSCs mostly differentiate into other instead of the neurons and elongate enterocytes. This restricts the regenerative effect of the NSCs. Conclusion, the NSC is severe as it is an important constituent of the cell transplantation therapy for SCI. Their therapeutic potential establishes the basic of NSC therapies. Many strategies have been designed in the preclinical testing with the promising results. However, more work is needed to achieve the full recovery and translate into clinical process. Thank you.
Thank you for your presentation, ma'am. Dear session chairs, if you have any question regarding the presentation, you can ask to the presenter.
What is the chance of recovery after, how long it takes for regeneration of this?
Ma'am, in this animal experiment, it took about 2 to 3 weeks and months to get complete, not complete, but 80% of the recovery. And there was this case in a human, but what it happened was in the primary state, like over here, the primary, right? The very initial stage when the patient got injured, he went to the hospital and they tried this grafting. It was done in, I guess, US, somewhere in US. And what happened was after grafting this neural stem cells and exercise and all, he gained about like 60% recover, 60% complete motion of his body. And like it took 3 to 6 months for him. Along with some immunosuppressants and naproxen for reducing the spinal pain.
But not so many cases are done on human. Only this is on the animal as of now.
It is like very rare because there is also, we can use MSCs also, like some clinical trial studies has been done on MSCs, which is mesenchymal stem cell or stromatal cells, which is more effective and easily like available. Because the neural cells, it relies on the extraction from the embryonic tissues and then its evolution into neural stem cells. So it is like very rarely studied because firstly, when you graft it on the human or animal, first we have to check it if it is survivable. Like I can graft neural stem cells in the patient, but if it is showing the effect or it is growing or not, that is the very first challenge. And to maintain and to do all this testing, like very less laboratories and doctors have tried it.
Okay, thank you.
Thank you, ma'am.
Thank you for your center. Veronica Sadar Bhagwan Singh University.
I was older, so I thought like
Good afternoon, everyone. Hello.
Yes, ma'am, you're audible. Good afternoon.
Is my slide is visible?
Yes,
yeah, it is visible. You can proceed.
Okay, thank you. Good afternoon, everyone. Myself, Vandika, and I am from Sardar Bhagwan Singh University. And my topic is artificial intelligence and machine learning. The content includes introduction. In introduction part, the brief intro of artificial intelligence, machine learning, and deep learning. And the second is artificial intelligence and their types. Third, approaches, elements, applications. Fourth, problem of artificial intelligence and challenges. Fifth, advantages and disadvantages of artificial intelligence and artificial intelligence uses. Seventh, machine learning. Eight, type of machine learning. 9, challenges and limitations of machine learning. And the last one is application. So the introduction part, first one, artificial intelligence. Any technique that enables computers to mimic human behavior is called artificial intelligence. The second is machine learning, a subset of artificial intelligence techniques that use the statistical methods to enable machines to improve with experiences. And the third one is deep learning, subset of machine learning, which makes the computations of multi-layer neural networks possible. So in this figure, the subtypes of artificial intelligence like machine learning and deep learning. And deep learning includes big data, data mining, data analysis. And the big data and the study of big data, data mining, and data analysis is called data science. So in the next slide, artificial intelligence. So what is artificial intelligence? Mainly artificial intelligence, the simulation of human intelligence processed by machines, especially computer system. And it also, artificial intelligence is a machine ability to perform the quantitative functions we associate with human minds, like such as perceiving, reasoning, learning, interacting with an environment, problem solving, and even exercising creativity. So in this slide, artificial intelligence, like intelligence of machines and the branch of computer science that aims to create it. So the types of artificial intelligence are: type 1, narrow artificial intelligence. So in narrow artificial intelligence or weak artificial intelligence, it includes narrow and predefined constraints, focused on single and limited tasks, uses natural language processing, and mainly for decision making. For example, speech or face recognition. The second, in type 1, the second is general artificial intelligence. General artificial intelligence is also known as strong artificial intelligence, or it includes mimics the human intelligence, learn and embedded human intelligence, or uses theory of minds artificial intelligence. For example, Fujitsu builds K supercomputers. Now in type 2, reactive machines, also called machine-less artificial intelligence. It includes memory-less, like no storage of inputs. No storage of inputs, reactive, like responding predefined inputs, statics, like no learning and performed. And reactive machines, it is the oldest form of artificial intelligence, like simple architectures. So the next is limited memory artificial intelligence. It includes learning through experiences, combination like react and learn, training like manual or automatic. For example, biometric person identification machines. The third one is a theory of mind, includes try to simulate like human emotions, current status like still in research phase. And the example is self-driving cars. Now the last one is self-awareness. Still theoretical idea and simulate human brains. Now the elements, there is a 3 elements of artificial intelligence, like massive amount of data, sophisticated algorithms, high performance parallel process. And the 3 steps are computers and programs, the Turing test, the Diamond conference. The approaches include cybernetics, symbolic, statistical, or integrated the approaches. Applications are healthcare and medicines, automotive, finance and economic, video games, and heavy industries. Problems of artificial intelligence and challenges: reasoning, problem solving, knowledge representation, planning, learning social intelligence, natural language processing, general intelligence, perception, motion manipulation. In the next slide, the advantages of artificial intelligence are: discussion taken by, first one is discussion taken by machine, which reduce room for error. The second is the machine does not get tired. It can work without taking breaks. Third, data identification and data analysis. Fourth, unbiasedness. And fifth one is the ability to take right decisions in a short span of time. On the other hand, the disadvantages of artificial intelligence, like ethical issues, of emotions and creativity, implementation cost, increased unemployment rate, lack of creativity. Where is artificial intelligence used? like in customer experience, supply chain, predictive analysis, real-time operation management, human resources, fraud detection, research and development, customer insights, knowledge creations, customer services, pricing and promotions, risk management and analytics. Now, what is machine learning? A branch of artificial intelligence concerned with the design and development of algorithms that allow computer to involve behaviors based on empirical data. As intelligence required knowledge, computer must acquire knowledge. Machine learning refers to a system capable of the autonomous acquisitions and integrations of knowledge. Why machine learning? Because of no human experts, black box human expertise, rapidly changing phenomena, need for customization and personalization. The types of machine learning are supervised learning, unsupervised learning, and reinforcement learning. The first one is supervised learning. So in supervised learning, also known as supervised machine learning, and it is defined by its use of labeled data sets to train algorithms that to classify data to predict outcomes accurately. Like example, used to predict house prices. So it includes classification like fraud detection, email spam detection, diagnostics, image classification. And the second one is regression, risk assessment, score predictions. Now, in unsupervised learning, it includes like that the training data is unlabeled and the system tries to learn without a teacher. First one is dimensionality reduction, text mining, face recognition, big data visualization, image recognition. And the second is clustering, biology, city planning, targeted marketing. The third one is reinforcement learning. It is a very different beast. It includes observe the environment, select and perform actions. For example, many robots implement reinforcement learning algorithm to learn how to walk. And the DeepMind's AlphaGo program is also a good example of reinforcement learning. It includes gaming, finance sectors, manufacturing, inventory management, or robot navigations. So in this, supervised learning, like in inputs, data with labels, and in outputs, mapping. As usual, unsupervised learning, and in the reinforcement learning. Challenges and limitations of machine learning. In the challenges easily identifies trends and patterns, no human interventions needed, continuous improvement, handling multi-dimensional and multi-varieties data, wide applications. And the limitations, high error and susceptibility, data acquisitions, interpretation results, time and resources. And the application of machine learnings are face detection, object detection and recognition, image segmentation, multimedia events detection, economical and commercial usage. Thank you.
Thank you for your presentation, ma'am. Our respected session chair, if you have any question regarding the presentation, you can ask her.
No, thank you.
Thank you, ma'am. Thank you for your presentation. Now let's move on to our next presenter, MD Ghulam Ghaus from Sardar Bhagwan Singh University. So your voice is not audible.
Hello.
Please.
Hello. Good morning, ma'am.
Morning,
Good
sir. Please.
afternoon, ma'am. Today is my topic in advance in precision medicine. Precision, precision medicine approaches the healthcare by the tailoring the medicine decisions, healthcare and treatment and interventions to the individual characteristic for each patient, and this includes that the considering the genetic environment and lifestyle factors to provide the personalized care. This is my contents. In content includes the introduction, principles, primary goal, population-based protocol, precision medicine, and genomic sequencing technology, evaluation of a precision medicine, and the challenges of the precision medicines. In precision medicine stands that are deeply transform the healthcare by tailoring the care precisions to individualize the taking into the account such as a personal characteristic and ethnicities, local environment and exposures. Such as if there is
Excuse
a
me, I
particular.
request all participants to mute themselves. Yeah, you can proceed.
Okay, okay. That exactly approaches to the care with a leverage that the big data of a precision medicines of a genomic that increases the improbabilities of help the identify the most of the appropriate cost-effective treatment. Even precision medicine derives into the unique genetic makeup of individuals to
Please
guides
mute.
of a medical decision.
Please mute your background voice.
This is my principles of the precision medicine. This is the effective of a care by by preventing medicines and new taxonomy and cost effectives. In principle, ineffective of the care principle, a physician do not fully understand how the certain treatments will affect at a particular patients. With a precision medicine, medical provider can apply the personalized treatment methods and personalized treatment methods for each of the patients. This includes the genomic understanding, utilizing genomic and informations to identify genetic variation that influence an individual health, such as a disease risk and response to the treatments. And in the preventive medicines, early diagnosis of the genetic cause disease is a possible through the genetic screening methods that integrate the data from targeting therapies and patient involvements. And new taxonomy is a medical process and new classification for disease and characterization with a modeling, early detection and prevention and continuous learning with the cost-effective treatments. And these include the precision targeting therapies based on the molecular diagnosis. In precision molecular diagnosis, precision medicine is a science. The primary goal. The goal of the precision medicine label to improve the health and treat disease by the tailoring treatment to individuals pick up where population and personalized approaches evolve. develop more and better treatment for cancers, and create a national voluntary research cohorts. And the precision medicines is to provide the more personalized and effective healthcare by tailoring medicine decision, treatments, and interventions to the unique characteristic of each individuals. By integrating genetic environments and the lifestyle informations, precision medicine aims to enhance the accuracy and diagnosis and identify optimal treatment strategies and minimize adverse effect. Precision medicine moves to the precision medicine move care for population-based protocols to truly individualized. Precision medicines move care from the population-based protocols to truly individualized medicine presents. In Obama 2015, Obama announced in the precision medicine initiatives in the United States. And then under the initiatives, medical care would transist from one side fits all approaches to an individual approaches with fit to the data of a patient genomic makeup, environment, and lifestyles, help the medicine processes, tailor treatment, and prevention strategies. In precision medicines, moves care from the population-based protocols to individualize medicines to achieve the precision. By the principle of interdisciplinary collaboration, encouraging the collaboration among the healthcare profession and researcher and data scientists and other stakeholders to integrate and advance in the field of precision and collectively to enables the era of medicine through the pharmacology and the policies that empower the patient researchers and provides to work towards the mission statements. Also, precision medicine moves to the population-based towards protocols to truly individualized medicines. The population's personalization, precision, quantum, healthy in the patient involvements, prediction modeling, and data integrations. Precision medicine also involved in a population health to promote the researchers in population science to fulfill the promise of a precision treatment and prevention in reducing the burden and cancer. Identify opportunities for the precision medicines to reduce the population burden of cancers. And secondly, the conduct population research to discover and evaluate the precision application along the cancer quantum implement in the cancer program. Evidence-based precision tool and applications. Genomic sequencing technology. In genomic sequencing technologies, researchers and clinicians who aggregate the most genomic exosomes and rich patient clinical history with advancement of machine learning technology. With the help of these outcomes, the precision medicine tailoring cares to the more effective treatments and prevent the disease require significantly and robust the data by the clinician and research by utilizing the genomic information to identify genome variation that influence the individual health and disease risk and response to treatments, and also helps to the personalized treatment, designing treatment and intervention based on the specific genetic patients to maximize the effectiveness and minimum adverse effects. This is the evolution in the precision medicines. Evolution in the precision medicine, these are the principles, forces shaping the evolution of the precision medicines. That's first in engineering and device-based medicines. That is the wearables, sensors, smart implants, and this is a remote health monitoring, relevance of a robotics, is a telemedicine and robotics. And second one is a molecular precision medicine, is a panomics profiling analysis of a disruption in a biological network. And third is the information based healthcare. And these are the challenges which which is faced during the precision medicine. The challenges overcomes to the precision as follows. Is marked the ongoing innumerable economic, regulatory, social, and a technical issue that needed a novel solution. Convincing, evidently,
Sorry for the
support
interruption, sir. Please
precision
continue
medicine.
the presentation.
Precision medicines, more research needs to make the case of a precision medicine adaptation, which would lead to the significantly improve the outcome. And that the significant advancement of the precision medicine is marked by the ongoing development in the technology, data analysis, and the understanding of the genomic. Education is the key challenge because of many physicians expresses little confidence in their the ability of the make of a genetic and genomic information is important. Educational efforts over the difference of discipline and medicine are needed. Thank
Thank you for your presentation, sir. Respected session chair, if you have any questions regarding the presentation, you can ask to the presenter.
Do you think this precision medicine will work out in India, country like ours?
Ma'am, nowadays they take 5 to 10 years between the precision medicines also work in India also.
Because still I have not heard of people using this technique for the treatment. As a precision medicine, personalized treatment has not yet seen.
But so, ma'am, please, in precision medicine, work on going on because lots of the technologies has also come with the advancement of the after the advancement announcement of the of Obama in 2015. The initiations was taken from in precision medicine also.
Okay, thank you.
Thank you for your presentation, sir. Now let's call upon our next presenter, S. Pavitran from Government Arts College.
Audible, ma'am? Good afternoon, ma'am. Am I audible?
Yes, sir, you are audible.
Good afternoon, everyone. Myself is Pavitran, research scholar. I'm pursuing PhD under the guidance of Dr. B.S. Sujata, ma'am, Associate Professor, Department of Zoology, Government Arts College. Here, I would like to present my research work on evaluation of honey, curcumin, and ethanolic flower extract from Lagersomia speciosa on excision wound healing and anti-inflammatory activities in experimental albino visitor rats. And starts from the introduction. The wound care and management of wounds is a complicated program, and research on drugs that increase wound healing in the developing area.
Screen is not visible. Please enlarge your screen.
Sure, ma'am. Excuse me, ma'am. Sorry for the inconvenience. I'm start from the introductions. The wound care and management of wounds is a complicated program, and the research on drugs that increases wound healing is a developing area in modern biomedical and pharmaceutical sciences. Every human being will experience a wound at some point in their lives. The wounds are results from physical or chemical injury or microbial infections. The healing is a repairing process of the tissues from the damage. Generally, the plant origin drugs have enormous therapeutic potential, which are due to the presence of certain active metabolites. They are called as the secondary metabolites. I select the plant L. speciosa as a research plant for my study. It belongs to the family Lythraceae. It is popularly known as bride of India. It has been traditionally used in folk medicine as a remedy for some illnesses. The objective of the present study is to evaluate the wound healing and anti-inflammatory properties of ethanolic flower extract of L. speciosa. It's aiming to confirm their ethnotherapeutic claims for treating injuries. This figure shows my research plant Lagerstomia speciosa. Next time, move on to the materials and methods. The plant L. speciosa collected from the PG Girls Hostel, Government Arts College, Coimbatore, and it were identified and authenticated at Botanical Survey of India, Coimbatore. The figure 2 illustrates the extract preparation methods. The collected samples were shade dried at room temperature, and the dried samples were powdered with the help of electric blender, and then powdered was extracted using the solvent ethanol. The 100 gram of powdered sample was soaked in 1 liter of solvent ethanol and kept for 4 days with periodic shaking. After that, the extract was filtered using Wortmann #1 filter paper and dried at room temperature. It was reported by Kunkatif 1994. Next, the phytochemical analysis. The qualitative phytochemical analysis of L. speciosa flower extracts was carried out according to the methodology of Harbon and Trias Evans. The GCMS analysis was conducted at the South Indian Textile Research Association, Coimbatore. It were identified and authenticated in some important compounds from the GCMS analysis. The ointment preparation. The ointment was prepared followed by the fusion method. The flower extract was prepared as an ointment using 10% petroleum jelly, and the ointment was kept in a sterile glass container and preserved at 4 degrees Celsius and used for the topical application. It was reported by I.M. Pillai et al. 2010. Next, experimental animal. I chose the male Western albino rat as a model animal for my present research. All the experimental procedures were approved by the Institutional Animal Ethical Committee, KMCH College of Pharmacy, Coimbatore. The skin irritation test followed by OECD 425 guidelines. The dorsal fur of the animals were shaved with an electric clipper, and the prepared 1% and 2% flower extracts topically applied on the animal skin. From the observation, there is no edema or irritations are formed. The excision wound creation method. The animals were anesthetized using ketamine hydrochloride, 100 mg per kg. A full thickness of wound circular area 2 into 2 centimeter was marked on the shaved skin, and the marked area was left open to create the excision wound model. It's reported by Martin and Malone, 1972. The figure 3 shows the excision wound creation methodology. The figure 4 shows the experimental design for wound healing activity. The group 1 is a control. They did not receive any treatment. Group 2 and 3 receives povidone iodine and neomycin respectively. These are the commercial available ointments topically applied. The group 4 and 5 receives the honey and curcumin 2% respectively. These are the traditional medicines followed from the ancient people. The group 6 and 7 receives 1% and 2% ethanolic flower extracts topically applied. Let's move on to the results. The results 1 is a phytochemical analysis. The table 1 shows the phytocompounds from the qualitative phytochemical analysis of the L. speciosa extract. It has almost all the active metabolites in the flower extracts like alkaloid, flavonoid, saponins, phenols, tannins, protein and amino acids, reducing sugars, steroids, glycosides, and phytosterols. The result 2 is a GCMS analysis. The table 2 shows the important compounds identified from the GCMS analysis of ethanolic flower extract of L. speciosa. The perfurol and the pentatecnol are the peak component of GCMS analysis. The result 3 is the wound healing activity. The table 3 shows the statistical justification of wound contraction rate of L. speciosa ethanolic flower extracts. The wound healing activity was measured by to calculate the wound contraction rate. From this table, on day 17 to 21st day contraction rate was highest in the 2% flower extract treated groups compared to the control as well as the neomycin ointment. From the results, the ethanolic flower extract significantly increasing the wound contraction. The figure 4 shows the day-wise wound contraction and the excision wound model. The conclusion, the plants are effective wound healer and they repair wounds in a natural way. The plant-derived natural compounds are significant source of medicinal agents for new drug design. The many bioactive compounds are the main source of medicinal plant. The plant L. speciosa consists of phenolic compounds like tannins and flavonoids are responsible for faster wound healing. The phenolic compounds stimulate the platelet growth factor in the late inflammatory phase. It enhances the healing mechanism. And finally, the futuristic vision of my research is focus on developing an advanced wound healing technologies like to make a smart herbal bandages or dressing that can actively monitor and facilitate the healing process. Thank you, ma'am.
Thank you for your presentation, sir. Our respected session chairs, if we have any questions regarding the presentation, you can ask.
See, the marketed ointment will have different composition, but you have prepared in liquid paraffin jelly, correct?
Yes, ma'am.
Then how will you see how you have compared? Because marketed ointments will not have paraffin jelly only; they'll have other constituents also, no?
Yes, ma'am. The marketed ointments have simple compositions. Based on the compositions, I'm selecting the petroleum jelly for making the ointment preparation, ma'am.
Only petroleum jelly will not be there only. Please refer properly. And one more thing for curcumin, why you have taken curcumin?
Actually, I'm choosing curcumin and honey is traditional medicines. Whenever we meet an cut or a wound, the people go and put a turmeric powder for the primary.
That I understood. You can go to your section of result. Yeah. You said 1% is better. 1% this is better when compared to the control.
No, ma'am, 2%.
Sorry, 2%.
2%, yes, ma'am.
But there is no much difference in the contraction of the wound, no? Even curcumin, you're showing 84.4% and 93% you're showing. And that too, it is working after the later stage, not in the beginning stage. There is no much difference, correct?
Yes, ma'am. Yes.
Okay, fine. Thank you.
Okay, ma'am. Thank you.
Thank you for your presentation, sir. Now let's move on to our next presenter, D. Niveta from Government Arts College. Niveta is unavailable at the moment, so now let's move on to our next presenter, A. Benshira from Government Arts College.
Good afternoon, ma'am.
Good afternoon, ma'am.
Shall I present my?
Yeah, you can proceed.
Ma'am, anti-cholesterol effect of methanolic red leaf extract of Legostomia speciosa against Gengus carticus syrup, which is ingested to the albino rat, is my topic. We all know that cholesterol nowadays, cholesterol is a waxy substance which found in our blood high cholesterol develops fatty deposits in blood vessels. Eventually, these deposits grow, making it enough blood to flow through arteries. So it causes strokes and heart attacks. So a person is considered to be hypercholesterolemia, that is high level of cholesterol, which when certain levels are exceeded. So it causes the high cardiovascular diseases. Then next, Gengus cardicus herbal syrup, which is the medicine we used. So Gengus cardicus herbal syrup, which reduces blood cholesterol and cleanses cardiac cells. It maintains a heart artery cleanse and protection by support strong arteries from clogging by plaque and ensure effective blood flow. Here, medicinal plants have been used over centuries. So for remedies and diseases, anti-cholesterol of plant origin have enormous therapeutic potential, which are due to the presence of many important secondary metabolites. It had anti-cholesterol activity compounds like alkaloids, phenols, flavonoids, reducing sugars, glycosides, phytosterols, steroids, and tannins. So the objectives of studies, the aim of the study is to evaluate the anti-hypercholesterolemic effect of methanolic red leaf extract of Legasthemia speciosa. So to find out the beneficiary plant constituents in Legasthemia, to identify the chemical constituents and medicinal value present in the Legasthemia speciosa. And through this experiment, we found out the medicinal properties of compounds present in the red leaves of Legasthemia speciosa. To determine the pharmaceutical effect of Legasthemia speciosa against the Gengus cardicus syrup in lowering of blood cholesterol levels. Sorry, ma'am. So here how the extract is prepared and where the plant is. The plant is collected at PG Girls Hostel Government Arts College, Racecourse, Coimbatore. So which is scientist Dr. M.U. Sharif, Botanical Survey of India, Tamil Nadu, TNAU, Coimbatore, and which the exact preparation, how the extract is prepared is attractive red leaves of Legasthemia speciosa is washed and then they are powdered and sieved. Then the sample of 100 gram per 1000 ml is prepared, and the powdered plant is cleaned, glass, and stored in methanol for 3 days. The whole mixture then went under filtration by a piece of clean white cotton cloth, and the solvent of the extract was reduced under room temperature and stored. Here the phytochemical analysis, the quality analysis, extraction of the compounds reveals that it contains the many important metabolites such as flavonoids, glycosides, reducing sugars, tannins, steroids, phenols, saponins, alkaloids, phytosteroids, amino acids, and proteins. Here are the experimental plant extraction methods in picturization. Next is the phytochemical red leaf extract. What are the compounds and how much they are present in the leaves? Here alkaloids, flavonoids, saponins, phenols, tannins, amino acids, and proteins, reducing sugars, steroids, glycosides, phytosterols. Next, experimental animal model. We have selected some animal albino rats. Human being is selected for the present study. We just strain albino male rats, which is weighing about 120 to 130 gram, are selected. Their house is in a well ventilated temperature and it contains 12 hours light and dark circles are given. So next is the acute oral toxicity study. Immediately after dosing the rats, we observed continuously for first 6 hours of any toxicity signs, that the skin, fur, eyes, mucous membrane, or circulatory, respiratory, or changes in behavior pattern. They were then kept under observation of 5 days of the experimental design and the
good morning, madam. My name is Muhammad Ishal. So mine is in the main hall at 120 presentation.
Sir, please.
Main hall, right?
Sir, please give a few minutes. I will get back to you.
Main hall means this is the going on now.
Yes, sir. Yes, sir. Please mute yourself. We'll call your name.
Now Benshina is presenting.
Yes, sir. Yes, sir. Please wait a few minutes.
No. In the main hall, now the candidate name is Benshina presenting, right? That is the hall, right?
Yes, sir. Yes, sir. We'll connect with you soon, sir.
Benshina presenting now, right? That is the hall, right?
Yes, sir. Yes, sir. Please, we'll call your name when it's your turn. So please mute yourself.
So no need to enter to breakout hall.
No, sir. You can stay here.
Okay. Now,
Sorry for-
after that, mine will start or no?
Yes, sir, we'll inform you
Yeah,
when
but
the...
sir.
Sorry.
Yeah, Dilan seminar la. So I will wait for my turn, right?
Yes, sir, yes, sir. Please wait for your turn. Sorry for the interruption, ma'am.
Ma'am,
Yeah, you can.
Okay, ma'am. This is the experimental designs. Next is first animal, that is animal served as control group, which is administered with a 0.5 ml distilled water, and group 2 animals received low dose Gengus cardiacus syrup with a 5 ml per kilogram of their body weight, and group 3 animals received high dose Gengus cardiacus syrup of their body weight, and animals received low dose MRLE 250 mg per kilogram, and animal received high dose MRLE 500 mg per kilogram of their body weight. These are the experimental design of our. Next is the result. They conduct acute toxicity and biochemical analysis. Acute toxicity, the crude methanol extract is non-toxic for the rats, so they are tolerated. They are tolerated 250 to 500. Sorry, ma'am. 500 mg per kilogram. No biochemical and histological changes were recorded. In acute toxicity study, no mortality or toxicity reactions was recorded in rats after administration the methanolic crude extract of Legostomia. Red leaf concentration 250 and 500 mg per kilogram. No death occurs during the toxic studies. Next, biochemical analysis. Levels of biochemical parameters were significantly lower in the pre-treated with the L. speciosa. The groups of rats pre-treated with L. speciosa extract demonstrated, and it contains elevation of biochemical parameters with 4 and 5. The sample of syrup J. Gengus cardiacus treated group 1 and 3 shows significant low level of cholesterol compared to the control group. So the heart rate also significantly reduced to that experiment. Next, conclusion is cholesterol is an essential constituents of biological membranes, and acting as a precursor for the synthesis of bile acids, hormones, and vitamins. High circulating cholesterol is associated with a hypercholesterolemia, atherosclerosis, and stroke, and increases the risk of cardiovascular diseases, fatty liver, and carcinogenesis. The medicinal plant Legostomia speciosa appear to be rich in secondary metabolites widely used in traditional medicine to combat and cure various ailments. So the finding of the study revealed that Legostomia speciosa red leaf extract are having anti-cholesterol properties with a dose 500 mg per kilogram, and anti-cholesterol effect is not sufficient with 250 mg per kilogram. The anti-cholesterol effect of methanolic red leaf extract of L. speciosa could be due to the presence of their compounds containing flavonoids, glycosides, and phenols. Therefore, L. speciosa could be a potential herbal medicine as adjuvant with existing therapy for the treatment of hyperlipidemia. The present work provides, as a result, conclusive scientific evidence for traditional use of L. speciosa in treatment of anti-cholesterol activity. Thank you, ma'am.
Thank you for your presentation, ma'am. Our respected session. If you have any question to the presenter, you can ask.
No, you have told that you have done an acute toxicity study with 250 mg and 500 mg. Which guidelines you have followed? Hello?
Hello, ma'am. Hello, ma'am. 500 mg, ma'am.
No, which guideline you have followed? This is not the standard guideline. And what parameters you have assessed?
Am I audible, ma'am?
What parameters you have assessed that details you have not given, no? You have not shown the graph, what reduction has happened and what has happened.
Yes, ma'am. Actually, 1%. Hello.
Yeah, you tell that.
Yeah, ma'am. The parameters. Ma'am, actually, the medicine is given to and it that the heart rate reduced the cholesterol level and the heart rate also reduced during their experiment, ma'am. We point something like 0.1% of the medicine is treated with that, ma'am.
Okay, thank you.
Thank you for your presentation,
Okay,
ma'am.
ma'am.
Let's move on to our next presenter, Dr. V. Shalini from National Institute of Siddha.
Chudu.
Is my slide is visible, ma'am? Hello.
Yes, ma'am, it is visible.
A warm greetings to one and all gathering here. I am Dr. V. Shalini, third year PG scholar, Department of Gunapadam. I would like to present my topic, physicochemical characterization of Siddha herbometallic formulation Nagaparpam. Siddha system of medicine is one of the ancient system of medicine. Rising popularity of Siddha medicine in recent years have increased the demand of Siddha preparations. A parpam means an ash obtained through incineration. A metallic parpams are highly valued and have their own importance in Siddha formulations. Nagaparpam is such a herbometallic formulation used in Siddha system of medicine for the treatment of various inflammatory conditions like hemorrhoids, fistula, and ano associated with the pain. The present study was performed to characterize the quality control parameters for the Nagaparpam by using sensitive tools and advanced techniques. This is the method of preparation of Nagaparpam. The ingredients of Nagaparpam has Nagam, Vale rasam, that means purified mercury. The Uttamani, that means Percloria damia leaf juice, required quantity. And the Tutti, that means Aptilon indicums leaf juice, required quantity. This is the picture. The Nagam, that means zinc, that rasam, that means mercury, purified mercury. And the Uttamani, that means Percloria damia, Tutti, and Aptilon indicum. The raw drugs are identified and authenticated by the experts in National Institute of Siddha. The authentication numbers of the herbals and metals are denoted. The method of preparation of Nagaparpam. The purified Nagam was taken and put into the ulai stirred with the whole plant of Percloria damia and until it becomes parpam. And the calcinate Nagam and a half amount of the Vale rasam with Percloria damia leaf juice for 6 hours and made into villai. That means cake. That it allows to dry in sunlight, and the villai was covered with Aptilon indicums leaf juice and paste, and it was subjected to calcination process with the cow dung cakes. The process is repeated 2 more times. Finally, the test drug Nagaparpam was collected with the help of surrounded and stored in airtight glass container. The last picture is the final product. These are the indications of Nagaparpam. In this presentation, physicochemical analysis of Nagaparpam was analyzed using as per the Ayush testing protocol parameters, the organoleptic characters, and the physicochemical analysis. The XRD, semi-DUX, and ICP-OS methods were analyzed. The organoleptic characters of Nagaparpam is state solid and odor is odorless, color gray, the taste bitter and shape powder, touch soft. The physicochemical analysis of Nagaparpam is LOD, a loss on drying 105 degrees Celsius is 0.43% and total ash 99.13% and water soluble ash 2.20% and acid insoluble ash 2.09% and pH of the Nagaparpam is 9.43%. The fineness of particle is 99.86% pass through mesh is 100 in sim. In the XRD, the graph of the XRD shows a peak in the zincite. The composition of zincite is 100%. The semi-DUX shows the graph in oxide in very peak level. This oxide is 68.31%. The report of this semi-DUX analysis confirm the presence of potassium, calcium, zinc, iron, and traces of mercury and lead-leading compounds. Lead-containing compounds, and the size varies from 5 micrograms. The elemental analysis of ICP-OS shows arsenic, cadmium, mercury, lead is below detector level, and the zinc is 74.88%. These are the graphical representation of ICP-OS of Nagaparpam. Coming to the discussion part, standardization of parpam is utmost necessary to confirm its identity and to determine its quality, purity, safety, effectiveness, and acceptability of the product. The crystallographic structure of the Nagaparpam shows the zinc comfort is 100%. The Nagaparpam elemental analysis shows the zinc has 74.88%. The morphological of Nagaparpam in semi-dux shows size ranges from 5 micrometer. Although the particle sizes of different batches showed the similarity, it seems that these particles are aggregated of much smaller particles. And to the final, the conclusion part, so Nagaparpam is complex material. Physicochemical analysis through modern techniques will be most attractive for the standardization of Siddha medicine. This study would be definitely help in building confidence in use of such a herbometallic formulation on Nagaparpam for medication by ensuring safety, quality, and efficacy. These are the references. Thank you. And finally, Siddha is a bundle of information that has to be explored and explained for the understanding of Siddha's knowledge. Thank you for the patience. Thank you.
Thank you for your
No, this which condition you're using this in your lab? You have tested anything, or you have given to the patients?
Ma'am, this is the preclinical study we have to done in SCRA lab, ma'am. SCRA Arumbakam lab, ma'am.
Okay, fine. But this must be used by the Siddha people, no?
Yes, ma'am.
Parapam is used, no, frequently.
Yes,
But you are mixing with
yes, ma'am.
This you are using this zinc-based formulation in the Siddha medicine, no?
Yes, ma'am. We are using, ma'am.
So what condition they are using? But the percentage is so high.
Ma'am, this is we are using only zinc, ma'am. That's only reason the percentage was high, ma'am. We are using these medicines for piles, fistula, that conditions, inflammatory conditions are associated with pain. We have used this medicine, ma'am.
Okay, fine. Thank you.
Oh, thank you, ma'am.
Thank you for your presentation, ma'am. Now let's move on to our next presenter, Pedipa P. from Chettinad School of Pharmaceutical Science. Prathipa is unavailable at the moment. So now let's move on to our next presenter, CBS from Chettinad School of Pharmaceutical Science. CB is unavailable at the moment. So now let's move on to our next presenter.
Hello, ma'am.
Yes, ma'am.
Hello, ma'am.
You're audible, ma'am.
Yes, madam. Hello.
Yes, ma'am, you're audible.
Ma'am, am I audible?
Yes, ma'am, you are.
Is my slide visible?
Yes, ma'am, it is.
Yes. Myself, I am Sebi. Here I am here with my oral presentation entitled The Cytotoxic Evaluation of Samsvera sufruticosa in APR leaves. Cancer. Cancer is the second leading cause of death. That can cause 3.9 million people's death. And here I am here to evaluate the anti-cancer or the cytotoxic potential of the plant Sansevieria subfruticosa. My aim is to evaluate the cytotoxic potential of Sansevieria subfruticosa NEBR leaves. My objective is to prepare the alcoholic extract of Sansevieria subfruticosa NEBR leaves via maceration and to perform a fractionation to estimate the in vitro cytotoxic activity via brine shrimp lethality test.
1 second, you can do full screen, no?
NTD studies, and these are my literatures.
Miss V, please
Yes, madam.
do slideshow, no?
Yes, madam.
Please do slideshow.
Okay, madam. Okay, these are my literature reviews, which actually telling about this cytotoxic potential of some of the crude extracts of Sansevieria subfruticosa. This also, some of my studies also confirms the presence of some promising antioxidant as well as pro-oxidant potentials in the same plant. This is the methods that I followed. Then this is a coarse powder that I used. Then I did the maceration followed by the rotary evaporation. I got different crude extracts, which I did the fractionation with the solvents like hexane, dichloromethane, ethyl acetate, ethanol, and water. Then I did the phytochemical screening of the plant, which I got the total ethanolic extract, which I got ethanol, alkaloid, phenolic, flavonoid, then carbohydrate, proteins, terpenoids, and sterols, then N-hexenes, which give the presence of phenolics and flavonoids, then dichloromethane, which give alkaloids, then ethyl acetate, which give alkaloid, phenols, flavonoids, carbohydrates, terpenes, and sterols, whereas the water fraction gives the phenolics, flavonoids, carbohydrates, proteins, and terpenes. I also evaluated the total phenolic and the flavonoid content of the plant, which actually give the results. The maximum extractable total flavonoid and phenolics were found in the ethyl acetate fraction followed by the total ethanolic extract. Next, the important study that I did was the brine shrimp lethality assay. This was done in the varying concentrations of my extract. That is from 1000 to 1 microgram. And I calculated the percentage mortality for the extracts, the hexane, ethyl acetate, dichloromethane, and total ethanolic extract. Then the LC50 values were calculated, and thus were found to be, the promising activities were found to be from hexane, that is 130 microgram per ml, and for total ethanolic extract that was found to be 110 microgram per ml. The next study that I did was the anti-mitotic study. The extracts selected were the same as that of the above. The study was compared with the Wien-Christine as a standard. Then parameters were observed where the root length and the root number. All groups exhibited a marked decrease in the root number and the root length in compared with the control. Among all the extracts, the total ethanolic extracts showed the maximum activity. The mean number of dividing cells were the next parameter which was noted. The graphs were actually plotted, which actually I didn't put it here. And that was between the concentration and the percentage inhibition, which helped me to find out the IC50 values. The IC50 values were calculated, and that was found to be 101 for the total ethanolic extract, and followed by the hexane extract, which shown 0.26 microgram per ml. The next study I performed was the MTT study based on the breast cancer cell line, that is NDMB231. And the photographs were shared with the concentrations of 6.25 microgram per ml, 12.5 microgram per ml, 25 microgram per ml, 50 microgram per ml, and 1000 microgram per ml. There was a dose-dependent reduction in the cell viability was observed in MDA-MB231 cell lines, which were administered at different concentrations of sample. And the maximum cytotoxicity was observed with 100 microgram per ml of sample. And coming to the conclusion of my study, the present study confirms the cytotoxic nature of the ornaments. These are my references. Thank you for your time.
Thank you for your presentation, ma'am. Respected session chair, if you have any question regarding the presentation, you can ask to the presenter.
What was the IC50 value with the MDT-SA?
Madam, I couldn't find out the IC50 value. Why? Because it is not going below the 50 below the 50%. So if I have to find out the IC50 value, I have to repeat the MDT above the concentrations that I have mentioned.
Thank you.
Thank you for your presentation, ma'am. Now let's move on to our next presenter, Pradeepa P. from Chettinad School of Pharmaceutical Science.
Ma'am, can you see my screen? Hello?
Yes, ma'am. Yes, ma'am, I can.
Yeah. Good afternoon, one and all. I'm Pradeepa, research scholar from Chettinad School of Pharmaceutical Science. So here I'm going to present a review on medicinally important.
Sorry for
ma'am.
the I can't. Your screen is not visible.
Okay. No.
No, ma'am, it's not visible.
No, ma'am.
It's not visible.
It's not visible. Yes. Okay, ma'am. There's. No, ma'am.
Ma'am, it's not visible.
Oh my God. No, ma'am.
It's not visible.
It's not.
It's not.
Seeing like a visible, but.
Okay, ma'am. Otherwise, we can.
Okay, ma'am. Next I'll join then.
Presenter, we'll check what's the issue and we'll get back to you.
Okay, ma'am.
Sorry for the interruption. Now let's move on to our next presenter, Mohammed Ishal.
Yes.
Mohammed Ishal MP from
Yes, yes.
Chettinad School of Pharmaceutical.
Yes. Audible, ma'am?
Yes, it is yours. Sorry, sir.
I will share. I can share only if let the Pradeepa please close your share screen. Then only I can share.
Pradeepa,
I think here.
Okay. So you can share your screen now.
Yeah. Share screen. Yeah, it's visible now, ma'am.
Yes, sir. Your screen is visible.
Yes. So, good afternoon, everybody. Myself, Muhammad Ishal, research scholar of Chettinad School of Pharmaceutical Sciences, Chennai. And my topic is, this is actually a review presentation regarding my research topic. Actually, I'm starting the research work on two specific marine seaweeds, Padina gymnospora and Sargassum tenarium. Both are brown algae, light brown algae, which I collected from Tamil Nadu coastal area. I'm starting the research. So I'm presenting this is actually pharmacological review presentation. And this actually the marine resources is playing a very huge and vital role in the drug research and development, especially in antimicrobials. We have a lot of hope, and we can beat a lot of challenges like antimicrobial resistance from the marine source antimicrobials. I am showing some research review literature survey, and this especially lot of novel metabolites are emerging, which is having potent pharmacological properties, especially from the marine seaweeds, algae, and marine plants. So I selected two species of marine seaweeds, which is brown algae, Pardina and Sargassum, both coming under Phyophytae. And this specific seaweeds is easily available, not in the exactly coastal. We have to go a little inner side, then we will get these two species. I collected this Pardina gymnosperma. This is also known as ceiling fan blade algae. And this is a kind of brown algae which I collected. And another one is Sargassum tenerium. That is also a brown algae. This is the taxonomical status of Pardina gymnosperma, which is coming under the family Dictyotaceae and phylum Ochrophyta. And Sargassum also another one that is coming under the family Sargassacea. And this both species I done extensive literature survey, both containing especially phytophenols, terpenoids, and alkaloids. Then glycosides, flavonoids. These 5 classes are prominently noticed in the 2 species, which I found from the literature research. And what I felt is, compared to the Pardina, Sargassum tenerium having very limited, undergone very limited studies. Compared to Sargassum, Pardina having a little more research which is already done by the scientists. And this is some of the recent research papers, which is one is in 2023 published in Food Science and Nutrition Journal, and there noticed reported the antioxidant, anti-diabetic, and antimicrobial activity of the Pardina. And another one published in Journal of Plant Biology, which is also specifically experimentally proved the anti-diabetic and antimicrobial property of the hexanic extract of Pardina gymnosperma. And the journal, International Journal Plants, which is reported in 2023 in the same year about the antioxidant activity of the Pardina. And in the Journal of Fisheries Science International in the same year, this current year, they reported the anti-tuberculosis and antibacterial antioxidant activity of this Pardina. And another research review work I reviewed in the Journal of Food Science and Technology in the last year, 2022, about the Pardina, which unveiled the antibacterial antioxidant property of the Pardina. And the cytotoxic activity of the Pardina reported in Iranian Journal of Fisheries Science in the last year. They extensively unveiled the anti-cancer activity and cytotoxic activity, which reveals the anti-tumor potential of the brown algae, which is under study. And the another one in the last year also in the Journal of Applied Biochemistry and Biotechnology, which reveals the oral cancer against the oral cancer activity from the Pardina gymnospora. Here all I noticed in the review mainly the phytoflavonols, glycosides, and alkaloids, including tannins, showing the activity. Another one in the 2021 in Iranian Journal of Botany, they reported around 6 species of Sargassum. This is one of the very rare research journal I found for the Sargassum. Compared to the Pardina, Sargassum having a very little number of hits in the research which is already done by the scientists. That is explaining the Sargassum pharmacological activity. And another one, South African Journal of Botany in 2021, they reported also the cytotoxic activity and of the Padina. And in 2021, again, in the Journal of Pharmaceutical Research International, they reported the anti-cancer activity of the Padina. And this is another research work which is already done in Padina, that is for the anti-coagulant activity. This is one of the innovative approach which is done in this Padina brown algae for the antioxidant and antithrombotic activity. And that journal also reporting that the flavonoids and cardiac glycosides present in the Padina is responsible for the anticoagulant activity of the Padina. And this another one is in the journal, International Journal of Aquaculture, they're reporting the seaweed extract, regarding the seaweed extract of this Padina against the bacterial infection, and they are exploiting against the fish, in the fisheries science for boosting the immunity and resisting the microbial attack in the fisheries also. And another two is regarding the Padina also, and sargassum against the antibacterial study in World Journal of Fish Marine Science and Journal of Modern Biotechnology. And this is another one, this is also same regarding anti-bacterial. And this one, I want to highlight this one. In 2010, in Neuroscience Letters International, they published by Sugandhi et al. Regarding the Alzheimer's, anti-Alzheimer's activity and significant inhibitory activity of the acetylcholinesterase by the brown algae Padina gymnosperma. And antioxidant activity, I already mentioned. This is another journal in paper in published in 2007 in Journal of Applied Phycology. And what's the conclusion which I found in my extensive research of the literatures regarding these two species of specific light brown algae, that a lot of activities, a lot of ingredients are present. It's a pool of chemical constants having vivid kind of pharmacological activity. Mainly the past scientists, they studied about the antioxidant activity and even antimicrobial activity also little studied, but still there is a bunch of scope in that. And especially in the sargassum tenerium, what I found is very minimal research is already undergone in the pharmacological activities of the sargassum. So that is a lacuna of the research that we can think of forward. And especially the modern technologies like advanced technologies, extraction technology, analytical technologies are least minimally used. What I found in the literature, in 2 or 3 papers, they use the GCMS. But my planning is to go for some modern sophisticated extraction like a critical fluid extraction and LCMS I am planning. So these are all the conclusion I got from this literature review, especially for exploiting these two species. This is about my presentation. These are the references, man. Thank you so much.
Thank you for your presentation, sir. Our dear session chairs,
Thank
if you
you.
have any questions, you can ask the presenter.
No question. It is a review only. No. Okay.
Okay, thank you for your presentation.
Yes.
Now let's move on to our next presenter, Dr. Vikas Suresh Shinde from
When
SG.
shall I join?
Just a few minutes, ma'am. We'll move on to our next presenter, Dr. Vikas Suresh Shinde, SGM SPMS, Dhyan Vilas College of Pharmacy, Pune, India.
Good afternoon, everyone. Actually, sir is not available, so I am going to present his work. Can I share my screen?
Yes, ma'am, you can.
My screen is visible?
Yes, ma'am, it is. Please enlarge your screen.
Yes. Is it visible now?
Yes, ma'am, it is.
Okay. Good afternoon, everyone. Myself, Himlata Dange. The name of the topic which I will be represented over here is Seneco Eggworthy Flower Oil has a neuroprotective effect on rats exposed to aluminum chloride and 3-NP induced neurotoxicity. So what is neurodegeneration? The neurodegeneration is a progressive loss of the structure or function of the neurons. Many neurodegenerative diseases are caused by genetic mutation, protein misfolding, protein degradation pathway, membrane damage, and mitochondrial dysfunctions. There are the different neurodegenerative disease like amyotrophic lateral sclerosis, that is ALS, then Parkinson disease, that is capital PD, Alzheimer disease, that is AD, and Huntington disease, that is HD. The work is related to this Alzheimer disease and Huntington disease. This disease occurs as a result of neurodegeneration process. The scope of work, the ADHD and PD, that is a neurodegenerative disease, is a chronic neurodegenerative disease that usually starts slowly and get worsened over the time, ultimately leading to the death of the person. Nootropic agents, the agents which are used in the treatment of this neurodegenerative disease is known as the nootropic agents. The nootropic agents such as piracetam and choline esterase inhibitors like donepezil, etc. are being used to improve memory, mood and behavior, but the resulting side effects are associated with these agents have made their use limited. Several methods and compounds have been investigated as neuroprotectant, but the ideal agent has not been discovered as yet. Hence, there is a need to validate traditional knowledge in a scientific way. Natural plant phytoconstituents are the most abundant antioxidants and anticholinesterases for neutralizing stress-induced free radicals and increase the level of ACH and decreases the inflammation of the brain. So here we use the oil of the plant because the natural phytoconstituents show the antioxidant activity, and increase in the oxidative stress is a major reason of neurodegeneration. So here we use the plant oil. Here we use the natural phytoconstituent for the treatment of the disease or to decreases the formation of free radical, ultimately decreases the oxidative stress, or we can control alkaloids and terpenoids. Many earlier investigations have shown that terpenoids diminish oxidative stress and act as the neuroprotective agents due to their antioxidant property. As the terpenoids show the antioxidant property, and by showing the antioxidant property, they can act as the neuroprotective agents. Then Seneco Edworthy's plant were selected for the present research work, as no scientific data is available for its neuroprotective activity. So here we choose this plant. Objective of the work to assess the neuroprotective effect of Seneco Edworthy oil on different models of neurotoxin agents, to study the effect of Seneco Edworthy oil on various behavioral parameters. Locomotor, rotarod, elevated pulse maze, Morris water maze, and T-maze apparatus were used. Then the various biochemical parameters are analyzed, like level of acetylcholine esterase enzyme, then MDA, SOD, CAT, CHS, cholesterol, T protein, and histopathological and DNA fragmentation study. Here we use the model of AlCl3, that is aluminum chloride, and 3-NP for the induction of the neurodegeneration to isolate and identify the various phytoconstituents present in the oil. The material and method: collection, identification, and authentication of the plants, extraction of the plant material, preliminary phytochemical screening of the extract, then acute toxicity study, pharmacological evaluation, the neuroprotective activity of Seneco Edworthy, aluminum chloride induced neurotoxicity, then 3-NP induced neurotoxicity, isolation and identification of the phytoconstituent. This is the plant profile. The botanical name is Seneco Edworthy hook. Vernacular name is Sonaki. The family is Asteraceae, and distribution is present in India and Western. So this is the procedure for extraction of oil. The oil is extracted by using the dry powder. They extracted with the hexane, then passed through the anhydrous magnesium sulfate. Then solvent was removed on the rotary evaporator at 50 degrees Celsius, and the oil is stored at 4 degrees Celsius. Then here the GCMS study of the SE oil. The chemical composition from GCMS from GCMS study, the compound is identified. These are the various compounds along with their molecular formula. We found out that this caryophyllene oxide are mainly the responsible for antioxidant property. Then acute toxicity study. This acute toxicity study was performed according to OECD 423. The final dose is selected for study is 125 mg per kg and 250 mg per kg. Then this is the first model for the induction of the Alzheimer disease. For the induction of Alzheimer disease, the aluminum chloride is used. These are the grouping of the animals. We observe this behavioral parameter on the 7, 14, 21 and 28th day after the 3 hour of the treatment. Then this 3NP induced neurotoxicity model is used for the induction of Huntington disease. These are the group, and the behavioral parameters is observed on the 14 and 21 days after the 3 hour of the treatment. Then these are the various evaluation parameter. The behavioral assessment include locomotor activity, rotor rod activity, then elevated pulse mates, T maze, then Morris water mates. The statistical analysis, here the ANOVA is ANOVA followed by the Turkey-Kramer multiple comparison test is used for the statistical analysis. The biochemical parameters. The biochemical parameter for the biochemical analysis, the animal were sacrificed, the brain was removed, 10% W by volume tissue homogenate was prepared in 0.1 molar phosphate buffer. The homogenate was centrifuged at 10,000 rpm for 15 minutes, and the clear supernatant was separated and used for the further biochemical estimation. The solution is used for the estimation of acetylcholinesterase level, then estimation of lipid peroxidation, estimation of superoxide dismutase level, estimation of catalase level, estimation of glutathione level, and DNA isolation from the brain tissue. This is the pathophysiology, how neurotoxicity induced by ALCL3. The ALCL3 induced in the rats, it causes the AB deposited in the wall and cerebral blood vessels, then interferes with the epicatalyst metabolic reaction in the brain. Both are responsible for the neurovascular changes. This neurovascular changes is responsible for increases the level of acetylcholinesterase enzyme or increases the activity of acetylcholinesterase enzyme. The acetylcholinesterase enzyme is responsible for degradation of acetylcholine. So when the level of acetylcholinesterase increases, ultimately the level of acetylcholine decreases, which is the major reason behind the development of Alzheimer's disease. Then the neurovascular changes is further responsible for increase the level of MDA, SOD, catalase, and GSH. Decreases the level of GSH, which indicates the increases the formation of the reactive oxygen species, which are responsible for the neurodegeneration, which further progress into the cognitive changes and which are responsible for the onset of Alzheimer's disease. Then behavioral assessment, the various parameter we observe is body weight, locomotor activity, rotor rod activity, T maze, elevated pulse maze, and Morris water maze. In negative control group, the body weight is decreases, and in highest test group, that is SE oil 250 mg per kg, the body weight is increases. Then locomotor activity is decreases in negative control group and increases in the SE oil 250 mg per kg group. Then rotor rod activity decreases in negative group and increases in SE oil 250 mg per kg. That is our test high dose. Then in TMS activity, it's also it increases in negative control group and decreases in SE oil 250 mg per kg group. Then elevated pulse mates, that activity is decreases in negative group and increases in SE oil 250 mg per kg group. Then Morris water mates, the activity is decreases in negative control group and also decreases in SE oil 250 mg per kg group. Then biochemical parameters, here the acetylcholinesterase level in AD and lipid peroxidation level in AD. Here we see in negative control group, the acetylcholinesterase level is increases, and in test group, that in 125 mg per kg, the level is decreases and more level is decreases in SE oil 250 mg per kg group. Then in lipid peroxidation case, again negative control show the increase and the higher dose of test show the decrease in lipid peroxidation level. Then SOD level is SOD level. In negative control group, the SOD level is less, and in SE oil 250 mg group, the SOD level is high. Then catalyst level in AD, the catalyst level in negative control group is low, and higher test group is the more. Then GSS level, the GSS level in negative control group is less and more in SE oil 250 mg per kg group. These are some histopathological examination. This B indicate the negative control, which show the clear destructions of the cell. Which show the destruction of the cell, and the destruction is less in the higher dose of the test, that is SE 250 mg per kg. Then the DNA fragmentation is done.
Please conclude your presentation.
Hello.
Sorry for the interruption, ma'am. Please conclude your presentation.
Okay. In conclusion, the SE oil 250 mg per kg dose suppresses the ALCL3 and 3NP induced memory and learning impairment, acetylcholine esterase hyperactivity, then ALCL3 and 3NP overload, oxidative stress, inflammation, and apoptosis by activating BDNF and NDF and reducing oxidative stress. This study suggested that this oil of hexane extract, due to the presence of carbophylline oxide, may be shown as protective agents for AD and HD. The SE oil 250 mg per kg only show potency at higher dose and not at the lower dose to prove the effectiveness of the treatment for AD and HD. However, more in-depth research is required. The structural association between phytoconstituent present in plants and the relationship between dementia caused by ALCL3 and 3NP mediated processes both need more study. These are the certificates, the plant authentication letter. Then these are the IAAC approval. These are some references.
Thank you for your presentation, ma'am. Our dear session chairs, if you have any questions regarding the presentation, you can ask to the presenter.
No questions. It's good presentation.
Thank you for your presentation, ma'am.
Thank you.
Now let's move on to our next presenter, Pradeepa P. from Chettinad School of Pharmaceutical Science.
Able to see, ma'am?
Ma'am, please share your screen. You are not visible. Your screen is not visible.
This sharing option is not at all working, I think.
You can continue your presentation, ma'am. Pradeepa, are you there? Pradeepa, are you there? Okay, now let's continue to our next presenter. Sheetal Rana from Sardar Bhagwan Singh University. Sheetal Rana, are you there? Now let's move on to our next presenter, Aksha Rajput, Sardar Bhagwan Singh University. There's an update. Please, the e-posters have 5 minutes for presentation, 3 minutes for presentation, and 2 minutes for question and answer. Now let's move on to our next presenter, Suneha from Sardar Bhagwan Singh University. Okay.
Okay, start.
Hello. Hello, ma'am. Can you hear me?
Yes, ma'am.
Can you able to see my screen?
Ma'am, your screen is not visible. Ma'am, we'll give you another chance. So,
Okay.
we'll move on to the next presenter, Suneha. Next, we can move on to Manish Chauchan from Sardar Bhagwan Singh University. Manishi. Now let's move on to our next presenter, Salwa Abdul Salam from Chettinad School of Pharmaceutical Science.
Can you hear, madam?
Yes, ma'am.
Can I share my screen, madam?
Yes, ma'am, you can.
Okay, ma'am. Is it clear, madam? Can you hear? Can you see, madam, the screen?
Yes, ma'am.
Okay.
Please enlarge your screen.
Is it okay, madam?
Please enlarge it a little more.
Okay, madam.
Yes, ma'am, you can start.
Is it okay? Okay. Myself, Salva Abdul Salam, and I am a research scholar from Chettinad Academy of Pharmaceutical Sciences, Chennai. And here I am going to take a presentation on the topic.
One second, madam. Please, we are not able to see. It is mobile screen which is seen. The slides are not seen clearly.
Is it okay now, madam?
Ma'am, you please enlarge your screen. It's not visible.
It is not at all visible. it is a PowerPoint, and you can have a change the direction of mobile, and you can
Is it now clear, madam?
Ma'am, are you using the mobile version for sharing the screen?
Okay. Yes, madam, using the mobile version.
Ma'am, please better use the laptop for sharing the screen. Ma'am, at the last we can give you another chance. So now let's move on to our next presenter.
Okay, madam. Okay, madam.
Meanwhile, arrange for the laptop.
Okay, madam. Okay.
Okay, thank you, ma'am. Now let's move on to our next presenter, Salwa Abdul Salam. Now let's move on to our next presenter, Bijesh Vatakil from Chettinad School of Pharmaceutical Science.
Can you hear?
Yes, sir.
Hello.
Yes, sir. It's audible.
Let me share the screen.
Okay.
Can you see? Can you see?
Yes, ma'am. Yes, sir.
Okay. Shall I start?
Yes, sir, you can.
Hello. Let me introduce myself. I am Bijesh Vatakil. I am a research scholar in Chettinad Academy of Research and Education, doing the research under the guidance of Dr. R. Maitreyi. And I am here to present a comprehensive review of this Coccus nucifera lin on its phytochemistry as well as the pharmacological activities. And actually, it's my plan for the thesis work. Okay. So this Coccus nucifera, it is coming under the Acacia family. It's a common palm which is found in the different parts of the world, and it is usually consumed for the various purposes, especially the food and oil. It is widely used as a nutraceutical globally. And Coccus nucifera has a wide range of conventional medical application in inflammation, protozoal and microbial infections, cancer, hepatic illnesses, diabetes mellitus, pain, epilepsy, and fungal infection, and also depression. And these different parts of the Coccus nucifera, like leaves, flowers, roots, oil, and cotyledons, were explored for the scientific authentication in various illnesses. The presence of phytochemical like various amino acids like lauric acid, arginine, caprylic acid, linoleic acid, and palmitic acid, and also other constituents like vitamin C, saponin, phenol, and terpenoids, phenolics, and tannins were reported in the Coccus nucifera. And this Coccus nucifera was subjected to various pharmacological studies, like this scientifically authenticated on its antioxidant, antimicrobial activity, hepatoprotective activity, anti-cancer activity, anti-parasitic activity, anti-malarial activity, hypoglycemic activity, anti-helminthic activity, anti-epileptic activity, antidepressant, analgesic, peptic ulcer, anti-inflammatory as well, anti-hypertensive and nephroprotective activity, and various parts like endocarp, husk, coconut cakes, mesocarp, inflorescence, flowers, bark, and coconut milk, coconut water, virgin coconut oil were explored for its pharmacological activity. And the presence of these constituents also hint that this having hidden pharmacological activity. And this is a review paper on the Cocos nucifera on its phytochemistry as well as the pharmacological activities. And this reveals that it may be used as a promising, it may be used as a promising candidate for the drug discovery process. And so it is concluded that the researchers from around the world conducted scientific pharmacological evaluation of the Cocos nucifera to confirm the traditional medical uses such as antioxidant, anti-inflammatory, anti-protozoal, antimicrobial, anti-cancer, like that, different activities. And these documented scientific studies may aid in developing of a safer herbal chemical moiety in drug discovery. Okay, that's all. Thank you.
Thank you for your presentation, sir. Dear session chair, if you have any question regarding the presentation, you can ask to the presenter.
Only so less compounds are isolated from this plant?
No, madam. Actually, I already told some of the amino acids. Okay, lauric acid, different amino acids like lauric acid, arginine, caprylic acid, linoleic acid, palmitic acid, and various saponins, phenols, terpenoids, phenolic compound, and tannins were isolated from this compound, and various plant hormones like auxin, cytokinin, 1,3 diphenylurea, and there is enzymes like acid phosphatase, catalase, dehydrogenases were isolated from this plant.
Okay, thank you.
Thank you for your presentation, sir. Now let's move on our next presenter, Dr. Rekha Nayaka from JN Medical College. Dr. Rekha Nayaka, please present your screen. Now let's move on to our next presenter, Bharat SI from Chettinad School of Pharmaceutical Science. Bharat SI is unavailable at the moment. So now let's move on to our next presenter, H. Jasminajit Kaur from KPJ Healthcare University. Jasminajit Kaur, are you available? Mohammed Faiz B. Abghani from KPJ Healthcare University. Now let's move on to our next presenter, Anirban Banerjee from Guru Nanak Institute of Pharmaceutical Science and Technology.
Good afternoon, ma'am.
Good afternoon, sir.
Can I share my slides?
Yes, sir.
So can I start, ma'am?
Yes, sir, you can.
Good afternoon, ma'am, and good afternoon to the honorable evaluator in chair. I am Anirban Banerjee. I'm from pharmacology student of Guru Nanak Institute of Pharmaceutical Science and Technology. And today I am discussing about my research project under the guidance of Mr. Samrat Bose, Division of Pharmacology. And my project is the evaluation of anti-diabetic activity of Sweeteria macrophylla bark using by in vitro study. So here is a table of content. Next, introduction. So as we all know, the diabetes mellitus is a chronic metabolic disease and which is abnormally elevated the blood glucose characterized by, and according to WHO, worldwide more than 400 million individuals are suffering from diabetes, and 1.5 million of them are directly died due to this cause of diabetes, and it is the major causes of also kidney failure, heart attack, and retinopathy as well. On the other hand, Sweeteria macrophylla is a topical plant. Meliaceae family belong to the Meliaceae family, also known as the mahogany, and its bark represent potential alternative, and various bioactive compounds the present in the bark are influence the glucose metabolism as well as provides the antioxidant protection. So it is crucial for diabetes management. So our aim is by employing the in vitro method, the specific we are uncovering the specific anti-diabetic activity of Sweeteria macrophylla bark. And objective and major objective are obviously the investigate the anti-diabetic property as well as the mechanism of the action of the present of bioactive compound in the bark, and establish a analysis between the extraction in different medium also, and also it can be a further natural remedy, can be a natural remedies in the diabetes management in the future. So the justification, so why we use in this project Sweeteria macrophylla. So these the plant contain various bioactive compounds according to literature. Limonoids, flavonoids, alkaloids are major compound, and in the bark majorly present in limonoids, and limonoids is the major medicinal property. Limonoid present provides the major medicinal property of this plant, and according to literature also it is responsible for the anti-diabetic activity. So here we are taking this bark to the compare the extraction in between the hydroalcoholic extract and aqueous extract, and which is the more potent is the outcome of our project. So as we talk about the why we use Sweeteria, it is the treatment in case of streptozotocin and nicotinamide induced type 2 diabetes is macrophylla bark extract for consecutive 12 days reduces the blood glucose level. On the other hand, sweetenin, a major lemonoids present in the macrophylla Sweeteria, it provide the dose dependent hypoglycemic effect, and also the catechin and epicatechin and other flavonoids isolated from bark extract are provide the antioxidant activity by using DPPH assay, because antioxidant activity is also important for diabetes management. So material we use Sweeteria macrophylla bark, solvent for extraction, ethanol for hydroalcoholic extraction, and distilled water. And enzyme we used alpha amylase, also alpha glucosidase, and for DPPH assay we also use the DPPH reagent, and DMSO, and phosphate buffer for the maintain the media alkaline medium. So here is the methodology. So previously we collect and prepare the S macrophylla bark by the size reduction as well as drying properly. Then we extract the bioactive compound through the maceration in two different medium. One is the hydroalcoholic medium and another is pure aqueous medium. So both the extract in hydroalcoholic as well as the aqueous extract, we performing the phytochemical test. Some here is the some phytochemical test for the for identification the present bioactive compound. Next we are treating, well, we have treated the enzyme, both enzyme as well as DPPH reagent with the both of the solvent, both of the medium and extract, and the finally we observe the inhibition percentage and we validate the result and conclusion. So the result in case of interaction between S macrophylla and alpha amylase, for the aqueous, here is the aqueous extracts dose dependent percentage inhibition, and here is the hydroalcoholic dose dependent and percentage of inhibition, and both is the comparison. When we are compare these both of the result, the hydroalcoholic extract is better, superior, provide better inhibition than the aqueous extraction for the alpha amylase enzyme. On the other hand, same as alpha glucosidase, in case also the hydroalcoholic extract also provide more inhibition than the aqueous extract in when it is interact with the alpha glucosidase. Next, in case of interaction between DPPH reagent, here also the aqueous extract and the interaction between hydroalcoholic extract with DPPH. In that case also, the hydroalcoholic extracts provide more or free radical scavenging activity than aqueous solution, aqueous extraction. So discussion and conclusion, the hydroalcoholic extract demonstrate superior inhibitory effect in against the alpha glucosidase as well as the alpha amylase and as well as the DPPH assay. Here we are taking the concentration. The concentration increased 62 gradually to 10 mg per ml. It is provide the dose dependent inhibition. So the efficacy is increases then when we increase the extraction concentration in both solution. But here we also found that hydroalcoholic extraction provide more superior inhibitory activity in three of the two enzyme and also DPPH reagent. So here is the reference.
Thank you for your presentation, sir. Now,
Thank you,
dear
ma'am,
session
for
chairs, if
giving
you have any questions regarding the presentation, you can ask him.
Nothing much. Okay.
Thank you, ma'am.
Thank you, sir, for your presentation. Now, let's move on to our next presenter, Shreya Sarkar from Guru Nanak Institute of Pharmaceutical Science and Technology.
Hello, ma'am. Hello.
Hi, ma'am. Yes, we can hear you.
Am I audible?
Yes, ma'am.
Okay. Hello. Hello.
Hello.
My screen is visible? Hello.
Ma'am, Rekha Nayak will call you after this participant. Please mute yourself.
Because there is some issue with the thing. I have been told that I've called 3 times, so I just wanted to confirm. Am I audible?
Yes, ma'am, you are audible. After this presentation, we'll call you, ma'am.
Yes. Okay, fine.
Shreya Sarkar, you can continue with your presentation.
Okay, ma'am.
Your screen is not visible, ma'am.
Yeah, I am unable to share my screen.
Okay, we'll give you the chance again, ma'am. Now let's move on to the Dr. Rekha Nayaka from JN Medical College. Dr. Rekha Nayaka, please present your paper.
Do I have to share because I've already shared my poster?
Yes, ma'am, please share your poster here.
No, I've already shared my poster to you so that I don't know how to share the poster in the thing.
Okay, ma'am, just a few minutes.
Okay.
Now let's move on to our next presenter, Pradeepa P. from Chettinad School of Pharmaceutical Science.
Can you hear me, ma'am?
Yes, ma'am. Please present your paper.
Yeah. Can you able to share screen? Can you able to see my screen, ma'am?
Ma'am, no, your screen is not visible.
That is the only issue. Okay. Now?
No, ma'am, your screen is not visible.
Ma'am, can I share my screen? Hello.
Yeah, you can.
Hello, ma'am.
Pradeepa, are you
Hello.
able?
Yeah, ma'am, I can hear, but I'm sharing the screen, but it's not visible. But
Okay, ma'am.
I can see in my screen that I'm is visible.
Okay, ma'am, I'll share the screen. You please continue with your presentation.
Okay, ma'am. Okay. Shall I start, ma'am?
Yes, ma'am, you can.
Yeah. Good afternoon, one and all. I'm Pradeepa, research scholar from Chettinad School of Pharmaceutical Sciences. I'm going to present a review on medicinally important homemade herbal flowers. So this is my initial stage of ongoing research work. So I start a search or research my work from my home yard itself. So for the introduction, medicinal plants are nature's priceless gift to human. It contains many chemical substances which play a dominant role in the development of new drug. The crucial first step in the development of a new drug is the screening of active compounds from various plant parts. An attempt has been made to describe the medicinal significance of the flowers found in Palakkad in Kerala state and their therapeutic applications in this review. Medicinal flowers in Kerala. Kerala is an extremely biodiverse state in India with a long history of using traditional medicines. According to the recent survey, out of 4,600 flowering plants, 900 are have therapeutic properties. Flowers offer several therapeutic values and are considered to be very important in Ayurveda. Flower therapy or Pushpa Ayurveda is a special branch in Ayurveda which contains 800 types of flowers are mentioned. Flowers also play an essential role in human nutrition and diet. Many studies have shown that flowers are being used to make decoctions in different parts of the world to cure and treat diseases. Aim of my study is to investigate the nutritional profile, phytochemistry, and biological activities of different flowers and also identify toxin-produced flowers from my home courtyard. Objective to consider home yard medicinal flowers for our health benefits. Methodology. The different flowers were observed from my home courtyard and reviewed many research articles, review articles, and refer some Ayurveda books and understand the traditional methods of using those flowers and find their importance of therapeutic values. This is a table of survey on homeyard flowers. It contains some of flowers and scientific name, which contains hibiscus, rose, ixora, vinca rosea, marigold, chrysanthemum, alamanda, pomegranate flowers, mandaram white, shame flower, morning glory, butterfly pea, and their scientific names. These are the photos which I have taken from my garden. Next, move to the hibiscus plant. So, out of 300 species from my garden, I found Hibiscus rosa sinensis, which belongs to the family Malvaceae. The flowers are bisexual, large and showy, grow up to 25 cm wide and stalked and arising from upper leaf axils. It contains 5 free petals joined at the base and it may be color white, yellow or red. Hibiscus rosa sinensis is a national flower of Malaysia. The origin is Malaysia, that's why. Then flavonoids, anthocyanins, terpenoids, steroids, polysaccharides, alkaloids, amino acids, lipids, sesquiterpene, quinones, and naphthalene groups are the active constants which is present in the hibiscus. It contains many therapeutic values. It's used as an antioxidant, anti-inflammatory, antimicrobial, antifungal, anti-diabetic, anti-hypertensive, and other cardioprotective activity and anti-tumor also. Traditionally, we used hibiscus tea as a cardioprotective and also antioxidant property. The next is rose, which belongs to the family Rosaceae. It contains 200 species, and Rosa indica is my species. And flowers are axillary and symmetrical, and they are typically red with 5 petals, 10 cm in diameter, with prominent orange-tipped red anthers. Flavonoids, triterpenes, tannins, polysaccharides, phenolic acid, fatty acids, organic acids, carotenoids, and vitamins are the chemical constants. This carotenoids which gives the color to the rose. This is also have therapeutic values like antidepressant, antispasmodic, aphrodisiac, astringent, increased bile production, cleansing, antibacterial, and antiseptic one. Then rose hip tea is also used as a tea as an antioxidant, immune system booster, and aid weight loss and reduce joint pain and treatment of diarrhea. And also to improve our skin complexion, rose water also is be used. The smell, fragrance of the rose water, which gives antidepressant and anti-anxiety properties also. Next is Ixora. From my garden, I found this Ixora coccine, belongs to the family Rubiaceae. There are 544 species are there and which contains the alkaloids, tannins, terpenoids, flavonoids, saponins, and other essential oils. It's also used as an antioxidant, anti-tumor, wound healing, hepatoprotective and chemoprotective activities, and many other therapeutic values. Traditionally used as an anti-diarrheal and dysentery agent. Vinca, as we all know, it is an anti-cancer agent. So, Acanthus roseus, which belongs to the family Apocynaceae, and it contains the tubular flowers and have 5 flattened petal-like lobes and appears singly in the upper leaf axils. Vinblastin, vincristin, vinorelbin, and vinflunin are the chemical constants which is present in the Vinca roseae. So, out of this, apart from this anti-neoplastic agent, other anti-diabetic, antimicrobial, antioxidant, wound healing, memory enhancer, hyperlipidemic, anti-diarrheal, anti-ulcer, and anti-hypertensive activities also there. Flower extracts before in traditional ways is used for infants' eye wash. Periwinkle is another name of this Vinca rosae. And because of detoxification property and counteracting poison, it is used to relieve wasp stings also. The next is marigold. Taget selecta is a species I found from a garden and belongs to the family Asteraceae. Then carotenoids like neoxanthin, luteoxanthin, lycopene, lutein, beta-carotene are the chemical constant which is present in the marigold. And it's also used as antipyretic. It's used for the treatment of jaundice, ulcers, conjunctivitis, liver problems, and other skin problems also. The next is Chrysanthemum. Chrysanthemum aurifolium is a species which belongs to the family Asteraceae, and it was first appreciated in China as a medicinal plant. Anthocyanins, phenolic acid, luteolin, chlorogenic acid, acetylsin, and caffeolicunolic acid are the chemical constants which is present in the chrysanthemum. It's also used as an antioxidant, antimicrobial, hepatoprotective, immune regulation, and nephroprotective activities. Then next is Alamanda. Alamanda cathartica is a genus of flowering plants which belongs to the family Apocynaceae. The flower have 5 lobed sepals and a bell or funnel-shaped corolla of 5 petals, and yellow is the most species one. Hydrocarbons, alcohols, esters, ethers, aldehydes, phenolic compounds, flavonoids, which are all present in the plant parts, especially in the flowers. There is no articles I found. And antifungal, anti-cancer, anti-inflammatory, cathartic, diuretic, antipyretic, anti-hypertension, and also it is used as to improve blood circulation and jaundice and malaria. Then pomegranate flower, which belongs to the family Punicoideae, and the species found in my garden is Punica granatum. And chemical constants are tannins, terpenes, terpenoids, flavonoids, organic oils. Then common tannins found in flowers are ellagic acid, punicatintacin, and gallic acid. Uses antidiarrheal, astringent, antioxidant, antibacterial, and antimicrobial results are there. It is also used for the treatment of cardiovascular diseases, obesity, hyperglycemia, dyslipidemia, and mastitis. The next flower is Mandaram white. Baunia acuminata is a species and belongs to the family Cisalpinaceae. Flowers are fragrant with 5 white petals, 10 yellow tipped stamens, and a green stigma. Alkaloids, anthocyanoside, phenolic proteins, phlobatannins, steroids, tannins, flavonoids, anthraquinones, saponins, terpenoids, resins, balsams, amino acid, carbohydrate, sugars, and cardiac glycosides are the chemical constants which is present in the Baunia. Then it's used as an antibacterial agent, antioxidant, anti-inflammatory, nephroprotective, hepatoprotective, proteinase inhibitors, insulin release enhancer. Then another important one is shame plant. It's also known as touch me not, live or die, or shame plant and humble plant. Flowers are pink, globose head and prickly peduncles and small calyx. Alkaloids, non-protein amino acid, flavonoids, sterols, terpenoids, tannins, and fatty acids are the main chemical constants which is present in the Mimosa pudica. It's used as an antibacterial, anti-venom, especially against the cobra venom. The entire plant is used as an anti-venom.
Pradeepa ma'am, please, sorry for the
Yes.
interruption. Please wait a moment.
Ah.
Can everyone see the screen?
No. The screen, some table has been shown.
Is it visible now, ma'am?
Yeah.
Yeah, it's visible.
Thank you. Can continue your presentation, ma'am.
Yeah, thank you, ma'am. So next is shame plant, and that's we have discussed about the antibacterial, anti-venom, anti-fertility. Ma'am, screen number 25. It is. Okay, so this shame plant is used as an anti-venom for the cobra bite and other some snake bite also. Entire plant will be used for this anti-venom, and that is the important thing here. And it is also used as a used for dysentery, insomnia, tumor, and various urogenital infections. Then this is the morning glory. There are about 600 species are there. Pomea indica is the blue morning glory is another name and belongs to the family Convolvulaceae. And here the chemical constants are and have antioxidant property, and compounds like flavonoids, polyphenols, and phenolic acids are the present. Then butterfly pea is also known as Clitoria ternata, that is I found from my home court and belongs to the family Fabaceae. Then flowers are pea-shaped with a tubular calyx, consist of few sepals which are fused about 2/3 of their length. Then chimpherol, quercetin, and myricetin glycosides are as well as the anthocyanin also present in the flowers. Then antimicrobial, antioxidant, anti-inflammatory, cytotoxic, anti-stress, anxiolytic, antidepressant, and anti-diabetic action also are there. So from this plant traditionally used as a tea. That is, butterfly pea flower is a common ingredient in the many herbal teas, mixed drinks, and cosmetic products. It is rich in antioxidant, also used for the weight loss, better blood sugar control, and improvements in hair and skin health. Then plan of my work is collection of individual medicine flowers, shade drying and size reduction, preparation of aqueous or alcoholic extract, percentage of yield calculation, preliminary phytochemical screening, determination of acute toxicity, then an evaluation of medicinal activity in animal models. So again, I'm sharing that this is only for the initial stage of my ongoing research work, that is my from review articles. So these are my references. Thank you.
Thank you for your presentation.
Okay, thank you, ma'am.
Now, dear session chairs, if you have any questions regarding the presentation, you can ask her. Can we proceed to the next presentation, ma'am? Now, let's move on to our next presenter, Dr. Rekha Nayaka from JN Medical College.
Yes, ma'am. I'm here. You want me to share the screen, or will you be sharing the screen?
Ma'am, if you can share the screen as well.
I'll just try, because I'm not tried any time. I'll just try. Yeah. Can you see the
Yes, ma'am.
slide?
Please full screen, ma'am. Please enlarge your screen.
But I'm not able to see the thing here. 1 second. How to enlarge? 1 second.
At the right side, options will be provided, ma'am.
Okay. Sorry. Can you see the poster?
Yes, ma'am, it is visible.
So shall I start?
Yes, ma'am, you can.
So very good afternoon to everyone. The title of my poster is Implementation and Evaluation of the Effectiveness of Interactive Computer-Based Crossword Puzzle as Innovative Teaching Learning Method in Pharmacology. So the authors of this study are Dr. Rekha Nayaka, that is myself. I am professor in pharmacology from Jain Medical College, Kahir University, Karnataka, and Dr. Bhargav and Dr. Sachin Patel are the other authors. So the background of this study is, in India, medical students are taught by innovative techniques in order to make the process of learning interesting. So in relation to this, game-based learning and puzzles are the best method of innovative techniques, which has found to be effective in creating active learning. So implementation of these innovative techniques will create interest in learning process of the students, and also it will help the student to understand the concepts easily and to recall the concepts easily. And also this game based learning and puzzle based learning will create the critical thinking ability of the students, as well as problem solving ability of this. And to create an effective learning process, created this innovative teaching method, that is interactive computer based crossword puzzle. And this innovative method has been implemented among second year medical students, and with an objective of to assess the effectiveness of this innovative teaching in learning pharmacology. So the main objective of this study is to assess the effectiveness of this innovative teaching method in learning pharmacology among second year medical students. So the A novelty of this study is that the crossword puzzles were developed using the software by the faculty. So these there are four puzzles from two CNS topics was taken, that is general anesthetics and anti-epileptics were the topics. They're the CNS topics which we teach in pharmacology, which are the very difficult topic to understand by the students. So the faculty had developed this puzzles on CNS drugs, especially general anesthetic drugs and anti-epileptic drugs. And another important novelty of this study is that each student made to solve the puzzle using the computers as a learning tool. So the study was conducted among 60 second-year medical students in the Department of Pharmacology in computer-assisted lab of Jain Medical College. And after the approval from the Institutional Ethics Committee, the study was conducted among these 60 students. And before we start the study, lecture classes, and on the day of into two groups, that is 30 groups in each. So group A was taught by tutorial method of teaching, where we taught the salient features of 4 general anesthetic drugs and 4 anti-epileptic drugs, and post days was conducted. And yeah, before we start the tutorial method, pre-test was given to the post-test, and the group B has computer based puzzle group, and they were also solving 15 MCQs on the topic. Those to crossword puzzle, that is they were given 20 minutes to solve the puzzles. So 4 puzzle from each topics were given, and after 20 minutes students were made into small groups. Out of 30 groups, 30 students in a group, they were made into small group of 4 to 5 students in a group, and they were made to disclose the correct drug name of the drugs by solving the puzzles. They came with the correct name of the drug. And those group which disclose all the students, this 2-minute task was conducted. And after this debriefing session was conducted, where the faculty discussed about the solutions of the clues which was given in the puzzles, and followed by post-test was given. And after the post-test, feedback was collected, and even focus group discussion was done among the students to evaluate the perception of students about this innovative word teaching method. And followed by the prize distribution was done to the group which come up with the correct answers or the correct name of the drugs. So this is how we conducted the study, and the results of the study was analyzed by 2 parameters. One is effectiveness was analyzed by the comparing the pre and post test scores, that is the student performance in solving the MCQs in pre-test and post-test. And another parameter is student perception. So the effectiveness study has shown that this puzzle group of students shown effective learning as compared to SGT group of students, and with the p-value of less than 0.05. So there is significant learning seen among crossword puzzle group when compared to SGT group. And student perceptions analysis showed that 90% of the student felt that this interactive computer-based crossword puzzle is an innovative method of teaching, and it created active learning, joyful learning, and also effective learning. And nearly 96% of the student felt that this method of teaching clears the concepts in pharmacology, especially in CNS pharmacology, which we took it as a topic for the model. And 86% of the student felt that this method will create or helps in developing the critical thinking skills as well as problem solving ability of the student. So the conclusion of the study is that this innovative interactive computer based crossword puzzle teaching has created a significant learning among the students, and it is very well accepted by the student as an effective learning tool. And it has also shown that solving the crossword puzzles on computers has created very interesting and active learning as well as effective learning. And also this is improve the critical thinking skills of the students as well as problem solving skill of the student, as compared is that any innovative teaching method learning is better than the traditional tutorial or lecture based teaching. So these are my references. Thank you so much.
Thank you for your presentation, ma'am.
Thank you.
Our session chairs, if you have any questions regarding the presentation, you can ask me. Okay, thank you, ma'am. Thank you for your presentation.
Thank you.
Now let's move on to our next presenter, Salwa Abdul Salam from Chettinad School of Pharmaceutical Science.
Shall I log out?
Yes, ma'am, you can. You can stop sharing the screen.
Hello. Can I log out, madam?
Yes, ma'am, you can.
Hello, madam.
Ma'am, please, there will be option stop sharing the screen. Please press that option, ma'am. You can. Thank you.
Okay, ma'am, can I share my screen?
Yes, ma'am. You can start your presentation.
Okay. Can you see the screen, madam? Can you see the screen,
Your
madam?
screen is not visible, ma'am.
Can you see, madam?
Please zoom. Okay, ma'am. You can continue your presentation.
Okay, ma'am. Okay, myself, good afternoon all, and good afternoon to the section chairs also. Myself, Salva Abdul Salam, and I am a research scholar from Chettinad Academy of Pharmaceutical Sciences. And here I'm going to take a presentation on the topic neurotoxicity, HPTLC study, and in silico study of Vanda testata on aluminum chloride induced Alzheimer's disease. This is already my research article. And here the main study is to focus on the neuroprotective study in silico HPTLC of hydromethanolic extract of Vanda testata, which is also a Resna species and belonging to the family Orchidaceae. And going to the introduction portion, Alzheimer's disease. Alzheimer's disease, which is a neurodegenerative disorder and which is clinically characterized by a decline in cognitive function, memory, and pathophysiologically defined by the accumulation of intracellular neurofibrillary triangles and extracellular beta amyloid plaques in the brain. And here, the neurotoxic agent used is aluminum, and which leads to the development of anxiety disorder, depression, memory deficit, and symptoms similar to those of Alzheimer's disease. And here the plant taken is Vanda testata, which is mainly an epiphytic orchid, and they constitute an important in folk medicine because of their multifarious medicinal properties they have. They are used in many disorders like fevers, dyspepsia, nervous problem, bronchitis, etc. And the main phytoconstituent used is alkaloid, beta-sitosterol, gamma-sitosterol, glucoside, tannins, and they also have a long chain aliphatic compounds, fatty oils, resins, etc. And here, the materials and methods, the root and leaves are collected and they are identified and they are deposited in St. Albert's College, Kerala, and the voucher specimen is 479. And here, for this study, neuroprotective and in silico study, the animal used is Vista rat, and the maceration process used is the extraction process used is maceration. And here, the hydromethanolic extract is used water and methanol.
and which is the ratio of 17:30. And the treatment protocol includes here the animal is divided into 5 groups. The treatment total days is 20 days. Meanwhile, the body weight and the behavioral studies are conducted on the 0th, 17th, 14th, and 20th day. And on final day, that is the 21st day, the animal is sacrificed, brains were isolated, then the antioxidant levels and the neurotransmitter levels were determined. And the group is for the normal control, is normal saline is given, and the disease control, aluminum chloride is given, and the standard used here is rivastigmine in the dose of 0.3 mg per kilogram. And the low dose group is 150 mg per kilogram, and the high dose is 300 mg per kilogram. And here a Ayurvedic formulation is also taken, which is 1 ml per kilogram. And the evaluation parameters here are in silico study, including fast online and molecular docking, HPTLC study, and the behavioral study and body weight changes. Behavioral study includes locomotor activity and pole climbing, and the antioxidant enzymes detected are catalase, SOD, malonidase, and glutathione, and the neurotransmitters are acetylcholinesterase, noradrenaline, dopamine, and serotonin. Finally, coming to the result and discussion part, past online study confirmed the neuroprotective action of the constituents, and molecular docking confirmed the binding affinity of the ligands, the receptors, acetylcholinesterase receptors, and TPR gamma, explaining about the molecular mechanism of action. And the HPTLC study confirmed it consists of 7 to 15 phytoconstituents with the RF value showing. And here the group 2 animals, that is aluminum chloride disease control disease group, show reduction in the locomotor activity and there is an increase in time to climb the pole compared to the treatment group. And because in the case of disease control group, it is due to the neurotoxicity of the aluminum chloride, then increased level of malonaldehyde, reduction in the body weight, antioxidant enzymes like superoxide dismutase, catalase, and glutathione were observed in disease control group. And it is mainly due to the free radical generation, and it is corrected in the treatment group, that is low dose group and high dose group and Ayurvedic formulation group. And finally, in the case of neurotransmitters, elevated level of acetylcholinesterase enzyme and the reduction in the dopamine, noradrenaline, and serotonin were observed in the disease group, and it is reversed in the treatment group like 150 mg per kilogram and 300 mg per kilogram of phytomethanol extract of Vanda tessellata and Ayurvedic formulation. And finally, the study concluded that Vanda tessellata can be used as a remedy for the treatment of neurotoxicity. And these are the references. Okay, thank you, madam.
Thank you for your presentation, ma'am. Session Chair, if you have any question regarding the presentation, you can ask. Okay, thank you, ma'am.
Okay, thank you, madam.
Ma'am, can I share my screen? Hello, ma'am.
Hi, ma'am.
Can I share my screen?
Yes, ma'am, you can. You can continue with the presentation.
Okay. Good afternoon, all. My name is Shreya Sharkar. I am from Guru Nanak Institution of Pharmaceutical Science and Technology. Today, I am here to present my paper about neurodevelopmental assessment on zebrafish embryo by using vitamin B complex. Let's start from the introduction. So, neurodevelopment is most vital process which begins by gastrula formation, which is controlled by complex signaling pathway. It establishes a risk for later neuropsychiatric illness, challenging the historical divide between childhood disorder and adult mental health. Neurodevelopmental disorders arising in early phases persist throughout life, affecting emotions, behavior, and cognitive functions. As example, attention deficit hyperactivity disorder and autism spectrum disorder, emphasizing the need to maintain a minimum level of neurodevelopmental health. My next slide is about objective of this study. The objective of the study is to evaluate the neurodevelopmental effect of vitamin B complex on zebrafish embryo against folic acid by mechanical spinal cord transection. So, what is the relation of nervous system with vitamin B complex and folic acid? So we all know MTRR gene, which is located on chromosome number 5, which is synthesize vitamin B complex. And then comes folic acid. Folic acid undergoes a spontaneous reduction process to form dihydrofolic acid, and then it converted to tetrahydrofolic acid and subcutaneously to 5 and 10 methylene tetrahydrofolic acid. The final product of this process is methylene tetrahydrofolate, which is produced by methylene tetrahydrofolate reductase enzyme. The methylene tetrahydrofolate reductase enzyme is involved in nucleic acid synthesis, methylation of DNA, protein, and other processes which held in human body, and it is also responsible for neuronal development and various neuronal disorders such as schizophrenia and other neuronal disorders. This slide is about why we use zebrafish in this research. So, zebrafish is very much identical and prominent in nature. According to the various articles, they are almost similar to the human body and approximately maximum 70% genetic similarity found to the previous research. Their brain, neurons, CNS, and other organs are majorly similar to the human body also. So spinal cord transaction process is perfect model to the zebrafish, and it is absolutely preferred in dorsal side. This slide is about methodology. So the first step of methodology is egg collection and maintenance. We have to collect eggs and maintain it properly. The healthy eggs, visible and viable, they have to transferred in a separate container with E3 solution. We observed heart rate developmental growth under microscope in 3 to 4 days post fertilization. Then feeding. We have to feed embryos from 6 days post fertilization, and for the feeding of the embryos, we prepare 1 liter of 0.9% rock salt solution with brain shrimp capsule and incubated this solution at 26 to 28 degrees centigrade. After shrimp movement observed, we collected it and we used it for feeding embryos. This slide is about mechanical spinal cord transition in larval zebrafish. For this transition in larval zebrafish embryo, we use 3 types of solution. One is agarose plate. Agarose plate mainly used for surgery plate. Then we use MS222 solution. This solution is an anesthetic solution, and E3 medium is we all know the which is maintaining embryos. Then transition process of zebrafish embryo For spinal cord transition, anesthetize the 3 days post-fertilized embryo with MS222 solution. Place the embryo on agarose plate and use a hypodermic needle. Perform larval zebrafish spinal cord transection under a microscope. Then we used to charge the drugs. So we made 3 types of group. One is control, second one is vitamin B complex treated group, and third one is folic acid treated group. So in control group, Petri plate filled with 10 ml E3 medium with 6 transected embryos, which feed daily and monitored for recovery and growth up to 10-day post-fertilization. In vitamin B complex, we use vitamin B complex as a test drug for neurodevelopment observation in transected zebrafish embryo. We use 2 ml neurobiome injection containing vitamin B3, B6 and B12. We use 5 Petri plates with 10 ml E3 medium with various concentration of drug, which are 20 ml, 30 ml, 40 ml, 50 ml, and 60 ml, were administered to 6 transfected embryos each, and followed by daily feeding and monitoring to recovery and growth up to 10-day post-fertilization. Our last group is folic acid treated group. In a neurodevelopment study, we use folic acid as a standard drug. So 5 mg Folivate tablet we used, and there is various drug doses like 60 ml, 120 ml, 150 ml, 200 ml, and 300 ml were administered to transfected embryos in sterile Petri plate with E3 medium, and monitoring their daily recovery and growth over a period of 10 days post-fertilization. Now there is a part of result. So here is the body length. So the chart illustrated a noticeable increase in body length after transaction of 3-day post-fertilization embryo. However, post-treatment result indicate a decrease in body. Both the test and standard drugs exhibit significant outcomes with a gradual increase in result corresponding to their concentration. Remarkably, the highest concentration of both vitamin B complex and folic acid yield similar positive effects. Then the result for eye diameter. The data chart reveals a slight increase in eye diameter after transition the embryos on 3-day post-fertilization. However, post-treatment result indicate no significant changes attributed to the effects of both the test and standard drugs. Remarkably, the highest concentration of folic acid demonstrate a difference in outcomes. And this result is about neurodevelopment of the zebrafish embryo. So reviewing the data sheet on the slide, the neurodevelopment impact of the test drug is evident.
Sorry for the interruption, ma'am. Please conclude your presentation.
Hello? Hello?
Interruption, ma'am. Please conclude your presentation.
Okay, ma'am. There is a bending angle area. So upon reviewing the data sheet, it is evident that the bending angle on the 3-day post-fertilization before interviewing was considered normal. At higher concentration, folic acid and vitamin B complex demonstrate nearly similar effects, causing a substantial increase in the leading angle. Here is my conclusion and reference. Thank you.
Thank you for your presentation, ma'am. Our session shares. If you have any question regarding the presentation, you can ask the presenter.
It's a good presentation. Have you got CPCSA approval for your project? Hello, am I audible?
Yes, ma'am, you are. Shreya Sarkar, please.
Yeah. Shreya Sarkar.
Shreya Sarkar, are you available?
Hello, ma'am. Hello.
Yes, ma'am, you are audible.
Madam, have you got a CPCSA approval for this project? Hello. Hello.
Shreya Sarkar, are you there?
Okay. Okay, ma'am, ask us to proceed. I just wanted to know this one.
Okay, thank you everyone for your presentation. I will read the names, the one who was not able to present before. Please let me know if you're available. D. Niveta from Government Arts College. Sheetal Rana from Sardar Bhagwan Singh University. Apiksha Rajput from Sardar Bhagwan Singh University, Balwala, Dehradun. Suhaina from Sardar Bhagwan Singh University. Manshi Chauhan from Sardar Bhagwan Singh University. Bharat SI from Chettinad School of Pharmaceutical Science. H. Jasminjit Kaur from KPJ Healthcare University. Mohammad Faiz B. Abghani from KPJ Healthcare University. H. Jasminjit Kaur, KPJ Healthcare University. Please present your paper. Once again, I'll call all the non-participants. D. Niveta, Government Arts College. Please let me know if you're available. Sheetal Rana from Sardar Bhagwan Singh University. Apiksha Rajput, Sardar Bhagwan Singh University, Balwala, Dehradun. Suhaina from Sardar Bhagwan Singh University. Manisha Chauhan, Sardar Bhagwan Singh University. Bharat SI from Chettinad School of Pharmaceutical Science. H. Jasminjit Kaur from KPJ Healthcare University. Please present your paper, sir. Head Jasmine Jit Kaur, KPJ Healthcare University. Please present your paper. Head Jasmine Jit Kaur from KPJ Healthcare University. Please present your poster. Jasmine Jit Kaur, please present your poster.
Yes, thank you. Sorry for the delay. Good evening, everyone. My presentation is on a systemic review on anti-hypertensive effects of olive oil in hypertensive patients and hypertensive treatment. So this is my content. All right. So for the introduction, the systemic review papers will be reviewing on the hypertensive effects in extra virgin olive oil. All right. So hypertension is defined as a systolic blood pressure of more than 140 and diastolic of more than 90. Okay, which is taken at least twice on two separate occasions based on the CPG of Malaysia. Around 1.28 million people aged between 30 to 79 worldwide already has hypertension from low income and also middle income countries. All right. And about in Malaysia, 30% of the populations are hypertensive, whereby about 16% and 14% of them has already known and unknown hypertension, right? And the risk factor involving hypertension includes aging, BMI, and also diabetes. First line therapy for antihypertension is using ACE inhibitor, ARB blockers, CCBs, and then diuretics as well. While whereby non-pharmacological treatment includes having regular physical activities and also weight reduction if they are overweight and some dietary changes. So our focus here is on dietary changes, which involves incorporations of olive oil within the standard treatments of patients with hypertension who are already on their standard treatment. Studies has found that extra virgin olive oil has component that contributes to hypertensive effects, whereby studies has found 80% of oleoic acid and 1 to 3% of minor components which include polyphenol and other nutrition combinations are present in olive oil. So these studies will systemically review effectiveness of olive oil in hypertension treatment and identify the components of antihypertensive effects. So problem statement is most of the patients have hypertension which contribute to the risk of CVD in patients, and only there are few systemic reviews papers which is discussed on the antihypertensive effects of olive oil. So there need to be more papers, there need to be more studies that has systemically reviewed on the effect of extra virgin olive oils for preventions of hypertension. So the objective of my studies is to compare antihypertensive effects of olive oil in any stage of hypertension patients with comparing it with placebo or other supplement given or any diet which does not have an olive oil to perform a systemic review. Right. So we will determine the antihypertensive effect of olive oil according to how much of systemic blood pressure or diastolic blood pressure has been reduced, and also identify the components of olive oil. So this is the PICO setup which is used in the systemic review, whereby our populations would be adult with any stage of hypertension. It should be more than 120/90, and intervention group will be consumptions of olive oil, whereby the comparisons will be placebo and those given any supplements or diet without the components of olive oil, because we need to check the outcomes of olive oil that is provided in antihypertension actions. Okay, so hypothesis is a reductions of blood pressure in hypertension patient consuming olive oil indicates hypertensive effects is present in the olive oil. Okay, so the importance of my studies is that it is able to discover new alternative for the prevention and treatment of hypertension associated with cardiovascular problem. It can encourage pharmaceutical company and further research to be done on the uses and effect of daily consumptions of olive oil for cardiovascular health, and also healthcare professional can practice recommending alternative treatment despite the standard treatment, and pharmacies as well, and create awareness among society of the importance on the management of blood pressure. Right. So the literature review is that studies has expressed on the genes that states there is a risk of hypertension which is not sufficient, and very little salt restriction does help to reduce the blood pressure in severely hypertensive patients, but evidence for sodium intake role in development of essential hypertension is still insufficient. For patients age 50 and above, the gradual change from diastolic blood pressure to systolic commonly happens and indicate isolated systemic blood pressure in the elderly, okay, which can lead to more serious cardiovascular event. Okay, classifications of hypertension we can classify into primary, secondary, and isolated hypertension, and isolated office or white coat hypertension. Okay, the risk factors in this includes unhealthy diets, physical inactivity, using tobacco, consumptions of alcohol, excessively overweight and obese patient. Okay, whereby the non-modifiables are those with elderly age and also who already has got a risk of history of hypertension in their family. This is the risk stratifications for cardiovascular risk chart, and also the tables to denote what are the risk for patients, whether they are in the low risk, intermediate, or high risk. So this is Framingham General Cardiovascular Risk Chart, okay, whereby the components here includes based on the patient's age, gender, total cholesterol, HDL, LDL, smoking status, diabetics, vascular diseases, and antihypertensive medications. Okay. And management of hypertension according to the CPG, the first line includes ACE and ARB, followed by CCB, thiazide diuretics, and also non-pharmacological should be incorporated very significantly into the standard treatment, which includes weight reduction, sodium reduction, alcohol reduction, physical activity, healthy diet, smoking cessation, relaxation therapy, and also diet rich in potassium. Okay, outcomes of the therapy, olive oil benefits. Okay, studies have suggested that increase of OA levels in the membranes will affect the lipid structures of membrane and suppress the G protein mediated signalings, whereby this is associated with the cascade of control of phospholipase C and also adenylyl cyclase that causes blood pressure lowering effect. Whereas polyphenols can prevent chronic degenerative diseases like cardiovascular diseases, because provide protective effect to remove ROS species and modulate the expressions of gene in the signaling transduction pathway. Okay? And another meta-analysis also states that risk of chronic heart disease and stroke in olive oil consumptions, olive oil intake shows significant reductions in risk of strokes for the cardiovascular patients. So the methodology of the study design includes includes a paper of randomized clinical trial. The search engine that was used in these studies includes Google Scholar, Cochrane Library, and also PubMed. Okay, so articles was select based on the search engines that were selected. Only 3 search engines was taken because they have got benefits of a strong search engines. The design, validity, and capabilities of the articles which was collected from those search engines were done using the CASP checklist for the RCTs and also Prisma checklist and flowchart for the systemic review. And the evidence of table was then tabulated based on the articles that was extracted with the help of Microsoft Excel. Right, so these are the 3 search engines which we use: PubMed, Google Scholar, Cochrane Library, and also the MASH term that was used to search the articles includes terms such as hypertension, extra virgin olive oil, blood pressure lowering. Okay, and also we use the Boolean operation terms, for example, and, or, not. Okay, so and was to include both the combinations of studies, or was just to select papers with only olive oil studies, and not was to exclude certain papers which consisting of animal studies. Okay, so the selected papers was then appraised and filtered using CASP checklist, and further evaluations was done for the selected articles based on the inclusion and exclusion criteria. So for our studies of systemic review, the inclusion criteria includes publications for 10 years, for the past 10 years between 2012 to 2022, only English language articles, only RCTs related to the topic of the studies, and also hypertension patients who have any stage of hypertension but age 18 and above. And the article should mention extra virgin olive oil or olive oil as one of their major components, or the studies can also mention Mediterranean diet. Okay, exclusions was animal studies paper, and also patients who already have got comorbidities such as cancer, theochroma, phytoma, and adrenal gland cancers. So quality assessment for all the articles that was collected was done with Prisma 2020 flowchart for to identify, screen, and include the eligibility of the studies. Okay, and then CASP checklist for RCT studies was also used to assess the quality and validity of the articles for RCT. Okay, so data collections. data was extracted using the evidence table. So I'll show you the evidence table, and it was also constructed based on the PICO approach that I have shown earlier. Okay, so the outcome was assessed based on.
Sorry for the interruption, ma'am.
Sorry?
Please conclude your poster.
Okay. So the outcome was assessed based on the evidence table that was constructed for the final papers that was selected, and the evidence table I will be showing you shortly. And then risk of bias was also assessed for all the articles that was collected based on the CASP checklist in order to classify the level of biasness for each paper. All right. So this is the table of evidence. So you can see the components in the table of evidence here. Includes the number of studies, publications, the articles, the study design, various randomized blinded interventions, what are the interventions, and subjects, what are the inclusion, exclusion, the total number of subjects, whether there was any dropouts, what was the interventions, was it olive oil, and then what was the dose of the olive oil, and what was outcomes of the studies based on the control group and also the test group based for the systemic and diastolic blood pressure extracted. So this is the examples of the evidence table. So this is the mesh term that was used, the one I mentioned earlier, hypertension or high blood pressure, olive oil, and this was the three Google Scholars PubMed search engines that was used, and also Cochrane library. Okay, so studies was selected based on these three search engines, and also this was the search terms. So these are the number of articles that was collected. All right. Upon the collection, based on the MESM, Prisma flowchart was created. So based on the Prisma flowchart, the initial number of paper that was collected was 1,317, but later this was also removed because the duplication of studies, 17 studies, and also other papers was excluded, 1,258 due to some exclusion criteria. Okay, upon screening, there was only 42 papers, and 26 papers was excluded from the 42 papers. Okay, due to the CAFP checklist that was not eligible. Okay, so based on the eligibility of full text article assessed, so 6 articles was excluded and only 10 articles was included in our study finally. So the study's characteristics was based on the evidence tables and also the in-text citations and publications and title of articles. So this was assessed based on the evidence of table, the type of studies, the randomizations, the presence of blinded and also locations of studies, and how many subjects was there. Okay, so these are all the components which are already in the evidence of table. Okay, so final one is study outcome, which we analyze based on the baseline BP, and then we compare on the systemic blood pressure and the diastolic blood pressure. All right, so this is the evidence table for all the 10 articles that was created, and these are the main study outcomes for the general study characteristics. Okay, so all studies showed reductions in the systolic and the diastolic blood pressures in the intervention group. Okay, 2 of the studies did not show a reduction in the control group. Okay, whereas 3 studies did not show significant difference between the study groups of a control group and also the outcome group. Okay, whereas 3 studies blood pressure in the control group, and the final one study showed only systolic blood pressure reductions and not the diastolic blood pressure reduction. So this is the risk of bias that was assessed for all the 10 tables. So the examples of biasness that was we included was to test on the selection bias, and also performance bias, detection bias, attributions bias, reporting bias, and also other bias. Okay, so this is the results, and we have classified it based on the low risk, high risk, and unclear. Okay, so this was done based on the CAP checklist. Okay, so discussion, the blood pressure changes based on other studies as well. After 1 year interventions, 24 hours ambulatory BP in the MD group, Mediterranean diet group, showed decrease in the values of the blood pressures from the baseline. Okay. Another study concluded Mediterranean diet supplemented with EVOO has also reduced the blood pressure. For the inflammatory markers, for the intervention groups given Mediterranean diet and EVOO, systolic blood pressure changes was measured after 1 year follow-up, and it showed a reductions of 6 millimeter mercury, and the diastolic was 3 millimeter mercury. Okay, and the reductions was 6% in total cholesterol and 10% in LDL and 9% in HDL. And for the CD40 expressions, the Mediterranean and EVO group showed significant reductions in the T lymphocytes, monocytes, but not in the control group. Okay? So the inflammatory biomarkers assessment shows reductions in the of the ICSM and also in the Mediterranean diet EVO group after 1 year of intervention. Okay? So this showed a significant reductions even in the control group. Okay? And then the CRP values and interleukins values significantly reduced in the Mediterranean diet with the EOO group. Okay. And for the oxidative stress parameter, after 1 year of intervention, there were significant reductions in both systolic and diastolic blood pressure given Mediterranean diet with the EVOO and Mediterranean diet given with the nuts that contains the olive oil components. There was significant increase in also the total polyphenol extractions and plasma nitric oxide that was found in the BP levels. Okay, so the study concluded that Mediterranean diet in the elderly with hypertension can reduce the blood pressure due to the increase in the levels of the plasma nitric oxide. Okay, so we can conclude that the interventions of the EVO in the dietary group has impact on the blood pressure reduction and improves the control and reduce the systolic blood pressures and risk of cardiovascular disease. Okay, high level of monounsaturated fatty acid and polyphenol, and also the low levels of polyunsaturated fatty acid and EVO cause changes in the inflammatory biomarkers and oxidative stress parameter, which is also associated with the overall antihypertensive effects. Okay, so the limitations of this study is that we were unable to conduct any meta-analysis, and also they didn't cover any Asian region. Mostly was papers was collected in the Europe regions, and one was from India as well. Okay, so we recommend that antihypertensive effect studies of EVO in different regions as well to get better and stronger results. That's all. Thank you.
Thank you for your presentation, ma'am. Maitri ma'am, if you have any questions regarding the presentation, you can ask to the presenter. Thank you for your presentation, ma'am.
Thank you.
Mohammed Faiz B. Kahani from KPJ Healthcare University. Please present your poster, sir. Due to the cyclone and power cut issues in our region, we can't able to send the certificates by this week. Next week before Saturday, you will get all the detailed certificates, feedback form, best oral presenter and details, etc. through the mail. Hope you understand our situation. Mohammed Faiz B. Gahani, please present your poster. We'll call upon our next presenter, Apiksha Rajput.
Okay, ma'am. Once.
Ma'am, please make it full screen.
Good afternoon, everyone present over here. My name is Apeksha, and I'm from Sardar Bhagwan Singh University. And today I'm here to present my presentation on the topic Artificial Intelligence in Drug Discovery, its opportunities and challenges. So let's start with the introduction. Artificial intelligence is a wide-ranging branch of computer science concerned with building smart machines capable of performing tasks that typically requires human intelligence. As artificial intelligence is computer-based programs which can reduce a time-consuming process in drug development also. So moving to the part that how is artificial intelligence used in drug discovery. So we can see here it helps in reducing human workload. It helps in accurate decision making also, and it provides quicker validation of the drug target. It also helps in the cost reduction. It helps in achieving the targets in short period of time, and it helps in the optimization of drug structure design. Now we can see the present day use of the artificial intelligence in the drug discovery. So the most recent applications where it is being used nowadays are the drug target identification, the compound screening and design, the prediction of the drug interactions, or we can say drug target interactions, drug protein interactions also, and in the personalized medications, and in optimizing the clinical trials. Here we can see the role of artificial intelligence in the drug discovery. In many fields, it is used, like in the field of drug design. It is used in predicting 3D structure of the target protein, in predicting the drug protein interactions, in determining the drug activity, and in the de novo drug design. If we talk about the roles of artificial intelligence in polypharmacology, then here it is like designing the biospecific drug molecules, designing the multi-target drug molecules. Similarly, in chemical synthesis field, like AI is used in the prediction of the reaction yield. It is also used in the prediction of retrosynthesis pathways, in designing synthetic route, and in developing insights into the reaction mechanisms. The next field where AI is being used nowadays is drug repurposing. There, the use of AI is to identify the therapeutic target, is to predict the new therapeutic use. AI in drug screening, like it is used in the prediction of toxicity, bioactivity, physicochemical properties, and in the identification and classification of the target cell. In this diagram, we can clearly see there is a direct and indirect processes. Like if we do indirect process, like manually, we do the collection of the compound, and then we prepare primary assays like by using high throughput in vitro, and then the secondary assays, which we also try on the animals, like and it is called as preclinical studies. And then SAR analysis, like structure activity relationship. This is an indirect process, like we move to the clinical trials then, but with the help of the artificial intelligence, or by using it, we can do it very clear, very directly, in a very direct way. Like there you can see structural characterization of protein ligand complex. So by the use of the softwares, using the artificial intelligence system, we can do it in a direct way and more quickly. So there are also many softwares nowadays which can be used for doing the preclinical studies also. So this is the example how AI can be used in the preclinical or clinical research also. Here the technologies where the artificial intelligence has been used in machine learning, deep learning, in drug repurposing, in structure-based drug design, in generative models, in predictive analytics, and in pharmacogenomics. But despite of these all advantages, artificial intelligence faces some of the significant challenges also, like the challenge of growth, diversity, and uncertainty of the data. And the second one is the escapism about the data generated by the AI, like the doubts about the data. The third one is that there is lack of skilled personnel to operate the artificial intelligence based platforms in pharmaceutical industries, if we are talking about those industries, and the apprehension of replacing humans leading to job loss. Most of the humans nowadays think that the technology which is known as artificial intelligence can make them jobless, but it's not the thing, as these technologies also require human power or human support to operate. So from all this, we can conclude that the artificial intelligence is making every effort to advance the drug discovery process or system, and it has the potential to completely transform the drug discovery process. Also, these systems are only intended to make work easier and not to completely replace humans. So this all I can tell you. Thank you so much, ma'am.
Thank you for your presentation, ma'am. Our respected session says, if you have any question regarding this poster presentation, you can ask to the presenter.
Yeah, can you just list out some of the software used for preclinical and screening using AI?
Sorry, ma'am.
Can you just mention some of the software in preclinical screening which utilizes AI?
In preclinical studies, ma'am, like there are many softwares which we can use to do that process. Now, like we don't need we don't need more animals to do that. Now softwares can do this, and all of them are based on the artificial intelligence system. So they all are the computer based technologies.
Yeah, that's what I'm asking. Could you name a few?
What, ma'am?
Could you name a few?
A name? Yes, ma'am. XFarm, ma'am. XFarm is a software through which we can perform the practicals. Well, like we can do the preclinical studies without using animals. It can give us accurate results.
Mm-hmm. So
Yes,
some of
ma'am.
the advanced. Oh, thank you.
Thank you for your presentation, ma'am.
Thank you, ma'am.
Let's call upon our next presenter, Mohammed Faiz B. Abdghani from KPJ Healthcare University. Is there any other participant who hasn't presented, give the presentation? No? Our dear session shares, please share us your feedback for today's session.
Ma'am, have you provided any feedback form for the session shares, or because I haven't got it?
Next we'll give, please share us your feedback, like how this presentation. Everyone gave how their presentation.
No, it's a, is it a general feedback or individual presenters feedback you are asking?
General feedback, ma'am. You can provide your general feedback about this conference.
Yeah. So, as far as this session is concerned, all the presenters have presented in a very well manner, and it is a multidisciplinary presentation. So all the delegates had an opportunity to share the knowledge in an interdisciplinary manner.
Good afternoon, ma'am.
Yes, ma'am.
Hello, ma'am.
Yes, ma'am. We can hear you.
Ma'am, actually there is a technical issue, so I'm not able to present that time.
Okay.
Can
Okay.
I present?
You can present, ma'am.
Okay, thank you, ma'am.
Ma'am, your screen is not visible.
1 minute, ma'am. Is it visible now?
No, ma'am, your screen is not visible.
One minute, ma'am.
Is any other presenter there? Please let us know. Otherwise, we'll conclude with this presentation itself.
Good afternoon. Can you hear me? Good afternoon.
Yes, good afternoon, sir.
Yes, Dr. Enrico Magosso. I have a presentation later, so if nobody can present now, I can go ahead with mine.
Sir, one presentation is going on. After completing that, we'll call your name, sir. That time you can present.
Sure. Perfect.
Ma'am, 1 minute, please.
Okay, ma'am.
Is it visible, ma'am? Is it visible, ma'am?
Yes, ma'am. Make it up. Please make it full screen.
Okay, ma'am. Okay, good afternoon to everyone. My name is Suneha from Sardar Bhavan Singh University, and my topic of poster is role of 3D bioprinting. 3D bioprinting in the drug discovery. So start with the introduction. 3D bioprinting is a technology where bioinks mixed with living cell are printed in the 3D to construct natural tissue like 3D dimension structure. Currently, this technology can be used in various research areas such as tissue engineering and new drug development. Also, 3D bioprinting technology and bioprinted structure enable researchers to study functions of the human body uh, in vitro, and 3D bioprinted structure are more biological relevant compared to vitro studies performed in 2D. And now come to the approaches and overview which is presented. First, a preparation which is isolated from the cells and mixed with biomaterial, and 3 bioprinting approaches which are inject based printing, extrusion based printing, and light based printing, which is also called laser based printing, and lastly for this analysis and testing, and also for the drug induced we can analyze drug induced toxicity process work. It is an additive manufacturing process that uses bioinks to print living cells developing structure layer by layer, which imitate the behavior and structure of the normal tissues.
Now the advantages. Advantage of the 3D bioprinting is that it decreases the animal activities, biocompatibility with human cell and tissues. Effect of drugs can be examined more accurately, and it allows mimicking the real structure of desired tissues and organ. It is an automated complex process, and 3D bioprinting has the consistency and less human errors. Now the applications or of 3D bioprinting, or the area in which 3D bioprinting is used. As you can see here, the three component which makes the 3D bioprinting efficient. With first one is organ blueprint, and second one is bioinks, which include the cells plus growth factor plus gels, which is a uh, and third one is 3D printing technology, which comprises into a 3D bioprinting, uh, which which have the applications like regenerative medicine, which will use in the organ transplant and tissue repairs. And second one is in vitro models, like for use for the drug discovery, consumer care industry, disease modeling, and also in food industry, like animal free meat. Uh, in the food industry, it also has the 3D bioprinting also has the application, like it uses the powdered ingredient like sugar, meltable materials like chocolate or cheese, or 3D bioprinting animal free meat is being developed that makes use of either plant-based protein or cultured animal cells. Now the current advances of 3D bioprinting include the cardiovascular tissue. It creates a 3D cardiovascular tissue construct using different proliferated cell types. These tissues construct exhibit macroscopic beating and cell assembly of vessel, like when co-cultured with endothelial cells. And second one is musculoskeletal tissues. It fabricating 3D skeletal muscles construct that are precisely patterned. The bioprinted cell had high viability and responds synchronously to electric pulse. Um, and third one is in the current advances, neuronal tissues. Neurons have been bioprinted with high viability, and they monitor functionality of for over 2 weeks with a successful development of voltage gated potassium and sodium channels. And the fourth one is hepatic tissues. Bioprinted cells were positive for hepatic markers, and they were similar to native hepatocyte as they by albumin secretion and capability of morphological analysis. And last one is skin. Printing full thickness skin equivalent using alternating layer of bioinks and a dermal fibroblast cell suspension that showed high viability after printing. Now the conclusion. 3D bioprinting has the potential uh, to advance the process of drug discovery and development by facilitating the construction of more accurate in vitro models, speeding up the process of drug discovery and enhancing patient outcome through customized medicine. And also we can find the challenges that facing the 3D bioprinting, that need for the like better bioinks that are compatible with human body, and need for more sophisticated printers that can print 3D structure with high precisions. Thank you, ma'am.
Thank you for your presentation, ma'am. Our respected session chair, if you have any question regarding the presentation, you can ask to the presenter. Okay, thank you, ma'am, for your presentation.
Thank you, ma'am.
Now let's move on to our next presenter, Enrico Magasso from University Kuala Lumpur, Royal College of Medicine, Perak.
Very good evening. Oh, evening in Malaysia. Very good afternoon in Dubai. I want to start my presentation. First of all, I would like to thank the organizer and Ramia for his patience. So hopefully he's listening.
So please enlarge your screen.
Yes. I'm trying to actually. Is it? For the work that I want to present today, it is actually a feasibility investigation that we tried about preparation of ketoprofen for gargles, so for local oral delivery. And the important thing was that we tried to have it alcohol free. So we used SMADs. We concentrated our research on SMADs because we know that ketoprofen is a poorly water soluble drug. And we assumed, also from previous experience, that the microemulsification process that we started would have been actually specific. We know the specific and the nature of the oil surfactant ratio that we use, and also the nature of the surfactant themselves, and also the temperature used to. Okay. In our intention for the study was actually that we wanted to formulate this gurgle basically for post-periodontal pain treatment, and therefore for local buccal inflammation. Okay. In particular, we know that the oral mucosa, the buccal mucosa, has a 0.01 average square meter of surface area, and it has moderate and relative enzymatic activity. What is important from our pharmaceutics point of view is there's actually the oral mucosa has a high relative drug absorption capacity. In our study, we actually concentrated on ketoprofen, which is an excellent and quite potent NSAID. And we actually, our challenge was to have a liquid dosage form of a drug that is basically insoluble in water. It's basically water insoluble. In fact, ketoprofen is classified as a class 2 of the BCS. Therefore, it has a low solubility and a high permeability. Excuse me. In fact, the pKa is 4.45. So the aim of our study is actually to is a proof of concept that, of a formulation, and we wanted to obviously characterize and make it also palatable, which is a very important parameter. So basically, it's not just the preparation that we can do, but we know that for pharmaceutical products, it's also very important to, especially if they go in contact with the oral mucosa, they must have a good palatability. Furthermore, we actually concentrated on an alcohol-free system. Ketoprofen is easily soluble in ethanol, but we actually decided to avoid ethanol because there is a first, there is also a culture we are in Dubai now. So, in the use of alcohol for gargle. So it is actually more challenging and also more expensive from the pharmaceutical point of view to produce a gargle, mouth gargle, that it actually has these characteristics. And we wanted to prepare an alcohol-free formulation, which is also a microemulsion. Okay, so we wanted to challenge ourselves. Material, we got it from ketoprofen from China. Gelucir, we use Gelucir as a self-microemulsifying system. Was from Gatte-Fossé, France. And lavender oil, we also use lavender oil, not just for the flavoring, but also because it's a lavender essential oil. It is actually known for having some anti-inflammatory activity per se. So we wanted to add some more activity. First of all, the preparation. We use a melt fusion technique as recommended by Gatte-Fossé, which is the manufacturer. And we actually studied several range of concentration. We went from 1 to 2 to 1 to 6. In reality, we also tried 1 to 1, but was not immediately. We realized it was not feasible as in preparation. As excipients, again, we use lavender essential oil and sodium saccharin. So we also wanted to add some sweetener, which is not a sugar. So we concentrate on sodium saccharin. We also prepare not just the ketoprofen Gelucir ratio 1 to 2 to 1 to 6, but we also prepare blanks, meaning just Gelucir without ketoprofen, and also partial excipients. So for each excipient, we study the combination. Okay, so we prepare ketoprofen and Gelucir without lavender, without sodium saccharin, because we wanted to see any possible interaction. Okay, and they were all prepared in a similar way. We characterize obviously FTIR, Fourier transform infrared. With the particle size, we use even, besides master size and zeta size, we actually use Hooter counter as well, DSC, so differential scanning calorimetry, and we use France diffusion cells. And this one was to study the in vitro permeability test. We use, in fact, membrane that had a similar permeability as the buccal oral mucosa, and we evaluate it through UV spectrophotometry. So the result, I start the result showing how it looked like our preparation. As you can see, it is from 1 to 2 to 1 to 6. And as you can see, it is quite clear. So this was already something more of an interest. So offering a product that it is actually not milky, but but it is transparent. So it has some appeal. We measure the particle size. Okay. Here we discarded the 1 to 2, but we see the 1 to 3 to 1 to 6, because every study that we performed, we actually basically eliminated the range, the concentration of our preparation that was not so suitable in terms of results. So we try to optimize our researches to streamline our research. So in particular, this particle diameters distribution were not very different in the range 1 to 3 to 1 to 6. Okay, so this one was basically, there was around 2 micron. When it came to the hydrodynamic diameter, in fact, we noticed that, so the mean value, we noticed that actually the ratio 1 to 5 was the optimal ratio in terms of formulation. Okay, so we look at it. So this is actually what happened in terms of synthesizer, the distribution of the particle. Okay. And we also did the use spectrophotometer of all, of course. And so we noticed that there were no wavelength shifts, meaning we expect some stability. So we don't expect a major chemical interaction. Okay. And it was actually very, very interesting because although very simple, just UV spectrophotometer, but it showed that actually the 125 and 126 samples had a better spectra in terms of noise. What is very important, and the most important point for us was actually that the permeability study. Usually, permeability study with the trans diffusion cell is done for is mostly for a topical application of semi-solids in particular. So it was a bit of a challenge to try to do it with the already liquid formulation. But for us was important because we wanted to determine not just the amount, but how fast we could deliver, we could pass the membrane, therefore the simulated oral mucosa, with our formulation. And the results were very interesting. We did it in isosaline, water, and phosphate buffer, so pH 7.4. To mimic the oral environment, and we noticed that within 1 minute, was already there was already a sufficient amount of ketoprofen to have an activity. So actually was the aim that what we wanted. So a very fast, because as we know, for a gurgle or for a mouth rinse, actually the amount, the time, the contact time between the gurgle and the oral mucosa is limited to 30 seconds to 1 minute. It's no more than that in practical terms. So we didn't want a product that is it takes 10 minutes to be absorbed. So we wanted something immediately, and we achieved that. So the FTIR, these are the spectra, FTIR spectra. At the end, we selected the 1.5. I give you, this is just a summary, and the 1.5 is the one that gave the best result. And we noticed that this is all the spectra of the different with the different excipients in individual and in the mixture. And we noticed there were not major shifts or appearance of major peaks. It's not very clear from this image, but we went through quite thoroughly, and this actually indicated there was not a chemical interaction, right? So in conclusion, we proved that actually it is possible to have a microemulsion, which is actually we found that it was stable up to 36 months. Okay, so we retested using the particle size in particular, and it was able, our preparation, to permeate the buccal barrier within 1 minute. These are the references. I would like to acknowledge the Center for Drug Research, University of Science Malaysia, School of Pharmaceutical Science. They helped with the equipment, these two centers, and the Overworld program of the University of Pharma, and Mr. Paladin from May, which provided the free of charge the lavender, and the Eurochemo Farm for helping to get the gel seal. Thank you very much.
Thank you for your presentation, sir. Our respected session chairs, if you have any questions regarding the presentation, you can ask to the presenter. Thank you for your presentation, sir.
Thank you very much, and congratulations for the organization. And I hope next year I'll be able to be physically with you guys. Thank you very much.
Thank you, sir. Sheetal Rana from Sardar Bhagwan Singh University, please present your paper. Sheetal Rana, are you available? Thank you for attending second Dubai Pharma Summit and sharing your valuable insights and feedback. We appreciate your contribution and hope you found the event to be a valuable learning experience. And there's an announcement due to the cyclone and power cut issues in our region, we can't able to send you the certificates by this week. Next week before Saturday, you will get all the detailed certificates, feedback form, best oral presenter, details, etc. through mail. Hope you understand the situation. Thanks again, and we hope to see you again at another important events. Thank you, everyone.